Microbiologist of India, AMI, and a professor in the Department of Bio and Nanotechnology at Guru Jambheshwar University of Science and Technology, Hisar, Haryana. With this, I request our chair to please join us on dais. Can we have a huge round of applause for our chair for this session, Professor Namita Singh. Students, once again, I need to mention you, Kohart sir ne kaha tha, taaliyan rukni nahi chahiye. Can we have a huge round of applause for our chair? I welcome you, ma'am, once again to Dev Bhoomi Uttarakhand University. It is the time to introduce the co-chair of the session, Professor Ajay Kumar, a dynamic and a leader of our university of Dev Bhoomi Uttarakhand University, and he's joined us as a Vice Chancellor. Now I request sir to please join us on dais. A Pro-Vice Chancellor, sir, has requested to assist him for the stage. Can we have a huge applause for our Vice Chancellor of the University, Dev Bhoomi Uttarakhand University, Professor Ajay Kumar, who will be a co-chair for this session. I welcome you, sir, on dais. Over to the chair and co-chair. Thank you.
Good morning to all of you. So today, Professor Barry T. Rose with us. I invite Professor Barry T. Rose. Please come on the desk for this side. He will come this side or that side? आप करना चाहेंगे सर? नहीं, please. Let me introduce Professor Barry T. Rose, a truly global authority in field of viral immunology and immunopathology. Dr. Rose is Distinguished Lindsay Young Distinguished Professor of Microbiology at the University of Tennessee, USA, where his work has been defining the course of infectious disease research for over 4 decades. His career is marked by continuous funding from the National Institute of Health, USA, for his pioneer study in herpes simplex virus infection. Dr. Rose's research is critically important for understanding how this pervasive virus causes severe immune-mediated tissue damage, particularly in eyes, leading to conditions like herpes, stromal, which is a major cause of infectious blindness and damage within the nervous system. He is recognized as one of the first scientists to elucidate the complex role of various T cells subtypes, including regulatory T cell, in creating the delicate balance between the effective viral clearance and destructive immunopathology. A prolific scholar, Dr. Rose, has authorized over 400 research papers, placing him among the top 2% of cited scientists worldwide. His profound impact has earned him numerous, including the Alcon Award, for his contributions to vision research and the prestigious 2022 AAM BC Excellence in Research Award. Furthermore, he has shaped the next generation of scientists, having mentored nearly 80 doctoral fellow and graduate students. Out of this, more than 30 Indian scientists trained by him during his course of job. So I welcome Professor Ted Rose.
Thank you very much. Oh, it's working, yeah. Thank you, Namita. She makes me sound like a god, but I'm not. I'm just an ordinary person. And one of the great pleasures of my life has been that I've been to India many times. And what's more important than that, I've had at least 20 or 30 trainees from India in my lab, many of whom are here in India working as full professors running research programs. And so it's a great pleasure to be here to talk. This topic I'm going to talk about, however, may not be all of yours cup of tea, because I see that the strain of microbiology you're more interested in is making new ways of breaking down the environmental contaminants, etc., which is very important. But I, of course, work in medical disease, and so I'm interested particularly in dealing with virus infections. Now, all of you, this is not moving on. Okay. These young men were here for 20 minutes. I thought they had fixed it.
Now this changed.
Okay, is it working? Have you? Oh, yeah. Okay. We all became virologists. Everybody in the world became a virologist because of the outbreak of this problem, COVID-19. And so many people have become expert virologists that they know how to deal with these infections. And so many people have all of a sudden become quite concerned about using vaccines against viruses. I don't know if there's any problem here in India, but certainly in America, a lot of children are not receiving childhood vaccines. And this is going to cause us a great problem in the future. Now, vaccines are not perfect, but they are by far the cheapest and most efficient way of dealing with any infection of any kind if you're able to make an effective vaccine. But immunity is sometimes a problem. And I'm going to talk about, I need this. Okay. I'm going to talk about infections where immunity to the virus actually is the cause of the problem. And then I'm going to try to tell you how we can perhaps deal with that situation. All right, but I'm having no. So just by the side, whenever you're infected with an infectious disease, not everybody gets symptoms and so forth. Most people do not, and so one doesn't worry too much about it. So the root cause of disease in the unhappy few who get severe disease is something we're really looking into seriously, because we don't know why some people get severe COVID, for example, and others do not. Some people, when they recover from the infection, still suffer signs and symptoms for a long time after. We don't understand why that is, but one day we will find out. So let's talk about viruses. How do viruses cause disease? Well, there are some agents which we describe as intrinsically virulent. That means they have the wherewithal to be able to encode events which are directly damaging to the host and may actually kill the host. And the best example of this in human viral infections was, of course, smallpox, which has been now eliminated from our society. But there's a whole bunch of other viruses which can become causes of infection and disease if your immune system is damaged in some way, either because you're infected with another agent, such as HIV, or if you are genetically predisposed. And so if your immune system is not working fully effectively, a whole bunch of other viral agents can cause disease. And the final cause of the problem is that the virus itself doesn't cause the lesions, but instead it is the response that you make to the infection that is the cause of the pathology. We refer to those as immunopathologies, and here's a list of some of these which occur in you. Severe symptoms of COVID, when they happened in the respiratory tract, for example, were not caused by the virus itself. They were caused by the immune response to the virus infection. What's happening? Okay. Oh. That will make me louder. I don't want to be. Turn it down. I must. Ooh. No, it's terrible. Let's go back to where we. Can I get it quieter? Okay. I must say, life in India is full of excitement because you have actually no clue of what's going to happen next. So I'll try to continue. This is a list of infections which affect you and me, and they're caused by us generating a reaction against those infections. And most notably in India is dengue shock syndrome, which is a major problem in India. And that's caused by the host making inflammatory cytokines to the virus. And I know a lot of people are trying to solve the problem of that disease. But there's several other infections, and some of them are listed there. And I'm running out of time, I guess. Okay, but there's good news. Whenever there's a host response to a virus infection, it's only some components of the host reaction system that are causing the lesions. Going on at the same time, one has a list of other activities which will downregulate that reaction. And so we have two types of things going on, good things and bad things. And so to control this scenario, all we have to do is to find ways to rebalance the action of these different components. The question is, how do we do that? How do we accomplish that task? All right, well, here then is the balance, if you like, where we have the good guys on the right side and the bad guys on the left side. And since you're not immunologists, most of you, I can tell you that we immunologists can recognize a bunch of different activities which, when they are functioning, cause tissue damage and cause lesions, damage and sometimes death. On the right side, we have another list of characters which are reacting, and if they dominate the scene, there will be no lesions to observe. And so the task then is to try to emphasize the activity of those events on the right and suppress those on the left. So there's a whole bunch of strategies to do this. And in fact, one of my Indian colleagues, now an assistant professor in Texas, and I wrote a review about that. And this review was published a little while back now, and the number is there if any of you are intrigued enough to read it. It talked about rebalancing immune activities to control viral immunopathology. So we'll just very briefly go through some of those. So the problem there is in the middle, viral immunoinflammatory lesions, and we want to get rid of things which are increasing that activity. And we want to get rid of those. So one thing we can do is remove the inflammatory cells themselves, which are responsible for causing these effects. We're able to do that. We can block the activity of those inflammatory cells in a number of ways. We can also restore the function of cells which had lost their useful activity, and this is a valuable line of approach. It's called immune exhaustion, and we're actively using the reversal of immune exhaustion to control certain cancers. And one of your native fellows, Rafi Ahmed, is one of the leaders of this field, and many have made the case that Rafi Ahmed perhaps should have shared in the Nobel Prize, which was given to other people who were also working on this subject. So you can also expand the activity of cells which we call regulatory cells, and I shall make a little few mention of that. And this year, I should say, regulatory T cells was the topic of the Nobel Prize in Medicine. And then finally, we can eliminate other things which are causing problems. So there's a variety of ways to manipulate the immune response, and we have not enough time to go into each and any one of those, but if anyone wants to grab me afterwards, I'm happy to expand the story with you personally. All right, so what we have done is focus on the cells which principally organize the inflammatory reaction. And these are cells called T cells, and they're pro-inflammatory T cells. And sometimes those pro-inflammatory protective T cells become exhausted, as I mentioned, and one have used that. And I have a colleague there who's currently in Pune, who she and I have written a review about this subject. Okay, so here then, with this exhaustion thing, on the left we have the exhausted cell, and it's exhausted because it's upregulated molecules which the host has mediated the response of that cell. And what you can do is get monoclonal antibodies which engage those exhaustion molecules and use those for treatment, and one can get the cells fully restored to function. That's a very expensive thing to do, and in fact, those of you who are looking for alternative forms of medicine, it would be very useful if you could find reagents which could reverse exhaustion, which are far cheaper than monoclonal antibodies to use for that purpose. And I know many of you young students are trying to find alternative ways to manipulate things like that. Okay, I mentioned this business of regulatory T cells. Well, we were some of the pioneers in this area, actually. And one of my students, Susmit Sivas, who was a native of Ljubljana, he came into my lab and he decided that we should look at cells which are regulatory. He did some nice experiments, and as a consequence, we had the good fortune to be the first people who ever wrote a paper about the role of regulatory T cells in a virus infection. I wrote a review about that with another pioneer, Yasmin Belkade, who currently is the director of the Pasteur Institute in Paris, and that was a very highly cited paper. So regulatory T cells are valuable. This year they got the Nobel Prize. We didn't merit it, but some people did. And so the topic is of relevance. Now, the issue is, can we control inflammatory responses by manipulating regulatory T cells? And the answer to that actually is yes, with qualification. But we work on this disease, which is hepatic stromal keratitis, which is a lesion which occurs in your eye if you have the misfortune of being infected with herpes simplex virus. Now, when you're infected with herpes, it's normally a superficial infection of the skin, the eye, the genital mucosa, or something like that. But after herpes infects, the virus hangs out indefinitely in your body, in the local nerve ganglion. And it's not content to stay there, because periodically it reactivates and comes back into the lesion, into the site where the virus infected first, and causes a lesion. And if that happens in the eye, it can cause that eye to lose its function. All right? And so, again, with one of my very talented Indian students, this time Sharan Sharawat, who's at the Mahali Institute of Technical Research, where he's a full professor now, we wrote a review about how you can manipulate regulatory T cells. And there's a whole bunch of ways of doing that, and we won't go through those today. But it's a very important topic to bear in mind, because there are so many potential ways that you can minimize problems with autoimmunity, certain cancers, and chronic viral infections if you could find ways of successfully manipulating regulatory T cells. I won't go through those various things, but you can read the review, and I put the number down there. If anyone wants my slides, by the way, after I finish, you're welcome to have them. Okay, this was the critical experiment that Susmit Sivas did, where we studied this disease in a mouse and caused it to develop that lesion I showed you like the humans had. And what Susmit showed was if you removed regulatory T cells from the mouse, the lesions got much worse. And so the picture on the right is showing a far more severe lesion. Other students of mine, in fact, I had a Spanish PhD student that did this, showed that if you add extra Tregs to the system, you can stop the lesion from occurring. All right? And so what I really am into now, though, was started in my lab by yet another Indian national. This gentleman's name is Kartik Varanasi, and he is he comes from Hyderabad in southern India, and he was a brilliant guy who came into my lab not knowing anything about immunology or virology, but he knew he was a very good biochemist. And so he persuaded us to forget everything we were doing and to start looking at a biochemical approach to these inflammatory lesions. And so Kartik did a very important experiment by showing that if he affected the activity of glycolysis in a mouse which was infected with our virus, then the outcome of the lesion was quite different than animals where we did not influence glycolysis. All right? And so what he did was, and I'm going to forget the first part there, that what we really need to do, by the way, is when the patient comes into your office, they have the problem, and we want to be able to remove the problem. And we can do that by manipulating metabolism sometimes. There are a few drugs, like rapamycin, for example, which a young lady in this, she's not so young now, actually, in the audience here, did some experiments in my lab long ago on rapamycin. And now she's the only gray-haired lady in the whole place. And so we're a twin. Shilpa Deshpande, who was also one of my students, did some work a long time ago. We should have followed up on it. Is an excellent way of turning off an inflammatory response with rapamycin. But there's rapamycin mentioned, and there are other ways of doing it as well. And that's the data. I won't show you that. So what we have done with Kartik Varanasi was realize that the two main combatants in this saga, namely the pro-inflammatory cells which are causing the damage, and the regulatory T cells which are alleviating the damage, they actually have different ways of generating their energy and all kinds of other metabolic processes. For example, the regulatory T cells use fatty acids and glucose. They use oxidative phosphorylation to generate energy, whereas the pro-inflammatory cells primarily rely on glycolysis and fatty acid synthesis. And so what Kartik did was he infected mice in the eye with herpes simplex virus, and then a few days later he treated them with 2-deoxyglucose, which is a drug which will prevent glycolysis from happening. And this led to a marked suppression of the expression of these lesions. And if you looked at the local lymph nodes of those mice, you could show that the amount of regulatory T cells in relation to the inflammatory T cells was changed. As a consequence of 2-deoxyglucose levels, the representation of Tregs increased, and these suppressed the inflammatory reaction. However, going on to study this further, we were able to show that if you treated mice right early on with 2-deoxyglucose, the mice indeed did not develop the lesions we were worried about, but they did something else. They died. And that's what's called the doctor's dilemma. When you cure disease, you don't want to cure disease by causing the animal or person to die. But this is what happens if you use 2-deoxyglucose too early. And the reason for that, we now know, was because when you treat straight away with 2-deoxyglucose, there is no protection in the local nerve ganglion against the virus. So when the virus goes to the local nerve ganglion, it normally stays there, and it stays there in part because there's an inflammatory reaction dominated by these cells, which in another location, the eye, cause damage. But in the trigeminal ganglion, for example, those cells are valuable and they prevent spread. And so this slide here shows you that the virus is not controlled fully, continues to replicate. And when that happens, the virus goes up to the central nervous system, enters into the brain, and causes herpes encephalitis. And that sometimes happens in humans. Herpes is a common cause of encephalitis, but is still a very rare manifestation in humans. All right, so we had to look for some other way of doing this. And there's a wonderful review written by a gentleman called Luke O'Neill, who reviewed the various ways available for you to manipulate metabolism. And here's a list. We've only looked at glycolysis and oxidative phosphorylation, but there's a lot of other things you could look at, and other people hopefully are further investigating those things. One good thing is that we looked at metformin as a way of achieving this, and metformin is probably being used by several people in this country because it's one of the favorite therapies for diabetes, and many of you have or will develop diabetes, and you might receive metformin as a form of treatment. Well, it turns out metformin, which affects oxidative phosphorylation, if you use metformin to do this treatment, then again we get the good effects and we don't get the bad effects. So in other words, the animals survive very well. And the reason for that is because if we look at the expression of these inflammatory T cells in the ganglion, where I told you it's necessary to stop the virus from going to the brain, you'll find that whereas 2-deoxyglucose knocks out those cells, as shown on the right columns of those graphs, when you use metformin, the function and representation of those cells is fully preserved. And so we get the good effects and not the bad effects. So the conclusion is, if you're going to use metabolic reprogramming, you have to choose the right drugs to use for the occasion you're dealing with. I do believe, however, it is a very valuable approach which needs to be further explored in many clinical type situations. And is there a future? Of course there's a future, and you young people in the audience here are, of course, the future. So if you want to do something where you may yield fruit and make some nice discoveries, you should get involved in studying how to reprogram metabolic events. By the way, long COVID, which is this disease I mentioned earlier, probably is a problem with metabolism. And in fact, metformin is used to treat some patients with long COVID. All right, so that's the end. I don't know how much time I've taken. I've jumped some slides, but I have a very valuable group of people who did all the work. Khaled Varanasi, I've mentioned. Sid Baylor, I didn't mention. Sachin Mukha, Ujjwal, God. Look at all these Indians I had in my lab. How I could not have survived in this world of science without all of my Indian students and postdocs. I did have people from other countries as well, but thank you very much for your attendance, and thank you for providing me with these people.
Thank you, Professor Ross. Now, house is open for question. House is open for question.
Any questions
if you
from
have
the
any
audience?
questions, you can.
Any questions from the audience, from the students? You may raise their hands. The mics will be coming to you. Okay, ma'am has a question.
There's a lady over here.
Hello.
Excellent talk, I must say. And my question is that 2DG you used for eye treatment for hepatitis, right? Encephalitis.
Yeah.
And I know that during COVID time, 2DG was used for the treatment of COVID patients. So is there any evidence that 2DG is being used for other virus infections?
I'm not a clinician, and I'm not aware of anybody, but it could be the case. People use weird stuff to treat things at times, and it wouldn't be logical to use it because of the danger which I talked about. So,
Any other questions anyone has from the audience? Okay.
Well, the talk was really very interesting and gave you ways and means by which, in fact, you can fiddle with the pathways also, which...
sorry, I can't really hear what you're saying.
The talk was really very interesting, in fact, because it gave you ways and means by which you can fiddle with the virus replication and the implications which are more pathological. So my question is primarily that the intermittent fasting, does it also affect basically as far as the progression and effector functions of that virus molecules or those proteins are concerned? How it works? Because it will basically manipulate your glucose concentration.
I don't know the answer to that question, but there is diet does very much influence the balance of the type of T cells you've used. In fact, another one of my Indian trainees, Dr. Sombria, Deepak Sombria, studied the effects of pre-treating a mouse with sodium propionate, which serves to adjust the types of T cells that the inflammatory response uses. And that, these fatty acid, I forget what type of fatty acid it is, but it's fatty acid, and it works beautiful to minimize these inflammatory reactions. So I think diet is very important. In fact, certain types of fats are anti-inflammatory. And so there's a lot of mileage to be made. In fact, many of these weird compounds which ancient medicine has used, and I know curcumin is very popular in this country, it actually does have a pharmacological effect, which is quite valuable. So I think that's a very viable approach to look at this topic. Okay, is that it?
All right. Now request our chair and co-chair, Professor Namita Singh, the General Secretary at AMI, and Professor Ajay Kumar, the Vice Chancellor at Dev Bhoomi University, to kindly share a token of respect and gratitude to our plenary speaker, Dr. Barry T. Roos. Can we have 100 applause for our plenary speaker who has delivered beautiful sessions to us? I request chair and our co-chair to kindly share a token of respect and gratitude to our speaker. Still as the voice of the clap is so less. Can we have a huge applause?
Hello. Hello.
Thank you, sir. Now I request the President and the Chairman of ASMC, Professor R.C. Kohart, sir, to please join us on dais, please. I request Kohart, sir, to kindly share a token of gratitude to our chair, Professor Namita Singh, the General Secretary of AMI. Sir, I request you to share a token of respect and gratitude to our chair, Professor Namita Singh, the General Secretary at AMI. Now I request sir to kindly share a token of respect and gratitude to our co-chair, Professor Ajay Kumar, the Vice Chancellor at Dev Bhoomi Uttarakhand University. Can we have 100 applause? Thank you, sir. Thank you, sir and ma'am, for being the chair and co-chair for this session. Now, there's an announcement regarding the sessions that we have after the high tea. The session number 5, the metabolic and molecular frontiers in microbial biotechnology, will be happening at Cinema Hall, first floor in the main building. Session 6, the microbes for the restoration and degraded environment would be powered by high media, would be at the main pandal. Session 7, India Ideas Brew Science Over Coffee, would be at the Seminar Hall, Hotel Management Building. And I request all the dignitaries, all the participants kindly proceed for the high tea. We'll be meeting back once again after 20 minutes here for the next session. We'll be here by 11:45, so I request all the people who will be the part of session 6, microbes for the restoration of degraded environment powered by high media, kindly reach the main pandal by 11:45. Thank you.
कौन सी applicability? अच्छा, you don't have the
In next 2 minutes, we are starting with the session 6 here. So I request all the participant delegates to please join us as soon as possible. If you have your friends and family who has been staying out, kindly please call them to be here. In next 2 minutes, we are starting with the session 6 at main pandal. The volunteers at the back, make sure there should be no noise during the sessions. Kindly maintain the decorum during the sessions. The volunteers at the back side, make sure this thing happens. Thank you. Ma'am, shall we proceed? Thank you. So I welcome you once again for this day 3 of this conference, and we are starting with the session 6, Microbes for the Restoration of Degraded Environment, which is powered by High Media. This session focuses on the vital role of microorganisms in environmental restoration and sustainability. Experts discuss how the diverse microbial communities contribute to the biomedication of the polluted ecosystem. Includes soil, water, and industrial waste environments. The discussion highlights the latest innovations in microbial technology, emphasizes their use in degrading pollutants, restoring ecological balance, and supporting circular bioeconomy models. It aims to inspire researchers, students, and professionals to harness the power of microbes for a cleaner, greener, and more sustainable future. It is a time to introduce the chair for this session. Professor Athya Kapile is a chief scientist and the head of the Environment, Biotechnology and Genomics Division in CSIR, NIRI, Nagpur. She holds a PhD in life sciences from the University of Hyderabad as an expert in environmental microbiology genomics. Her pioneering research focuses on microbial community analysis, wastewater treatment, and restoration of polluted ecosystem through biotechnology. She is actively beyond research. She actively serves as a Vice President, Asia and Pacific of the Organization for the Women's Sciences and for Developing World, OWSD, and actively promotes women participation in the science and innovation. With this, I request our chair to please join us on dais. Can we have a huge applause for the session of the chair, Dr. Athya Kapile. I welcome you, ma'am, to Dev Bhoomi Uttarakhand University once again. Now it is a time to introduce the co-chair of the session, Professor Prashant. Professor Prashant is a distinguished professor at the Department of Biosciences and Bioengineering at IIT Mumbai. He holds a PhD in microbiology from the Indian Institute of Science, Bangalore, and completed postdoctoral research at USA and Switzerland. He has published extensively in high-impact journals, guided numerous PhD students, and contributed significantly to microbial biotechnological research in India. With this, I request our co-chair, Professor Prashant, to please join us on the dais. I welcome you, sir, to Dev Bhoomi Uttarakhand University. With this, we are starting with the first keynote session by our eminent speaker, Professor Athya Kapile. Over to you, ma'am.
Hello. Hello. Hello.
So I just, yeah, that's the one. Which one I use? Up and down? Well, does this work? Hello? I think this is good enough, right? I can use both.
If you wanted to use this one, these are slide up and slide down.
Okay.
Yeah.
That's going. Good afternoon, ladies and gentlemen. Thank you to the organizers for inviting me to share my research findings here. And a special thank you to Dr. Nirjara and Dr. Nabil for being such wonderful hosts. Since yesterday, you have all been hearing about how microbes rule the world, how they are involved in every aspect of our lives. So I worked for 30 years in the field of environment remediation. And so I would like to walk you through what we have done in the past few years regarding how the microbes influence the environment and how they basically look after the environment. Okay. So there are a number of groups that are basically focused on when you talk about microbes in the environment. They drive the biogeochemical cycles. All the nutrient cycles that are happening in nature, in the oceans, in the land, that's all because of the microbes and their actions. The carbon, nitrogen, phosphorus, sulfur, all these cycles are basically mediated through the microbes. Biodegradation, yes, of course. Biodegradation is a very important and a huge branch of microbiology, and microbes are responsible for keeping the environment clean. The only bottleneck here is that the load that we are putting in is so much that the microbes also need a little more resistance to clean up the mess that we have created. So we worked with, pesticides, we worked with different organic, industrial waste, we worked with industrial effluents, chemical ETPs, and of course emerging contaminants. And then there are the decomposers, who are also equally important. There is a whole group of microbiology that is focused on studying the functional diversity across the ecosystems, how the populations are maintained. So this is something that we have done. So we've looked at, we focus mainly on bacteria and the different ways in which they can carry out biodegradation. So we did metagenomics. We have applied the tool of metagenomics in many of our studies. This is just one of the simplest biodegradation pathways of phenol. So we looked at petrochemical refinery treatment plant, and we basically did a comparison of the different treatment processes within that. And I'm just showing you one example of how metagenomics helps us to identify the entire pathway. We have monitored each single arrow that you can see in the conversion. Each is responsible by some particular gene. We can even look at the abundance of the number of genes and decide which is the pathway that is actually being operated at the environment niche. And so whether we need to do bioaugmentation with the other pathways or whether we need to assist to get this pathway moving. So this is one way. Alternatively, we have looked at the taxonomy of the different microbes and associated those with the degradative potentials of different hydrocarbon groups. In the, the hydrocarbons actually comprise of more than 100 different compounds. So which microbes can interact with which? This is a study we did for an CTP, that's a common effluent treatment plant. For those of you who are not aware what a CTP is, so it's basically the government of India had set up this very new concept that wherever you have small scale industries like pharma, textiles, chemicals, so they do not have the funds to create their own ETPs. So there is a centralized facility called the Common Effluent Treatment Plant where all these industries send their wastewater to be treated. The complexity with biodegradation at a CTP is that today you are having wastewaters from say textile and tomorrow you are going to be having wastewater from pharma. So the array of compounds that are going into your treatment unit, the activated sludge that basically contains all these microbes for biodegradation, they have different compounds that they will be treating every day. And so in a CTP, the degradative efficiency is always compromised. So what we did was that this is like I said in Hyderabad, and it gets mainly from chemical and pharma. So we did the metagenomics of the activated sludge, and all these, can you see this? No, it doesn't matter. We wanted to look at what are the degradative pathways that are operating within this treatment plant. All the dotted arrows that you can see are the genes that are present in that ETP, and the bold lines are the ones that are not. So if you look at say aniline is not present, aniline dioxygenase is not there, and if your wastewater contains aniline, it's going to pass out undegraded. So what we do here is we have done this study for them and then got the necessary knowledge for bioremediation. So when you do bioremediation of one particular substrate, then it's easy because you're dealing with only one particular compound. But in an industry, it doesn't work like that. You have hundreds of compounds that need to be degraded before they are released into the environment. So the tool of metagenomics really helps us to understand what pathways are already present, and so we make an informed biodegradation, bioaugmentation process. So using classical microbiology and the genomic side, we decide whether The environmental niche needs biostimulation. Sometimes the genes are present, but because of other different stress factors or because of low carbon, excess nitrogen, they are not able to actually carry out the function that they are meant to. They are dormant. So we do biostimulation to activate them, to get them go into the degradative pathway, into the catabolic, activate the catabolic pathways. Or when something is missing, we find, then we suggest bioaugmentation. We have successfully done this in at least 5-6 different treatment plants across the country. But today I want to little bit focus on the ecosystem services. What the bacteria do actually in the ecosystem, in our natural resources, because as you know, India is blessed with abundant water. We have enough of rivers, lakes, every state and city, but sadly they are all now so polluted that we, despite being blessed with such an abundant natural reserve, we are in a water crisis state today. So we thought that we would look at what the bacteria help us to do in, say, in the river ecosystem. So this is the example of a case study that I am going to show you about the Ganga river. And we have studied the biogeochemical cycles that are happening in the river, the presence of contaminants, that physicochemical analysis, pathogens in the river, the antimicrobial resistant genes that are there, and we have compared across different right from Gangotri to Diamond Harbour. We had 22 different sampling locations. It was actually a huge team. I cannot take the credit for that. I just happened to be the project leader, and NEERI was the coordinating institute, but it was a real huge team. So we had Professor Datta Madamwar from, actually it says Motilal Nehru MNNIT was Dr. Shivesh, who's based at Prayagraj. Charotar University is Dr. Datta Madamwar. FixGen is Dr. Rooplal and his team. And we had tied up with this lab as our sequencing partners, accelerators. So 5 different institutes combined, and it was a huge team of more than 50 people. One of them, Abhay, is also sitting right here. And Dr. Prashant was one of the reviewers of this project. So I think we've just integrated so many people into this huge program. And it really was a very huge program. Sampling from Gangotri to Diamond Harbour, trying to cover the entire stretch of the river, 22 locations of sediment, water, and the biofilms. So this is just some pictures. We collected the samples from all these places. And then we did, of course, NEERI is famous for that. So the physicochemical analysis, what's the BOD, COD, what is the, we compared. So the thing was, when this project took off, it was just COVID time. So if you look at the first half, the BOD is really very low. So it shows that, today if you go and take the BOD in these same locations, it's going to be much higher. But at that time, when we were just within the COVID period and coming out of it, it was like, the human activity, the anthropogenic activity was at a minimum. Of course, the sewage from all the nearby villages and cities is still flowing down. But the floating population that comes to the river, which also is a very big major contributor to river pollution, that was all gayab. So there we could see, a good part of, it's also, I always said that it's also a very good example of how humans are actually polluting the rivers. When you take during the COVID and post-COVID, you can really see a big difference. We looked at the dissolved oxygen levels across, because that is actually what maintains the health of the river. And the good thing was that we could correlate our physicochemical findings with what taxonomy we could get from the metagenomics, studies. Like, wherever you have a little high DO, you have a high presence of firmicutes. These are gut microbiota, and you know that they are producing a lot of oxygen. So we had at some places very high DO levels. So we could correlate our physicochemical with, the bacterial diversity. And then one of the, the growing health risk is, of course, antibiotic resistance. I don't think there would be anybody, seated in the hall who would not have even one relative who has not faced this themselves or somebody in your family. So many drugs are now, not acting on so many people, especially, hospitals. So, from being a boon to being a bane, you've got to even study that. Although, as I say this, I do not take away the importance of antibiotic treatment. It's still one of the very integral parts of keeping us healthy. But this is an emerging health risk. And so we thought of looking at it across the river. So we've done a bit of study in across different scenarios for antibiotic resistance. So across the Ganga, we have looked at the estimated health risks because of this. So we looked at 2 different gene banks for the antibiotic resistance. You can see both overlap very properly. We looked at 16 different classes, and we find they are majorly all present across the river, even in Gangotri, where you expect that you would have minimum kind of a thing. We also looked at different pathogens. Other than the escape pathogens, we looked at the presence. So you can see this is the abundance. So there are some which are low, some which are very high. We did this project for the NMCG, and their main interest was, the presence of E. coli. So the thing is that when you're monitoring E. coli as a parameter for surface water, so when you are looking at it, say in a swimming pool, then that's a different scenario. You're looking whether it is safe or not. But when you look in the environment, like into a river, then you have, it's not just because of human population. You have cattle there, the runoffs are there, you have poultry runoff. So all these kind of things also contribute to the number of E. coli, and one of the major tasks that was done was to look at whether the E. coli is from human or animal origin. So if you just look at the presence of E. coli, so if you look at the, oh, it hasn't moved. Good thing I checked. It got stuck. So no, it's moving here, but it's not moving there. It's moving here, but it's not moving there. Okay, take care. Here. Which one? Here, no? I prefer this. Where are we? Yeah. So if you just look at the O2U numbers, then you can see, the number of bacteria versus E. coli. So the actual percentage of E. coli within river systems is really very low. So the alarm that we raise about, E. coli infection is, I think, a little bit to be diluted. And what one of our team members did was, he got sequences of E. coli from different origins, and he developed a methodology, to understand whether the sequences that we got, whether they are, coming from, they are homologous to the control sequences that we have used. So he has taken from different cattle hosts. We have looked at pathogen 0015 semen, and we looked at lab strain E. coli, and also, different humans and a wild type. So this way we have been able to categorize. Now this is not a, whole complete study. The more number of sequences you take as control, you will get a broader definition. You could even use here poultry, you could use, from poultry origin, you could use from different birds, so there are a whole lot of sequences that you could use. So just to demonstrate that this could be done, we had just, taken from these 5. And then, like I said in the beginning, we want to look at different, the cycles, the nitrogen cycle, carbon cycle, sulfur cycle. And so at each location, we have looked at, this is the nitrogen cycle. So you know that mostly from sewage, your nitrogen phosphorus levels are always very high. This is what actually causes eutrophication in the lakes. But a river rejuvenates itself, and so it has a more healthy balance to dealing with this kind of a sewage problem. So at each step, we looked at what are the different genes for nitrogen. And you can see the rightmost side. I don't this. I'm not able to. Oh yeah. Is it there? Anyhow, you can see the ammonification ones. So that is at Howrah. So you can see how contaminated it is, because those are genes that represent the anaerobic cycle, the anoxic cycle for nitrogen. So in a river, you don't really expect to see them, but you can see that they are strongly present at Howrah. So it also indicates how much of level of pollution is there at different things. So I will just end by, with this slide, that bacteria, they don't just actually respond to the ecosystems. They are what is actually controlling. All the health of the river is basically controlled by the bacteria in them. The biodegradation of organics that are coming into the river is controlled by the bacteria. So if we want to really develop methods to clean our rivers and lakes, we need to actually look at decentralized options to treat the incoming waste, through microbiological and biological routes. Thank you.
Can we have a huge applause for our eminent speaker sharing a beautiful presentation on how the rivers can be revamped up. Now I request Mr. Sunny Verma, the controller of the examination, to please join us on dais. Sir, I request you to kindly share a token of respect and gratitude to our keynote speaker of today's session. My dear audience, can we have a huge applause for this beautiful session by our chair and a keynote session by Professor Kapil. Thank you, sir. Thank you, ma'am. Now it is a time to listen from the another eminent speaker of today's as a keynote session by Professor Prashant. Over to you, sir.
Okay, I think I'll try it from here. Okay, if I require, then only I'll go for it. Just hold on for a second. Okay. try with this. If it doesn't work, then I'll come to it. Okay, hold on. Okay. No, leave it. That's my day. Yes. Yeah. Good. So, good afternoon, everybody. Well, I'm Dr. Prashant from IIT Mumbai, okay, Department of Bioscience, Bioengineering, working for last 25 years in the main theme of bioremediation, keeping environmental microbiology as one of the important focus of the lab. So what I'm going to do today is talk about one of the new species which we have identified from Pseudomonas genus based on the genome analysis, further by genome annotation, and based on its very novel, unique ecophysiological properties. So the name of the strain which we have given it as a Pseudomonas bharatica, that is based on the soil which we have used it for the isolation. So the strain was isolated from the soil which was contaminated with the petroleum products from Bangalore. So when I was doing my PhD, one of the senior isolated this. So the strain was that time identified as a Pseudomonas putida. And then it was basically labeled as CSV, and 86 is the year of isolation. Recently, when we went for its detailed genome analysis based on the genome analysis, annotation, various other properties it has, we identified it as a bharatica, giving the thing back to the country with its own name. So now we have a new species, and since it is written as a CSV86T, that is the type strain. So this is the first time reported in the literature, and it's now basically accepted by the Burgess manual to be listed into it. So I'll go ahead talking the detailed properties of CSV86. I'm not going to divert anything anywhere, but I would like to thank Chair Atya for giving a very good brief description about the environment kind of system present over there. So I'm going to skip my few introductory slides and go straight into the CSV86 for your purpose, and also it will save some time for me and go into detail of some of the experimental work. So now also I'm trying to target the audience, the one which are working into the research labs. Okay, so I'm going to not only going to talk about the overall conclusions of the experiment, but I'll try to give you some basic hints about how those experiments were conducted by not using heavy equipments, but just by using a simple microbiological techniques. So I'm going to be a very classical microbiologist, including biochemist and all omic aspects into it. So well, my lab is very well set for the microbiology, biochemistry, and what we do is basically isolate the microbes by using an enrichment culture technique by using a single carbon as a carbon source. So that could be any carbon source. It could be a naphthalene, methyl naphthalene, or any organic solvent as a carbon source, and get it done. We are looking for the cultures which are very fast, which can degrade all these aromatic compounds as a source of carbon within 12 to 14 hours, not more than that. We don't have time to wait for weeks. We have very short time, because that's today's industrial need in terms of the wastewater treatment or land remediation. So our main goal is to get the very efficient microbes which can degrade these compounds at a very fast rate and at a very high concentration. So the strain which I'm going to talk, Pseudomonas bharatica, is the one which can degrade naphthalene as high as 5% of naphthalene weight by volume into, that means 5 grams in 100 ml. And that is as a solid naphthalene, which can be utilized by this strain. So by using the various topics or the domains available in the life science, that is biochemistry, microbiology, molecular biology, omics, now we are into the biosynthetic or the synthetic approach, which I'll be coming towards the end as application. So I'll skip some of this introduction, which was given very nicely by Atya. I will not spend time. These are the effects which we know it, okay, which is taking place in the environment. And keep it in mind, it's not only the animals which are getting affected, we are also getting affected. To give you one of the best example is all the bottled water what we have it has lot of contamination of phthalate isomers. And these are very good reproductive endocrine hormone manipulators. And because of this, many of the amphibian species are converting from male to female and female to male, and also it's responsible for affecting their ovulation cycle. So you have a very drastic effect of some of these things, so one has to be really very careful while using any of these products. So when you want to use some of these strains in the literature, we have thousands and thousands of different bacteria which are known for their remediation capabilities, which can be used as a sole source of carbon and energy, which could be over a day, 2, or maybe a week. But the major limitation what we face for using these strains in the environment is either the biotic stress which is exerted onto the species, the interaction among the communities, but the worst part which is affecting bioremediation is the one which I have shown it over here in the pink or the red box. That is the carbon catabolite repression. To explain what is carbon catabolite repression, I'll just give you a brief introduction over here. As you know that E. coli or Bacillus subtilis, they utilize glucose as a sole source of carbon and energy. That's classically known as a lac operon, where when you give a lactose and glucose together, glucose is preferred. While utilizing the glucose, all enzymes responsible for lactose transport as well as utilization are shut down. That is the lac operon concept. You all are aware of it. Similarly, in E. coli, you have the glucose utilization prior to succinate, and if they have any ability to utilize aromatic compound which are considered as a highly reduced negative resonance energy and highly complex compound are basically preferred towards the end. On the other hand, if you look at the organisms like Pseudomonas and Burkholderia, which is a common soil microflora, they have what we call it as a reverse carbon catabolite repression, where organic acids like succinate, pyruvate, or any of the TCA cycle intermediate organic acids are utilized prior to the glucose, and that's called as a reverse carbon catabolite repression. And most of the Pseudomonas or Burkholderia species are reported in the literature, they basically prefer reduced aromatic compound which are treated as a complex secondary carbon source are taken towards the end. So that's the reason when you apply these cultures into the environment, you have a first simple carbon source utilized, then glucose is utilized. As a result, you have the aromatic compounds accumulation going on, and hence they are persistent in the environment, and they get bioaccumulated into the system. Are there any cultures or the organisms available in literature or experimentally which can reverse the order of the carbon source utilization? In other words, can they preferentially utilize aromatic compound as a sole source of carbon and energy, leaving succinate and glucose? So I'll give you one favorite example of mine. If I keep 100 gulab jamun and only Karela sabji right over here, and I'll ask audience to come and collect whichever you like it, I'm sure there will be a lot of fight for gulab jamun. Nobody will go for Karela because nobody likes Karela and its flavor. But are there anybody in the audience who will first go to Karela and then later on, if gulab jamun is available, then it's possible? And yes, there are organisms in the environment. So environment is a big scientific laboratory where you don't have to use restriction enzymes or anything there. They are constantly evolving, and Pseudomonas bharatica is one of the species which absolutely follows the very different pathway for the carbon source utilization hierarchy, where all aromatics, benzoate, methyl naphthalene, naphthalene, or any organic solvent, aromatic organic solvent like benzyl alcohol, are utilized prior to glucose, and they are co-metabolized with succinate. In literature, with Pseudomonas, many organic acids suppress aromatic carbon source utilization. Here in CSV86, Pseudomonas bharatica, we have a very different process taking place, which is a globally taking place, where neither succinate nor glucose suppress aromatic utilization. So hence, this strain is a very wonderful candidate to apply into the environment for the bioremediation purpose. So what is the basis for getting this thing? Because everybody knows that lac operon is universally accepted process, but that is not the case in case of now bharatica. Now, to get to this level, we took almost like 5 years to prove by using the growth experiments, which has to be completely reproducible. So before that, I'll give you a small gist of this entire CSV86. We have sequenced the genome of CSV86, which is around 6.8 MBP, which is one of the largest genome available. We have all aromatic degradation pathways present into the organism, which we have functionally proven not by looking only at the metabolites, but also by looking at the enzyme activity, oxygen uptake, isolation of different metabolites, and looking at the active transcription of the operons which are present onto the genome of CSV86. I'm not going to go into details of this, but then the entire summary is given for the metabolic pathways. So metabolic networking is very well nicely studied in this organism. So just to understand the difference in the growth, I have given here the 3 different types of growth profiles over here. One is a carbon source like glucose, glycerol, and fructose, which are very commonly present in the environment. Then you have aromatic compound, which are shown in the green, and you have organic acid, which are shown in the orange color. Now, if you look at the overall growth profile of these organisms, the organic acids and aromatic compounds are basically utilized very rapidly as compared to simple carbon source like glycerol, acetate, or fructose, which is a very unique property which is available with CSV86. I'm not going to go into detail of why this is very unique and what are the molecular mechanisms which we are studying into detail, but for today's thing, I'm going to avoid those things. So here at the bottom on the left side, it shows the specific growth rate on each of these carbon source, which I have shown it over, monitored over here by the growth. So I'll just explain you, this is for now for the MSc and PhD student, how this experiment was done. It's a simple growth curve experiment. While doing the growth curve experiment, we managed to monitor the carbon source available into the extracellular medium. So the culture is grown onto naphthalene plus glucose as a carbon source. Okay? So before that, what is shown over here in the brown is the growth curve of CSV86 on naphthalene alone. So you see the carbon source is utilized within 8 to 10 hours completely. Cells are reaching the stationary phase by 10 hours. The squares are the one which is showing a growth curve on glucose, which is a little delayed, which takes almost like 26 to 28 hours to grow into the stationary phase. So when we looked at the double carbon source, that is naphthalene plus glucose, at same concentration, molar concentration, what you see is the olive color inverted triangles shows a typical dioxic growth profile. Okay? And during this growth profile, what we monitored is the reduced sugar concentration, that is glucose concentration from the medium, and what we see is that till 10 hours to 11 hours, you have a glucose concentration remain constant extracellularly. It has not been utilized. Okay? And once the cell enter into the second log phase, you have a rapid decrease of the glucose concentration. Now, this is the one evidence, and the second evidence for this is that during the first log phase, we see the naphthalene, which is added in the form of the crystals, are getting completely utilized. And as a part and parcel of utilization of naphthalene, what we see is the olive green color, which is shown here in the inverted triangles. That is the typical color of CSV86 when it is growing on naphthalene. So by using this visual observation, naphthalene crystallization, and utilization of C14, monitoring C14 glucose from the medium, we basically come to this conclusion. Now, this is not true only for the naphthalene plus glucose. The number of different aromatics along with the glucose, or naphthalene plus acetate or naphthalene plus glycerol, basically showed the typical dioxic growth profile, where aromatics are utilized in the first log phase, glucose is utilized in the second log phase, which I'm not going to go into detail, but here what I'm basically trying to say is that during the first phase of the growth phase, where the aromatics are utilized, all enzymes responsible for aromatic utilizations are induced, while the enzyme responsible for glucose utilization are suppressed. The same enzymes are basically now getting induced when glucose is getting utilized. The same thing was done by using the oxygen uptake studies and by various other biochemical methods to come to this conclusion. So like other pseudomonas, what it shows is a reverse carbon catabolite repression. That is, organic acids are preferred over glucose, and that's why we see the organic acids are getting utilized in the first phase, and then you have a glucose utilization in the second phase. So this works as an internal control for all our novel experiment or the results what we observed it. And in case of aromatics plus organic acid, what we see is the co-metabolism, where the both carbon sources are utilized together. So this is a very novel, unique physiological property which we observed, which is a globally taking place in CSV86. So further, we went for the qPCR and actual transport processes which are responsible for the aromatic degradation and all those things. So here I'm going to, for the simplicity, for the clarity, I have taken the example of benzoate plus glucose. What it shows is a dioxy growth curve. These are the two basically gene operon. One is for the benzoate utilization, other one is for glucose utilization. BAN-E is responsible for the glucose benzoate transport into CASV86, while the GBP is a periplasmic protein which is responsible for the glucose transport via ABC transporter system, which is an ATP dependent system. So when we looked first at the glucose transport, when CASV86 is grown on benzoate plus glucose, we see the glucose uptake through the C14 is taking place mainly into the second log phase. Compared to that, when we look at the benzoate transport, C14 benzoate transport, we see it in the first log phase. So we have a clear-cut cyclic rhythm of benzoate utilization in the first phase and a glucose utilization in the second phase. We went further to look at the BAN-E expression, the enzyme of the protein which is present into the inner membrane for the benzoate transport, which is basically expressed in the first phase of the growth curve. Similarly, when we looked for the GBP expression, which is maximum during the second log phase of the cell. So what it basically tells us that it is not only at the biochemical level, but also at the transcription level, it is getting synthesized freshly. It is not the enzyme pool or protein pool is present into it and getting activated. No, it's a fresh synthesis of mRNA, fresh synthesis of protein, which is responsible for this activity. So overall, if I summarize the entire biochemical analysis for last 20 years by almost 15 PhD students for CASV86, this is what I can present the slide. What is shown over here on this left panel is all aromatic capabilities, abilities of the CASV86 to utilize aromatics preferentially over the glucose and any other carbon source which is shown in the yellow. Okay? And what is shown in the blue is the TCA cycle organic acid intermediates which are produced as a part and parcel of the aromatic metabolism. All those are co-metabolized. Neither glucose nor organic acids are able to suppress or shut down the transport or induction of aromatics. So that is a very, very important biochemically process which is taking place, suggesting that there is a major global switch regulator in terms of CASV86. So when we looked into it, we found that it has a genome plasticity. It has 3 ICEs present into there. One of the ICE is IC-NAH CASV86, which belongs to the CLC element, which basically carries the cargo, that is the genes required for naphthalene metabolism through the salicylate pathway, which we call it as a IC-NAH CASV86. Besides that, we have other 2 ICs. One is the PBG1, which is responsible for the heavy metal resistance. And then we have a PBG2 ICE, which at the moment we call it as a mixed function. At the moment, we do not have the detailed analysis what function it carries out into CASV86. But we have a very nice, these 3 ICs present into it. And we call it as a conjugative elements because these plasmids or these genes are able to transfer into recipient. In case of CASV86 for the naphthalene, we can transfer the naphthalene degradation genes to another strain, CASV89, through a conjugation, but at a very low frequency, which is around 10⁻⁻⁷ as compared to the very well-known characterized conjugative plasmids. Well, this has a lot of ecophysiological properties. It has various transports, but what is important here is that besides aromatic degradation, preferential degradation, it has a heavy metal tolerance, it has a salinity tolerance, it has a fusaric acid resistance, biofilm production, antifungal property, it has a PGPR activity. So it becomes a very important microorganism where we can apply it into the agriculture field now. So we have done certain experiments in the agriculture with the open pot culture along with this bacteria, few other bacteria where we can basically look for the effect, plant growth promoting effect in presence of the various pesticides. Okay? Naphthalene, methyl naphthalene are one of the insecticides which are used in the formulation of the various pesticides. So our interest now is to have such a nice, beautiful host which has a preferential utilization, has a good bioremediation potential, has a good PGPR activity also. Now, can we basically enhance the metabolic capacity of this microbe, CSV86, basically to have a more aromatic compounds degraded? So one of the examples which I'm going to give you from our lab is the Pseudomonas species, which we have isolated, which can degrade carbaryl within 10 hours. Now, carbaryl is the product which was produced by the Union Carbide in Bhopal, which is responsible for the Bhopal gas tragedy. I guess you all are aware of that. We are still facing the problem because of the mutagen which was produced by the interaction of water with methyl isocyanate, which was used for the carbaryl synthesis along with the 1-naphthol as a base product. So here, this culture, C5PP, was extensively characterized for CSV86, and we know the metabolic pathway which is shown here in the green. The green pathway is the conversion of carbaryl, which is transported into the periplasm, which converts into 1-naphthol and then further metabolized through the naphthalene route, which is shown in the black color. Okay? And then it basically metabolized to a TCA cycle intermediate via salicylate and gentisate route. Now, if you compare this pathway along with the Pseudomonas bharatica pathway, which is shown in the blue on the right side, what you see is that naphthalene is again getting converted into 1,2-dihydroxynaphthalene, which is a common intermediate for both carbaryl and naphthalene. So our interest is to take first 3 proteins, that is MCBT, the one which is responsible for transporting carbaryl into CA5PP, then the carbaryl hydrolase, which will convert carbaryl into 1-naphthol, and the third enzyme, which is a cytoplasmic enzyme, which is converting 1-naphthol into 1,2-dihydroxynaphthalene. So in this case, we are targeting 3 different proteins at 3 different locations: outer membrane, periplasm, and cytoplasm in one shot into CSV86 for the engineering purpose. Atya, just let me know how much time I have it. So then I can, 2 minutes? Okay. So this is the total IC. I'm not going to spend much time over here, which basically tells you the different promoters available. What we decided to use the PNA promoter, which is the original promoter of CSV86. We have done the different studies, which we found that this promoter is leaky promoter in CSV86. So to design having these 3 different proteins at different compartments in CSV86, we use the wild type promoter which is present in CSV86. Along with that, we have the entire genome sequence for C5PP. We know this entire pathway is present in the 3 segments, in 3 operons, which we have proven it transcriptionally. And we have taken the upper pathway operon, specifically the MCBT, CH, and 1NH, along with the SEVA vectors. So this is the SEVA vector, which is basically developed by Victor Lorenzo in Madrid. We have some of these vectors available. If anybody is interested, you are welcome. We have a permission to distribute among the Indian communities, where these are the shuttle vectors which can be used in Pseudomonas as well as in E. coli for cloning expression purpose. Having a different antibiotics, different promoters, and one can have a multiple cloning site along with the different modified repertoires into it. Okay? So we use the SEVA chassis for it, and we basically constructed 2 different constructs. One construct is without MCBT, without the transport, because we were the first one to say at a bioinformatics level that MCBT is a probably outer membrane protein responsible for transport of carbaryl into the cell. But that is not experimentally proven. So we did 2 constructs. One is a PNA, that is a promoter which we use from CSV86, CH, that is the carbaryl hydrolase, which is present into the periplasm, and 1NH, which is present into the cytoplasm. Along with that, we have a MEC2 construct, where we have a MCBT, which is a outer membrane protein, which is responsible for transport of carbaryl into CSV86 now. So with this, we did this experiment. The circles at the bottom shows the CSV86 wild type doesn't have the ability to grow on carbaryl as a carbon source. That's true. When we basically put MEC1 construct, that is only 2 enzymes, carbaryl hydrolase, which is in the periplasm, and CH, which is present into the cytoplasm, it basically gives the ability to CSV86 to start utilizing carbaryl as a carbon source. The moment we put MCBT into this strain, we see the further positive effect onto the growth curve, suggesting that MCBT is now a transporter responsible for transport of carbaryl into the cell. The other way to prove, which I am not showing it over here, is that we have deleted MCBT from C5pp, wild type strain. It shows the defect in the growth on carbaryl. So with this, we saw that, well, it's taking carbaryl, but now pathway is that carbaryl to 1,2-dihydroxynaphthalene to salicylate and catechol, that is going through the naphthalene route, which is shown here in the blue. Now, after introducing carbaryl genes into CSV86, does it retain the preferential utilization ability? That is the most important one, because we want to have that system going in there. So when we did, I'm not going to go into details of biochemical analysis. We did it by the enzyme activity. We did it by using the whole cell oxygen uptake as well as by the quantitative PCR. What we found out that we have a dioxic growth profile. In presence of carbaryl and glucose, culture is preferentially utilizing carbaryl and later on it's going as a glucose. So what it basically tells you that even if you modify CSV86 with the new pathways into it, it still holds on to its global regulation of naphthalene or the aromatic degradation in prior. So similarly, we went for other two examples. I'm not going to go into details. I will just give you the final conclusion of the entire thing over here. We basically went ahead for further engineering this strain for the gentiset production by using CSV86, by using salicylate as a substrate. And also we have engineered CSV86 now with the lower metabolic pathways. Now the cell can degrade substrate like salicylate, one way via gentiset, the other way via catechol, which are very rarely occurring phenomena in the nature. So at this stage, I'll conclude saying that we have now Pseudomonas bharatica CSV86 with a novel, very unique ecophysiological property and a property of physiological, the preferential utilization. Good part is that the property is very stable. It doesn't get affected by adding a new genes into it. And we have basically optimized now CSV86, engineered CSV86 for the carbaryl degradation. And we have now basically newly genetically engineered pathway in CSV86 to enhance the metabolic diversity at the lower intermediates like salicylate to gentiset and salicylate to catechol, which are the major intermediates for various polycyclic aromatic hydrocarbons. Okay? So at this stage, I would like to thank all the fund givers, collaborators. I have listed only few of them, various PhD students present, and no doubt, microbes. I was really lucky to have some of these two unique microbes. That is, one is the Pseudomonas bharatica, CSV86, and the other one is Pseudomonas species C5pp. Okay? Thank you very much.
Thank you, sir, for sharing the insightful thoughts with us and telling us more about this. Now I request our Pro Vice Chancellor, Professor Sandeep Sharma, sir, to kindly join us on dais and share a token of respect and gratitude to our keynote speaker, Professor Prashant. My audience, can we have a huge applause for our keynote session by Professor Prashant. Thank you, sir. Thank you, ma'am. All the participants, students, at the back side, as already been mentioned yesterday by Professor Kuhad, that anybody has any suggestions or any idea that they want to inculcate further, we have kept the box on the right-hand side of mine next to the stage. You can drop it there. It would be taken care of. Thank you. Over to the chair and co-chair for the further sessions.
Thank you so much. So we get on now with the sessions. Each speaker will have 15 minutes and Dr. Prashant will ring a bell at 14 or 13. and then a warning bell at 13 minutes and then at 15 minutes. So our next speaker is Dr. Chinmay Kumar Sarangi. He's a leading authority in the field of extractive metallurgy and resource utilization. Dr. Sarangi serves as a senior principal scientist in the electrometallurgy department at the CSIR Institute of Minerals and Materials Technology, that is IMMT, Bhubaneswar. His distinguished academic qualifications are rooted in tier institutions, having earned his BTech at NIT Roorkee and both his MTech and PhD in metallurgic and material engineers from IIT Kharagpur, where he was awarded the Institute Silver Medal for securing the first position in his MTech program. Before joining CSIR IMMT, Dr. Sarangi began his career as a scientific officer at BARC in Mumbai. His core research contribution lies in developing innovative, energy-efficient, and environment-friendly process for the extraction of metal values. He's a specialist in electrometallurgy and membrane electrolysis, and his work is critical for national self-reliance, focusing on the recovery of strategic and critical metals like tellurium, cobalt, and tungsten from complex industrial waste. He holds several patents for technologies developed in his lab, including those for metal recovery and fly ash processing, which he has successfully transferred to the industry. His commitment to translating research into commercial impact makes him a key figure in India's circular economy and sustainable resource development. Dr. Sarangi, we look forward to your presentation.
Thank you, ma'am. Very good afternoon to all of you, respected chair, co-chair, delegates, faculties and students. So I am Chinmay from Bhubaneswar. I belong to CSIR IMT, where mostly you will find the research related to mineral and metal industries. Now it would be a little bit eye-opening that why this biotechnology in the field of mineral and metal industries. That definitely it is because of the lack of resources and that technology which we need to have for particular resources. So that I am going to address in this talk. So mostly these are the works which we have done at IMMT and how biotechnology plays a big role in particularly dealing with these critical metals and their extraction from the resources which India is having, that we will be discussing. So few case studies also we will cover. And so that's why this title goes as Biotechnology Approaches for the Recovery of Critical Metals from Low-Grade Resources. Because the technology which we have, those are for high-grade resources. There is no difficulty. You can extract them with good efficiency and all. But what happens to low-grade resources, that we need to cover here. First of all, just I think most of you must be aware of critical metals. For our students, I'll just tell you in brief why we are so much of curious about these critical metals. As you know, Government of India has declared, even it happens to all the countries across the world, that we need to go for clean energy, national defense security, space research, and aerospace, medical as well as transportation applications. So there are certain metal components are required which we don't have, particularly supply resilience is there. There is no consistency in the supply of those metals. And the resources which are required to extract them, that are also not available. So that's why for the economic growth as well as for your safety, any country needs those metals. So accordingly, these are the important metals from that concern. Now we have issue here that we have to be dependent on other countries. Suppose, for example, a lot of metals you will find that China is having the control in the trading market. The moment they see that the specific country is buying certain metals, they stop their trading. So thereby, if you have your own indigenous technology, then only it will be helpful for you to survive. So that is why critical and about 30 critical metals have been identified for India, which we have a scarcity in availability. So that's why it is necessary to go for domestic intervention. So there we need to identify those low-grade resources which is economically not exploitable at this stage, But if we can have our own technology and technology intervention can be there, it can be made strategic. Similarly, international intervention that we have to identify certain resources which other countries can provide. So that's why geopolitical connections and those relations are also very much important here. So overall, you can say that we need sustainable technologies which can give you both economical as well as environmental friendly. Earlier, people were not much cautious about the environment, but nowadays, before setting up a plant, first of all, environment is the first criteria that it needs to be protected. So let's have a look at the conventional technology where we are dealing with hydrometallurgy. Basically, we use water and acid or alkali, or electrometallurgy, that electrical energy, pyrometallurgy, where you have to provide heat in the form of coal, coke, or other natural gases. But in all these conventional technologies, what happens that anyhow, you have to provide external source of energy in order to do certain reactions. But when you provide these reactions through these resources, what happens, you also emit carbon dioxide and other emission gases, which are also not good for your green environment. So there, this biohydrometallurgy concept comes into picture, that it can provide you a greener technology, as well as it can also deal with low energy input. So all those advantages become, make it a potential technology, and those are like eco-friendly with low energy input, suitable for low-grade ores, and it is also scalable, and simple technological outlay you can prepare out of it. Low capital investment, because in any technology, when you go for deployment, you have a capital investment as well as a maintenance one, that the operating cost. Although operating cost matches in both the cases, but capital investment is much below as compared to the conventional technology. So usually what we do, this critical metal bearing resource, if you have, that goes through a pre-treatment. Pre-treatment means naturally either you will find them in the form of sulfides or oxides or some other carbonate form, but sometimes it is not feasible to process them directly through the conventional technology. That when you go for leaching, suppose in acid or alkali, there is a possibility that these metals may not behave properly or may not respond. So thereby this pre-treatment is required. Once you leach them, that means the metal you are bringing into the solution in ionic form, and then you have that solid liquid separation, so that you have a leach liquor. So that leach liquor contains your metal ions. But problem is that along with your desired metal ions, some co-extracted metal ions will come. Those are impurities for you. And in order to separate them, you have to go for subsequent operation like solvent extraction, ion exchange. So those technologies will be helpful. Once you have a pure solution, then you can go for a material specific product, or you can go for a metal deposition through electrowinning. Now the biotechnology, here the approach we can apply, the scope is that on the top head, upstream. So upstream process, particularly pre-treatment or this leaching, where you need a reagent which needs to oxidize or reduce. So thereby you can incorporate this biotechnology. That is the area where people are working in, and they want to switch from a regular high temperature metallurgy system or from a acidic or alkali system, which are not good for energy point of view or from environmental point of view. That is the whole idea. So here few examples I have given, that copper, nickel, cobalt, rare earth elements. Those are the areas where subsequent research has gone. And a few of them I will also address, which we have worked on. This is one case where we extracted this copper metal from a low-grade ore. So usually your cutoff grade for copper ore is 0.45%. So nowadays people are also going with a lot of efforts to 0.4%. But this is a source which is available in Malanchkand, that Hindustan Copper Limited, that contains only 0.53% copper or less. And there mostly it is chalcopyrite, that's sulfide based. Now, if you go for a conventional technology, it will not work. Either you will not get the kind of recovery which you are desiring, or it will not respond at all. So there, this biotechnology helps a lot. So we have incorporated this bioprocessing approach, so that Acidithiobacillus ferrooxidans bacteria that helps in leaching out that copper into the solution. So thereby, in the heap form it was prepared. At room temperature it operates, so that in order to go for high energy systems. And once you have the solution, with copper ions. Then it goes for purification through solvent extraction. That is a liquid-liquid extraction system where the copper is extracted by organic solvent and then it's stripped with sulfuric acid. And then you have a pure copper sulfate solution which goes for electrowinning so that you can deposit that copper metal sheet on the cathode. And once you have those cathode sheets, then you can go for melting, casting and downstream processing to get your regular components. So that is the whole idea. So here 1 GPL of copper was prepared from those bioleaching process over a period of 13-14 days. So minimum 10 days you need to get that 1 GPL concentration of copper. Once that is ready in the backflux tank, then it goes for the subsequent purification followed by the electrowinning. And more than 97% current efficiency we got through during this electrowinning of this copper metal. This is a schematic you can see on the left side that the heap along with the backflux tank where along with that compressor system that the bacteria survival as well as the solution which is getting ready from the heap after leaching. And these are some of the photographs on the right side. You can see heap followed by the liquid, that bacterial solution and the electrowinning setup and these copper sheets which we got with a purity of more than 99.9%. So that also we call it 3N copper sheets. So this is marketable, directly it can go to industry. Another example is that ball mill spillage. So these are when you grind those ores, a lot of spillage comes out, which is a very low grade resource, even it is less 0.26% copper. And there most of the things are insoluble in acidic system. So directly here also when we opted for the bioleaching, that didn't work. So thereby a thermal activation was required, and that thermal activation at a 600 degree temperature, what happened, it goes for a phase transformation. And that phase, since it is a volume expansion process, it creates the crack. And because of those cracks, that penetration of the bacterial solution was possible, and then it could leach out. So that's why in the graph you can see that with different flow rates of those bacterial solution, we are getting 1.5 liter per hour maximum possible recovery. And this is a experimental setup, which is a model which is applicable in our heap leaching system. So it's a column leaching setup. In the similar manner, there is a sprinkler at the top of the material. So ball mill spillage is charged in that column, and the sprinkler is providing that solution, which is leaching into the backflux unit, and from there the subsequent processing is carried out. So here you can have a look at the flow sheet where the sequence of operation, as I told you, it goes through. So I need not to repeat that. But look at that SEM photographs where you can see the initially those two top photographs. There you can find that it is a very smooth surface. The moment it is undergoing through that thermal activation, there is a wavy structure, so that there is a change in that phase, physical changes in that phases. And finally, the cracks and pits are forming in those third layer of SEM photograph, which is an indication of the fact that the leaching has happened and your copper metal has been dissolved from that material. This is another example that chromite overburden, where nickel is a very important metal for India because we don't have any resource of nickel in our country. Mostly secondary resources, either scrap, waste, that is what we have. Now, in order to have and you know nickel is very important from alloy application as well as batteries, lithium-ion batteries, what you are using in your regular applications, where a lot of nickel is required. So there, India doesn't have any primary resource, only secondary resource. That's recycling you can do. That is one way of doing. But the only source India is having land-based resource, that is chromite overburden, which is available in Sukinda Valley in Odisha. But the problem is that it has a very low concentration of 0.7%, which is not exploitable because minimum you need 1% of nickel in order to make it a commercially viable technology. And cobalt also is there 0.05%. So a lot of research has gone into this chromite overburden. Conventional technology, as you can see, this flow sheet, which goes through a reduction roasting, then ammoniacal leaching, and then you are extracting this nickel through purification route, and we get this nickel cathode and cobalt solution. And cobalt solution can also go for the similar process to get cobalt metal. But the problem is that it is economically not feasible because of the low concentration of nickel. Now, the idea here is that can we incorporate biotechnology here on the upstream processing? So that is what we did here, but it was not that easy. This nickeliferous chromite overburden, what happens, this nickel is present in a log manner in the goethite phase. So mostly you will find these phases, goethite, hematite, and in that this nickel is being blocked. So until unless you have a channel to take out that nickel out of those phases, it is very difficult. That is why this iron reducing bacteria has been used here. In the earlier case when I was talking about copper, there we are using oxidizing bacteria. Here it is a reducing bacteria. And because of that, we are able to extract that nickel out of that COB, chromite overburden. So that we have done successfully. And these are the results, what you can see that acicular goethite is transferred into a globular magnetite, and further the feasibility of getting nickel out of that matrix is possible. So this is the flow sheet, as you can see, which goes through bioreduction followed by this acid leaching, then purification and metal deposition. So 99.9% nickel was obtained from a 0.7% nickel bearing material. So at the end, I would say that sustainability and challenges are very much there in the biotechnology also. One is kinetics. It's a slow process that has to be taken care of. Then only it will be able to compete with the conventional technology. And other regulatory social acceptance as well as technological intervention is also required. So with that, I would conclude here that there is a growing demand of these technologies at this stage. We have the resources, but we don't have the technologies. And in order to get those sustainable technologies, technological intervention is required. So a lot of, as we are talking about the incorporation of biotechnology or bioleaching into the system, that needs to be taken care of. Then only we can have a positive environment and a good technology to take further. So with that, I will conclude that. Thank you.
Thank you, Dr. Sarangi, for a wonderful talk and for maintaining time. So what we are going to do is, since we are running really very late, we will shift the questions to the end.
Sure,
if there is still time, then we will have a round of question answers. But right now, we would like to...
Now request our chair to co-chair to kindly share a token of respect and gratitude to our speaker, Dr. Chinmay Kumar Sarangi. Audience, can we have a huge round of applause? This is what I need to tell you once again. This cap is a resemble of our state Uttarakhand, carrying a Brahm Kamal on the top, which is the state flower of our nation. Thank you, sir. Thank you, sir and ma'am. For the next session, I request chair to co-chair to kindly introduce him.
So I'm very happy to invite Dr. Abhay Bajaj. Since he has a flight to catch, we are just, breaking the order a little bit. Abhay? So very briefly, we work together, Abhay and me, and so I know that he is a really dynamic and a brilliant scientist. Right now, he's a principal scientist at CSIR IITR Lucknow, and he also serves as an associate professor with ACSIR. He holds a PhD in environment biotechnology and MSc in life sciences. His career spans multiple prestigious research institutes across India, underpinning expertise in environment genomics and microbial technology. His professional journey spans National Innovation Foundation, Ahmedabad, University of Delhi, NCMR at the National Center for Cell Science in Pune, CSIR NEERI, very, very briefly though, and now with CSIR IITR. His research interests center on environmental microbiome, biodegradation and bioremediation, and the development of biomolecules and bioactives for human and environment health. He has led and contributed to major research projects funded by CSIR, DBT, and MCG. He has over 45 publications, 75 database submissions, and extensive sequencing microarray data sets, multiple presentations, conferences, and workshops, demonstrating his commitment to his scientific career. Over to you, Abhay.
Thank you so much, ma'am. Respected chair, co-chair, delegates, faculties, and students. In the next couple of minutes, I'll be talking about the wastewater microbiome, how the urban wastewater microbiome is contributing towards the development of antibiotic resistance in the environment. In the previous presentations from our chair and co-chair, you all are now primed towards the pollutants and the microbes. I need not go into the details for it, but then just a brief, we are struggling today with the legacy pollutants, and again, in addition to that, there are pollutants of emerging concerns like the pharmaceuticals, nanoparticles, plastic debris, biopesticides, and various cyanotoxins. These definitely will be the legacy pollutants in the coming times. So the question is, how microbes which are in the environment in all the three matrices, air, soil, and water, interact with these pollutants. We all know the pristine environment, we call it as a baseline situation where the environmental impact of these pollutants creates an effect. The effect may be the short term. So the effect may be the short-term effect and the long-term effect. By the time we realize the long-term effect, we are not in a position to go back to our baseline data. But thankfully, we are in an age of an advanced genomic tools, which we can use to monitor the environmental matrices and understand what is the environmental impact of these pollutants and how microbes are playing their role. So, just a simple example I can quote is sparrows. We don't see sparrows. The children nowadays don't see sparrows, but the generation of our chair and co-chair and me, we have seen the sparrows. So this is a long-term effect of maybe the industrial impact of, or we can say the pollutants on the environment. So prima facie, who is the primary receptor of these perturbations which are happening in the environment? It's of course the microbes. And these microbes develop mechanism, they undergo evolution, and we need to use these approaches to understand, to characterize and understand how microbes adapt to these pollutants. The environmental processes has been described using metagenomic tools in the previous lectures, but then I'll briefly talk about the microbial signatures related to these environmental processes, be it the biogeochemical cycles or the population interactions like heterotrophy or the phototrophy. There's always a microbial signature to it, and that is what we do in metagenomics. We retrieve the microbial genome directly from the environmental sample, regardless of the nature of the sample, and we study the taxonomic, we do the taxonomic identification, we study the functional attributes, and we try to recover the genomes of the bacteria which we are not able to culture in the lab, because those are the key players which decides the ecosystem functioning. So I'll take two cases. First, in the first case, I'll be talking about the urban wastewater, where we studied the prevalence of the antibiotic resistance gene. So it's an environmental resistome. What is a resistome? It is a collection of all the antibiotic resistance genes present in a particular matrix. So before going into the findings of what we did, how this resistance emerge in the environment. So there's a natural pool of bacteria which is there, and there are various contaminants, as I mentioned before. These may act as a stimulant. They create a selection pressure on these bacteria, and then they undergo, there are different mechanistic ways which the bacteria has to counteract these stimulants, these pollutants. So one could be the reduced permeability, specifically to the antibiotic I'll talk about. The second one could be they may actively efflux this antibiotic from the cell, or the modification of the target site where the antibiotic works on the bacteria for killing it. Or what it can do is it can directly inactivate the antibiotic. So these are the mechanisms which a single bacteria has to counteract these antibiotics when it faces the challenges from the antibiotic. But there are ways with the bacteria to deal with this condition. It's a horizontal gene transfer. So what happens is that when there is a mix of the resistant and the susceptible population, because of this selection pressure, the mechanism of horizontal gene transfer comes into the play, and there is an increase in antibiotic resistance bacteria and the genes linked to the mobile genetic elements. So this is the basic idea of how it behaves in a single cell model. But we were interested to understand how it happens in the environment. So what we did is, we attempted a drain metagenomics to understand how it is happening in the particular at a city level. So we mapped the whole city of Nagpur, and we all know we discharge the sewage from the households, from the industries, and the commercial places, and it goes to the sewage treatment plant. So the question was, we were intrigued to know what is happening before it reaches the sewage treatment plant, and how the sewage treatment plant is actually benefiting or functioning towards maybe contributing to the development of antibiotic resistance, or it is minimizing it. We wanted to know that. So what we did is, we mapped the whole city, and it's like nearly 80 samples we collected across the whole city in 2 phases. We generated nearly 1 TB of data, and metagenomic data, of course, and we did the taxonomic studies, the functional attributes we studied, and also the resistome profile. I'll briefly share the findings, what we found. We identified, I'll not talk of the general diversity, how it was, what are the numbers of the bacteria and all, but then our main aim was, can we use these genomic tools which can be used for monitoring purposes? So we looked at what are the taxonomic markers which are differentially present and have been and could be used as a tool for monitoring the efficiency of the STPs. So we could identify 4 major taxonomic markers at the genus level. One is limnohabitans, which was predominant in the STP, which clearly reflects that limnohabitans. So again, this is where the traditional microbiology and taxonomy helps us. Culturable studies helps us. We know limnohabitans belongs to a freshwater environment. So it was not found in the drain system, which is an open or the closed drain system, but in the treatment plant, we could identify that. In addition to that, there is another Tenibaculum. It is an opportunistic pathogen for the fish. So after the treatment of water, we discharge, I mean, this is how it goes everywhere in the country. The water is discharged into the natural environment, maybe the riverine system. So this is one of the taxonomic marker we identified, which could be an opportunist pathogen for the fish and can be a threat to the aquaculture. We studied some functional markers also. Like we studied how the distribution of genes for drug resistance, specifically the antimicrobial drug resistance, for the infectious diseases, for the membrane transport, which is again one of the mechanism for the development of this resistance in the bacteria, and also towards the degradation of xenobiotics. We identified these markers which were differentially present in each kind of setup, in the drains and the STP. We also predicted the phenotype from the genotype. We did the resistome analysis. The arrows are a little hidden by this, but if you see in these 3 in these 3 columns, the one which are showing the highly red region. So in the STP, that is the activated sludge, where since there is a retention and there is a community dynamics going on, which leads to the accumulation of antibiotic resistance genes. This is what we get to know from this. And in the open drain setup, we also identified a high prevalence of ARG subtypes, resistance gene subtypes. This specific sample belongs to the wastewater from the hospital setups. There is the commercial area which belongs to the hospitals. We collected the samples from there. This was very interesting. I mean, it is expected, but then we could actually see how it is there in the environment. We also did the profiling for the chemicals, the emerging chemicals of emerging concern, like antimicrobials, drugs, food additives, and cosmetic compounds, and we have the plastic additives. We could profile them in this drain setup, and we identified that, yes, outlet of the STP has a higher concentration of these emerging contaminants, which suggests that definitely there is a selective pressure of these contaminants in the environment on the single cell bacteria, which is forcing them to have a genetic makeup towards antibiotic resistance. So this was in the environmental setup which we found at the city level. So now when we wanted to know how it is actually there in different other matrices. This is one urban wastewater we have used. So what we did is we tried to study a very simple parameter, dissolved oxygen, how it impacts the distribution and the emergence of antibiotic resistance. Again, we used the samples, the already available metagenomic data sets from the human gut, chicken gut, soil, ocean, and fresh water. We found there is a correlation between the abundance of ARGs at the different DO level with respect to the distribution of oxidative stress genes in the samples and the abundance of pathogens. All these three things were correlated. Then what we did is we went on to do the bioreactor studies, simulation studies. We did the simulation for 6 months, and to understand, is this oxygen, first question was, does oxygen determine or does presence of oxygen shapes the emergence of antibiotic resistance? Second was, we wanted to understand how this happens and what is the correlation between the DO level, and how it is related to ARG subtypes, antibiotic resistance genes, and of course, the major player, mobile genetic elements. So I'll briefly talk about the mobilization and co-occurrence of antibiotic resistance genes. In this, I have highlighted with the red. So at the 150th day, after we have stabilized the bioreactor for 2 months, we ran it for another 4 months, and we found that there is an increase in antibiotic resistance genes, specific subtypes of these genes at the 150th day. I'll briefly go back. In the bioreactor, we set the 3 conditions. One was the higher DO, around 7, then the medium DO, around 3 to 5, and then the normal, lower DO, which is very minimal oxygen we tried to provide. We found that presence of higher level of DO affects the relative abundance of antibiotic resistance genes. Similarly, when we looked at the mobile genetic elements, we find both are correlated. The mobile genetic elements are also enriched in presence of higher oxygen level. So we tried to look at what antibiotic resistance gene are linked to which mobile genetic elements. So mostly class 1 integrons we were able to identify which were in higher abundance, and during the simulation studies we found that the presence of oxygen shapes the resistome in a way where class 1 integrons are enriched. So this is the findings which we got, and this study suggests that a simple parameter like DO can actually regulate the shaping of antibiotic resistance in the environmental setup, and we need to have the mitigation strategies to intervene at this point of regulating or reducing something like an oxidative stress, so that we can actually stop the dissemination of these ARGs from the clinical pathogen to the non-clinical isolates, the environmental isolates, because that is what is actual threat to our public health. So to conclude, the key lessons from the environmental resistome which we could understand is, we need to parallelly study the mechanism, how they are happening in the environment. Second is management. Of course, government is taking a lot of effort and is implementing a lot of action plans to deal with this. And last not but the least, the monitoring, which is actually the mainstay to address this issue of development of antibiotic resistance in the environment. So towards the end, I just like to thank the conference organizers and the AMI, especially Dr. Nijra and Dr. Nabil for having me here, my research team, my research lab directors who have generously supported me to conduct these studies. My collaborators, one of them is already here, Dr. Atya Kaple. The work which I have shown here has been generously supported, discussed, thoroughly discussed, criticized. We had a lot of discussion during the work with the collaborators, and we were able to identify, that was the aim, and I'm really thankful to my collaborators for that and the funding agencies. Thank you so much.
Thank you, Abhay. Very nicely put and great work. I hope you take this forward. Now, on behalf of the organizing committee, we'd like to give you a small token.
Yes, I request the chair and co-chair to kindly present a token of respect and gratitude to our speaker, Dr. Abhay Bajaj, scientist at IITR Lucknow. Thank you, sir. Thank you, ma'am. It is time for the next speaker session. I request chair to overtake.
So we have a next speaker, Dr. Selin Acharya. She is a scientist from BARC Mumbai, and she is going to give a talk about the adaptive response of metallophilic prokaryotes to uranium toxicity. So Dr. Selin Acharya is a senior scientist at the Molecular Biology Division at BARC Mumbai, Trombay, a microbiologist by education. She received her postdoctoral training in chemical biology from University of Warwick, UK. She has over 28 years of research experience in the area of environmental microbiology. It's a very long-standing experience. Her research is primarily focused on the mechanism of metal tolerance and metal detoxification in the microbial ecosystem. Her special interest lies in the area of microbial interaction with the uranium, which are the key importance for the uranium bioremediation, which is a very important aspect which BRC is taking care in terms of looking into the environmental aspect. So it's a very important. Her current research projects are in the area of metal efflux, metallothionein, protein secretion, and metal homeostasis. She has received a DAE Scientific Technical Excellence Award, DAE Group Achievement Award, and she has a recipient of DBT Crescent Biotechnology Overship Assistantship Award, and then award from the Maharashtra Academy of Science. Membership of National Academy of Science India. Dr. Acharya is academic editor of Frontiers in Microbiology and BMC Microbiology Journal. She has guided several PhD students and master students. She has published over 75 papers in a peer-reviewed journals. She has 2 patents to her credit and 5 book chapters. So it's a very good accomplishment over a period of time, and she has delivered almost 25 invited talks and 4 keynote lectures. So we are looking forward to have your lecture on adaptive responses. Thank you.
Very good afternoon to all of you, and thank you, Dr. Fale, for such a kind introduction. And I'm Dr. Selin Acharya from Molecular Biology Division from Bhabha Atomic Research Center, Mumbai. So we at our division, at our lab, we are looking at the cellular and molecular mechanisms for the microbial interactions with uranium. Uranium, because it's a kind of, a mandated, it's a mandate from our department. And so I'll be kind of, sharing a few interesting stories from our lab. So I'll be talking about the metallophilic prokaryotes. So metallophilic prokaryotes are the organisms which are isolated from the metal-enriched environment, and it shows that kind of inherent metal tolerance. Now, the tenet of the microbial ecology says that every microbe can be found everywhere, but the environment selects. So what kind of the microbes they are going to perpetuate in a kind of environment, it is dictated by the environment. For example, here you can see the acid mine drainage formation at a former uranium mining site in Germany. So here the pyrite oxidation, which leads to the acid mine drainage, it makes the environment very, very acid. So the pH falls down to 1.5 to 2. So there only these kind of acidophiles like Acinetobacter bacillus, it can survive and perpetuate. No other strains are available here. Similarly, in case of the uranium-rich tailings, when the microbial composition was investigated, it was found that only these actinobacteria, which was able to biomineralize uranium to form the phosphate, uranium phosphate mineral, they could survive in that kind of environment. So these are some of the examples that how the environment dictates. And this uranium is the heaviest naturally occurring radionuclide. It's known mostly for its chemical toxicity rather than radiotoxicity. It's weakly radioactive. And the other thing is that there is no known role of biological role of uranium as such. And most of the uranium which is now getting kind of contaminated into the aquatic environment or elsewhere is because of the high usage of the phosphatic fertilizers or the nuclear processing technologies. So uranium is relatively a new metal for the microbes to deal with. Now, in two of the examples where I have cited here, for example, when this is one discovery from this lab where they have shown that the microbial metalloproteome, when they have investigated, they found uranium was mostly incorporated in ferritin in a strain called Pyrococcus, which was isolated from a hydrothermal vent. And in another study, they have shown, they have identified a uranyl binding protein from an organism of Microbacterium, which was isolated from the uranium-rich soil in France. So these are some of the examples to cite that yes, these organisms which are isolated from such environment, they have some kind of adaptive features which allows them to survive in those kind of organisms. Now, I will share two stories from our lab which will give you an idea on the extraordinary resilience of what kind of the adaptations these kind of microbes show. Now, the first one is marine filamentous cyanobacterium, that is Anabina torulusa, which has been isolated from the Trombay paddy fields in our laboratory. And here it was isolated during the flooding, the seawater, flooding of the seawater. So seawater remains one of the most important secondary resource of uranium, having 3.5 billion tons of uranium, and uranium is found in the form of uranyl carbonate there. Now this organism was isolated from there, and this was a serendipitous discovery, I would say. I mean, when we start this experiment, it looks green, and then after 5 days, it was getting bleached, and then after 16 days, you see a complete regeneration. So what was going behind the scene, I will just share that. And another example I'll give is the kind of the cellular and molecular mechanism of one of the organism which we have isolated from the uranium ore deposit in Dobiasiat, Meghalaya. So this isolation and identification was done in collaboration with Dr. Joshi's team at Northeastern Hill University, Shillong. So we have got that strain into our lab, and we have started, doing the experiments. So to start with the Anabina torulusa strain, we expose these cells to uranyl carbonate, that is a form of uranium in sea. So we see the kind of, the uranyl binding by 65% within 24 hours. And this uranium was getting co-localized with the polyphosphate bodies. Now, after these uranium is bound to this, so we need to extract it out to regenerate the cells. So we do that by 0.1 normal HCl wash. And when we were done that, we saw these kind of the pit-like structures which were on the surface. Now with multiple experiments, we could see that although the filament is intact by the chlorophyll fluorescence, what you can see on the right, but yes, there were these kind of pit-like structures which was not there when we were giving the wash with HCl to the control cells without uranium exposure. So with all kinds of experiments, we could prove the presence of acid-soluble surface-associated polyphosphates for the first time in this organism, which comprised of only 20% of the total polyphosphate content. Now, when we went for extending the uranium exposure, you can see by 24-36 hours, it starts lysing. By 72, 96, and by 5 days, you see complete lysis and this formation of the akinines, that is the spores. But on further exposure, by 8 days, 9 days, and 10 days, you start seeing the germination. This is a germling, and this is the germination, and it becomes complete green. And on the right side, you have the corresponding flask. So if you see at the top of the flask in 8 days and 10 days, you can see the specks of the cyanobacterial cultures, which are at the top, and the cell debris at the bottom. Now, what all was going on, we tried to find out, and we could see that, as I mentioned, uranium gets co-localized with the polyphosphate bodies. Now, we do all these experiments in the minimal media without phosphate, because otherwise the uranium will precipitate. So in that case, the metal-stimulated uranium stimulates the poly-P degradation. So once the poly-P levels comes down to less than 50%, the cell lyses, the phosphate is released, the uranium is precipitated, and by 5 days you can see this kind of precipitates at the bottom, and at the top, the lysed culture of the anabina is on the top. Now, we characterized these uranium precipitates with XRD and the synchrotron-based X-ray absorption spectroscopy at RRCat Indore, and we could see that it is the formation of uranium-6 meta-autunite or uranyl phosphate mineral which is being formed. So, we could see these are the precipitates of uranyl mineral phosphates. And then, what was the reason for the regeneration? Now, the phosphate, whichever was there, was already utilized for the precipitation of uranium. Now, the level of phosphate was less than 0.2 micromolar. Only then there was an induction of the alkaline phosphatase. And you can see the zymogram here. So in the uranium-treated cells, you start seeing that kind of induction. And this is to see at the right side. So it is 0, 1 day, and 16 days. So this is the uranyl phosphate mineral which is settled at the bottom. And it starts feeding on the dead cells for the organic phosphate substrate, and then regenerating the entire organism. So these are the the events, and you can see the extraordinary resilience of this organism to evade the uranium toxicity or to deal with the metal toxicity. So the entire events. Now, coming to my second story, which is of the Chrysobacterium. This was isolated from the uranium ore deposit. This is one of the largest sandstone type uranium deposit. Normally we have in Jaduguda, that is the most exploitable mine, where we have around 300 to 600 ppm of uranium. But here we have almost 1000 to 1200 ppm of uranium. So we have this organism was isolated. You can see the profile of this. This was the plate picture, and this doesn't have any kind of locomotory structures. And so for finding out these kind of adaptive features or any kind of further study, we started doing the complete genome sequence of this organisms, and you can see on the right we had the gene abundance related to DNA repair and oxidative stress, the transporters, the secretion, metal efflux. So Now, how does it resist the uranium? The first one, we could see that it was a lot of phosphatases were found in the genome. And we could see that this is the phosphatase activity. And what it does is, when an organic phosphate substrate is given, it cleaves and releases the inorganic phosphate, which precipitates out uranium or any other heavy metal which is given to that. So this is what is bioprecipitation means. And we could see that at all pH at 5, 7, and 9, it was able to do a superior uranium bioprecipitation was seen. And here you can see the uranium precipitates, which was intracellular at pH 5 at the cell surface and extracellular at pH 9. Now, when you have such kind of precipitates which are kind of an extracellular, you have to contain if you want to use it for application. So what we did was we immobilized these organisms in the calcium alginate beads, and then we have kind of passed the uranium carbonate solution and uranium nitrate solution. So here you can see after 24 hours, when 1 millimolar uranium exposure was given, the entire the precipitates are formed within the calcium alginate beads, and this is how it shines kind of green when under UV fluorescence, and that is kind of characteristic of the uranyl phosphate mineral. So that was also characterized by the XRD. Now also we saw the uranium binding by the cells as well as the biofilm. Now this organism, as you can see on the left side, this has one property called as gliding motility. Now we saw that there were no locomotory structures, and it was able to move with the help of the gliding motility genes. And it is a multi-protein complex, out of which some of them are secreted by something called as type 9 secretion system, which is only limited to the Bacteroidetes phylum. It is not found in any other any other phylum. So here you can see that kind of the assembly of these multi-proteins on the cell surface. It allows the secretion of the adhesins, which allows, again, facilitates it's for binding to the substrate. So this is kind of, as compared to the control, you can see in case of the uranium, it forms a lot of biofilm, and that is how it kind of, so here you can see the biofilm. It is a dose-dependent biofilm formation, and you can see the uranium association with the SEM EDX data. So you have the biofilm which is trapping uranium, evading the uranium toxicity. So this was another mechanism. Now uranium is a redox active metal. I'll take 2 more minutes. Yeah, so it is a redox active metal. We try to see a lot of free radical generation. And the membrane permeabilization and the protein oxidation in presence of uranium. Now, we started doing the transcriptome and the proteome in response to uranium. So you can see the upregulation of all these antioxidant enzymes like SODs, catalase, and thiol peroxidase, and which was also correlating with the proteomic signatures. Now, there was, in fact, apart from uranium, the soil also had some other heavy metals, and we needed to see that what other kind of, protein or any kind of, mechanism is there to take care of that kind of toxicity. So in that case, we studied about this PIB ATPases. So these are the metal transporters which are on the membrane. At the cost of the ATP hydrolysis, it effluxes out the heavy metals. So we saw the induction of PIB under metal stress, and this was a membrane protein, and it was quite difficult for us to kind of, purify this. And we could purify based on ATPase metal stimulated activity. We classified it as zinc, cadmium, and lead translocating PIB ATPase. And we could see when we overexpressed in E. coli cells, it could tolerate higher concentrations of cadmium and zinc. And so in case of, we made several site-directed mutants for the metal binding residues. So we could see the wild type kind of, hardly accumulated any kind of, the metal like zinc and cadmium, whereas the mutants showed higher accumulation in the cells, giving us an idea that yes, this organism was transporting or effluxing out the heavy metals. And so these are all the adaptive responses. If you see the metal transporters, the type 9 secretion system, the polysaccharides or the biofilm, and the biomineralization, the binding, and the antioxidant response. So this all together allows this organism to survive and perpetuate in that kind of uranium ore deposit, which is an extreme environment. So with that, I come to the end. So this is our team. So the work which I showed you, mostly Pallavi has done, and also Devanshi has done. So these are all my, the vibrant, my extended family actually. And this is what last year we celebrated 75 years of bioscience research in Department of Atomic Energy. So going by Dr. Homi Bhabha's dream of peaceful uses of atomic energy. So it started in 1948. So particularly in BRC, for the bioscience group, we have almost 200 plus scientific officers who are working in various aspects of food, health, agriculture, basic sciences. And in the middle, you can see Professor R. Chitambram, who was former AEC chairman and also chief architect of Smiling Buddha, that is the nuclear test at Pokhran. And unfortunately, we lost him this year in January. So, yeah. So with this, I end my talk. So this organism, as I mentioned, that it has this gliding motility. And so this gliding motility allows the cells to organize, and it forms this kind of spreading edges. So at the spreading edges, when we take the impression and have done the electron microscopy, you can see the cells arranged in these kind of hexagonal lattices, and these diffract the light, which causes this kind of structural coloration. So this is this work is being done by Rashmi, and she will be presenting in her oral presentation. So with this, I come to the end of my talk. Thank you so much. And I'm the only one who is preventing you from going for lunch. So I'm really sorry for that. Thank you so much.
Very, very interesting talk. Very nice, colorful microbes. Maybe they have some role in the environment for the bioluminescence.
Exactly. So, we are trying to
wonderful.
find that.
Thank you very much.
Thank you, ma'am. It's just not you. We have two more speakers to go for. Then we have the lunch. Now I request the chair and co-chair to kindly share a token of gratitude to our speaker, Dr. Cyril Acharya, the scientist at BARC Mumbai. Can we have a huge round of applause, please? Thank you, ma'am. Over to the chair and co-chair.
Yeah, ma'am, okay.
Our next speaker is Professor Naveen Kumar Arora, a renowned researcher in environmental microbiology and biotechnology at the Babasaheb Bhimrao Ambedkar University at Lucknow. He is a professor of environment science, a former dean, and the current president of the institution's Innovation Council. Dr. Arora is globally recognized for his pivotal work on plant microbe interactions and sustainable agriculture. His immense impact is highlighted by his consistent ranking among the top 2% most cited scientists in the world for 5 years in a row, the Stanford Elsevier. With over 150 research articles and 54 edited books published by Springer, he also serves as the editor-in-chief of the Journal of Environmental Sustainability. His excellence has been recognized with multiple awards, including those from the Honorable Governor of Uttar Pradesh and the Ministry of Agriculture, Uzbekistan. So, welcome, Professor Arora, and over to you.
Thank you. Thank you, Chair and Co-Chair. At the outset, I would like to thank the organizers, specifically Professor Kuhad. Thank you, sir. Professor Prince Sharma, Professor Namita, and all the organizers from the university, Dev Bhoomi University, for inviting me to give this presentation. So we are already running late, so I will try to be very quick and very fast to summarize my presentation. So I will see human population. Yeah. There is need for this. इसमें नहीं आएगा, यहाँ रखो. So we all know how human population is still increasing, and the population of our country is also increasing, and will be 8 billion by 2025. So the food security is one of the biggest issues which we are facing at present and in near future. To meet the demand, we need to increase the food basket by 70% by 2050, and that too by a sustainable manner. So food security is the second most important sustainable development goal. All the goals are as important as any other, but this food security is SDG 2, which is also known as zero hunger. So challenges to food security are many, like unplanned urbanization, land degradation, requirement for biofuel production crops, climate change, soil quality degradation, and increase in soil salinity as well. So this map shows how the land degradation is happening around the globe. The red zones are the sites of very high severe land degradation, and you know, North India is also red. So we are suffering from very high severity of land degradation in North India, particularly the Ganga basin, which was supposed to be the most fertile basin of our country, and now it is almost very badly affected by land degradation and soil salinity. So I come from Lucknow, Uttar Pradesh, the part of Ganga basin, and we have huge areas of degraded ecosystems, degraded agro-ecosystems with almost zero productivity due to soil salinity. So this slide I have taken from Syngenta, and you know, it is from 2018. And you see 1 out of 9 people were undernourished in 2018. But in 2024, I again checked the data, it is 1 out of 5 now. And it is, I am quoting United Nations, UNEP, 1 out of 5 people are now suffering from food insecurity. So we have come down as far as food security is concerned. We are going down instead of going up. So what are the major reasons since 2018 and 2024 that from 1 out of 9, we are now 1 out of every 5 persons are suffering from food insecurity. The major reasons were the COVID-19. After this, 2018, there was COVID pandemic. Then the wars going on, Russia-Ukraine conflict, the Middle East conflict, and also the land degradation due to climate change. See, there is a huge gap, and that gap is increasing regarding if you see the food demand and what is the actual food production. And in fact, we need to go for sustainable methods also, because land degradation, one of the biggest reasons is the use of agrochemicals like pesticides, fertilizers, intensive agriculture. In Tarai region of Uttar Pradesh and even in Punjab, farmers are taking 2 paddy crops in one season. So once I was in Lakhimpur district of Uttar Pradesh, in March, they were sowing paddy and taking out water from the ground and putting fertilizers and pesticides in March, when that is the most driest part of the year, March, April, May, and paddy cultivation was going on. Day, night, they were pumping the water from the ground and I could see the farmers using pesticides and fertilizers and all those things. So this is how the food security is all about. You see zero hunger. If you see the global scenario on the left, around 40% 40% population is suffering from food insecurity. You see the scenario of Asia, it is almost similar to the global scenario. Almost 35–40% people suffering from food insecurities, some from very severe food insecurities and some moderate to severe. In Africa, 80–85% people are suffering from food insecurities. In North America and Europe, only 5–10%, and in Latin America, again around 50–60% people suffering from food security. So what I want to show from this slide is that how unequal we are. Globally, the human race is so unequal. And if you talk about food security, this is the scenario. If you fetch the data for health security, water security, energy security, it will be same. So this is how unequal we are, and we are trying to achieve the targets of sustainable development goals by 2030. So we need to work more on these aspects. We need to work together. Now you see climate change, which is impacting the land, it is causing land degradation. Two sectors, energy and agriculture, account for 92% of CO2 emissions. Only two sectors, energy and agriculture. So we need to go for sustainable energy, more green energy sources, and also sustainable agriculture if you want to control the CO2 emissions. We all know, maybe students behind, I don't know if they know or not, that Paris Agreement, it was decided, and all the countries are signatories to Paris Agreement, that we will keep the temperature below 1.5 degrees centigrade. Means if we compare it with pre-industrial area, we will not allow it to rise above 1.5 average global temperature. But we have already breached the Paris Agreement. So Paris Agreement is gone, and I am quoting United Nations Secretary General, that Paris Agreement is gone. Now we should look beyond Paris Agreement, and we should look for 2 degrees centigrade target. And 1.5 degree centigrade target is causing such an havoc. So you can imagine what 2 degrees centigrade will cause. And now we are shifting the goalpost from 1.5 to 2. And we are not going to achieve any of the SDGs by 2030. Again, I am quoting UN Secretary General. We are not on road to achieve any of the SDGs out of the 17 SDGs. So I am going to talk about sustainable agriculture. I am not going to talk about sustainable energy and green energy, but I would like to focus on my work on sustainable agriculture. So you see, the silver lining is regarding sustainable agriculture, the global market for biofertilizers is increasing by 12%, compound annual growth rate. Global market for biopesticide is increasing by 15% compound annual growth rate. And global market for bioremediation is increasing by around 10% compound annual growth rate. So microbes are playing very important role in sustainable agriculture and bioremediation, and they are going to play a huge role in the coming time. As yesterday, day before yesterday in the inaugural session, Professor Kohad and Professor Prince Sharma were pointing out. So this will be, the future will be an era of microbiology. Already this is going on, an era of microbiology, but future will be, there will be more requirement for the use of utility of microbes in agriculture, in environment, in bioremediation, in enzyme, in medicals, all these branches. So I have just depicted how it is growing in the case of agriculture. So now I come to what we are doing in our laboratory. So this you see on the extreme left, these are the degraded ecosystems in Uttar Pradesh, central Uttar Pradesh. I am coming from Lucknow and Kanpur region, Unnao, Lucknow, Kanpur, Raebareli, these districts, central Uttar Pradesh districts. So these are completely degraded ecosystems. I will show you some more pictures. And we have transformed them from completely degraded to the green ecosystems, to the productive ecosystems with productivity as good as Punjab. So salt stress is happening in these are saline sodic soils. I will not go in the introductory aspects. I want to focus more on what we are doing. So soil salinization and soil sodicity is increasing day by day around the globe. You know, in India it is increasing globally, and it is one of the major factors impacting food security now. See, this is the global distribution of saline soils. Every continent is affected, And this is the global distribution of saline soils in India. Except for the Himalayan states of Jammu and Kashmir, Himachal Pradesh, Uttarakhand and Northeast, all other states have the issues of soil salinity and sodicity and degraded soils which are increasing every day. So this is how we classify the saline soils on the basis of electrical conductivity. If it is less than 2, then the saline soil is non-saline. It is 2 to 4, then slightly saline. 4 to 8 is moderately saline. 8 to 16 is very saline, and more than 16 is highly saline. Now we in Uttar Pradesh, where I am coming from, central Uttar Pradesh, the soil is very saline now. So we have isolated the microbes from these very ecosystems, saline ecosystems, and now we know these microbes, which we also known as plant growth promoting microorganisms. They are now being used for bioremediation, bioaccumulation, biodegradation, stress management in crops, and combating climate change. See, these are the original pictures of those fields nearby Lucknow and Kanpur. So we have adopted 10 villages, 5 in Lucknow, 5 in Kanpur district, which are suffering from soil salinity like this, zero productivity. So farmers in this region are very poor because of land degradation. They are suffering economically, their education of their children is affected, health is affected. So you see, land degradation is causing all the sustainable development goals are in problem. Means economic, health, food, security, gender equality, everything is affected because of land degradation. How food security, of course, because there is no productivity, food security will be affected. Now because of land degradation, their economic status is affected. Because of economic status, so their health is affected, and inside, if their economic issues are there, they will take out the girl child from the school first. And food security also, the women in the house will feed in the last because of the food issues, and they will, okay sir, I will take 5 more minutes. So we have isolated microbes from these soils. We held several meetings with them. You see, these are the pictures from those regions only, and we have isolated these salt-tolerant microorganisms from these soils, which include pseudomonads, bacillus, rhizobium, and these are different salt-tolerant microorganisms. I will just quickly go through because only 5 minutes I think I have. Mainly pseudomonads, you see, fluorescens, entomophila, putida, bacillus subtilis. These are all isolated from these very ecosystems. And rhizobium also. One study which I am not showing here, we conducted in these soils was 80% of the crops which the farmers are still growing are lacking. I am talking about the legume crops are lacking nodulation now. No nodulation in 80% of the legume crops grown in this region. So that is another issue. So what we have done, we have checked the mechanisms, how these microbes are combating salinity, all published in several good impact papers, good impact journals, and we have tried to find out which metabolites are working, which are helping, which mechanisms, and we could focus on mainly on 2 of them. Exopolysaccharides are produced across the genus. Means pseudomonads are also producing exopolysaccharides, huge amount of rhizobium, bacillus, all are producing exopolysaccharides in huge copious amount. So exopolysaccharides help them in various ways. They help in enhancing water uptake, nitrogen nodulation, in case of rhizobium, help in mitigating salt stress, and they are also useful in quorum sensing. So we also, during the course of the study, detected a novel exopolysaccharide known as malzitose, which was present across the genus, which was not only confined to one genus like Pseudomonas, it was present in Pseudomonas as well as Rhizobium. Malzitose, which we reported as a new exopolysaccharide from these bacteria from the saline soils. That means it was playing some important role. These are the SEM analysis, and this is all published. You can download my papers from my ResearchGate or Google Scholar. We also checked some of these strains against phytopathogens, because some of the phytopathogens like Microphomina and Alternaria were also prevalent. in these soils. You see the zone of inhibition here. We selected, these were selected salt-tolerant biocontrol agents. You see under high soil salinity, they are showing biocontrol against the phytopathogens. So then we developed the products. All these names are in Hindi because we are coming from Uttar Pradesh, Lucknow region, Hindi. So we prepared bioinoculants using these microbes with the help of these microbes and distributed them to farmers. And these are the names like we gave them in Hindi to popularize. And now farmers, you know, they know their names. They come to our lab and ask for ki, sir, hame wo जय पोषवर्धक चाहिए या ट्राइको रक्षक चाहिए, because they know the names even. As we did initial feed experiments on legume crop, chickpea, and applied these bioformulations there. In some cases we also added metabolite and it increased. So you see it was several fold increase in the productivity of the chickpea when we applied these bioformulations. We also checked on oilseed crop in presence of pathogen. This is how the crops get affected with the pathogen, do tube assay, then pot assay, and then we go for the field trial. You see, here is the this completely degraded field, farmer's field, that is the control, and here when we applied the bioinoculants. This is the picture of the same field. So we got some novel rhizobial strains, also salt tolerant ones, which are phosphate solubilizers, siderophore producers, zinc solubilizers, IEA producers, EPS producers, and also nitrogen fixers. During the course of the study, we developed a new method for siderophore detection. This is a highly cited paper where we can detect siderophore on microtiter plate, 96 strains at one go. In micro ml you need this media, and you can check the siderophore production in 96 or 180 well plate. Within 24 hours, very little amount of media is required, and you know, chrome azurel S dye is present in that media, which is a pollutant if we drain it down into the environment. So very little amount of media is required, and you can check 96 or 180 strains in one go in 24 hours. So we checked this formulation in lettuce. I will skip some slides because shortage of time. Then we have this, we had this MOU with Shri Bioaesthetics for the production of bioinoculants, and this Hello Biome is now produced by Shri Bioaesthetics. It is a Hyderabad-based company which is producing our bioinoculant. This is a consortia-based bioinoculant for saline ecosystems, for remediation of saline ecosystem, having 6 bacterial strains in it with diverse activities like phosphate solubilizer, zinc solubilizer, nitrogen fixer, and others. Then we now we are doing experiment with more than 400 farmers in wheat. In case of wheat, you see, this is the control, and this is after we used consortia-based formulation. This equivalent to Punjab. This is the control. This is the productivity equivalent to Punjab. And this is the drone picture of the same very field. On the left side is the where we have applied our bioformulation. On the right side is that control. So from almost negligible, we are getting productivity equivalent to Punjab. Now these farmers are growing in these very fields where nothing was growing. They are growing some vegetables also. We call it backyard farming or kitchen garden farming, which is making their food basket rich, and they are also selling these vegetables which are very salt sensitive in comparison to other crops. Some of these crops which they are growing in their backyard in this restored ecosystem, soil ecosystem. So the pH has declined, EC has improved, organic carbon has improved, microbial biomass has improved in the soil, which was almost white. Now it is, I have not shown the data of metagenomic analysis we have also done. So from complete degraded ecosystem, it has become, within 3 or 4 years, it has become a very highly productive ecosystem with productivity as good as any fertile ecosystem in our country. And now we have signed another MOU with Shri Ram, DCM Shri Ram in Delhi, for the production of these bioinoculants. So if you meet the farmers now, those who are having kacha ghars, now I have pakka ghars now because of this restoration of soil. Their economic status have improved, and almost 400 farmers in almost 5 villages are with us in Kanpur region, and some farmers are in Lucknow region as well. All this is, in the end I would like to just finishing 2 slides. This is the book series which we are publishing. I am series editor of this book series. We have already published 60 volumes since 2018 in this book series, 60 volumes. This is Scopus indexed. So I invite all of you to participate in this book. These 60 volumes are from different parts of the globe, and it is Scopus indexed. Within 7 years, we have been able to manage 60 volumes. And this is the journal Environmental Sustainability, which we also started in 2000. I am the editor-in-chief, founder editor-in-chief of this journal, Environmental Sustainability. It is having an impact factor of 3. So we started this journal in 2018, and now it is having an impact factor of 3 since last 2 years. So again, I invite all of you to communicate your papers, your research in this journal. This is my research team from my lab since last 7 or 8 years, since this the work which I have presented. Thank you.
Thank you so much, Dr. Arora, for that really inspiring talk. The hosts, organizers would like to present you with a memento.
Yeah, I request the chair and co-chair to kindly present a token of gratitude to our eminent speaker. Students at the back, please be active. Can we have a huge round of applause for our speaker? Is it a time to listen from our education partner, industry partner, sorry, from the high media? Over to chair and co-chair.
So our last speaker of the day is Dr. Sujata Hajra, AGM Molecular Biology, High Media Laboratories. Dr. Sujata, you have 10 minutes.
So can you please put it in the slide? I don't know. It's like how audible I am. Thank you so much, Chairman ma'am, co-chair sir. And I'm sorry for keep you waiting. You people are must be very hungry because I myself is very hungry. Okay, so I will not take much time. So, so far we were here listening that how the research is going on for the environmental samples. So, I am representing High Media, and High Media's molecular biology segment, which is under ImprovementMicrobial brand. So, if anyone is interested, we are having a booth over there only with all our like literatures and instruments available right now. So, should I get that mic? Better? Head mic? I don't know. So we actually pan India as well as abroad, we are exporting our products in our molecular biology section, which is all under ImprovementMicrobial brand. And we have our regulatory compliances as per the quality is concerned, because High Media is a 50 years old company. It's fine. So here, okay. So quality is the basic criteria for any high media product. I guess everyone knows, because high media's microbiology media is there since last 50 years. And for molecular biology also, we have this regulatory certificate and compliances for maintaining the quality of the product. So our journey started long back with extraction kits and PCR machines, automated extractor, as well as we are having sequencing services also. So in short, we want to have a complete solution for molecular experiments for genomics, proteomics, automation, and sequencing. So obviously, so far we were seeing that scientists are isolating organisms, soils, water to identify that. So the basic workflow obviously is the transportation and pre-processing along with DNA RNA extraction, PCR, and downstream applications, meaning doing the sequencing of the isolated microorganisms. So for sample transportation, at present what we are having from HyGenoMB, you can see I am not going to read it out because we are having for stool, soil, as well as this tracol is actually very can be a very interesting thing for you people who are having collaboration with one lab with another lab. So in extraction we are having silica based column method as well as magnetic bead based automated method. So these machines like this InstaNX Max 16, it is in display in our booth. So if anyone is interested, they can visit our booth, and it is a very small machine, very sleek machine. Its weight is under 8 kg, and so 16 samples can be processed at one time. It is magnetic bead based, and this kind of pre-filled cartridges come with the for the sample processing. So for soil we are having kit, for water sample we are having DNA as well as RNA kits. So for MAC24, it's like the sample, if you see the cartridges, so there the starting sample volume will be more. So when you want to have more sample volume, you can go for the MAC24 machine. So these are some data to show you that soil DNA, whatever we have done with this magnetic bead based machine, you can see the extraction, the purity is quite good, and the yield is like always we keep a competitor to check our quality. So for the water, so for water extraction, there are either you will be going for with filtration or without filtration. So I'm not going into all the detail of this. And so filtration, after filter is done, you can put the filter paper in the tube with the process with the lysis tape. And here we can show you some E. coli spiked with water sample to show that how we are checking that whether it is PCR-able DNA is extracted or not by this machine. Several other pathogens also we have spiked and we have checked. And so we have this pathogen detection, Legionella, Salmonella, E. coli, Vibrio, and total coliform. E. coli and Salmonella, we are having quantitative PCR to identify the LOD. So we provide the quantitative standard. So this viral panel is being developed during COVID only because we got this requirement that from the sewage water to identify COVID. So these are the panel which we have developed, hepatitis A, hepatitis B, as well as all these entero, astro, and rota. I'm just running through the slides because I don't want you people to be like, already it's 2:30. So Salmonella quantitation probe PCR and E. coli probe PCR, already I told that we provide the quantitative standard. And these are all the data. These are all our in-house data generated using our own machine, own reagent, own kit. And like AMR is right now the biggest threat at present what we are having. So for that, under that One Health approach, whatever we are having in AMR, so these are the five basic MRSA, VRE, and colistin, CRE, ESBL. We have these PCR kits, quantitative PCR kits, real-time PCR kits. If anyone is interested, I am just repeating it. Please come to our booth and get more information. And you can visit our website, highmedialabs.com, where under the molecular biology segment, you can get these details of this, about this AMR quantitative real-time probe-based PCR kits. And this is the latest thing. So for point of care for NG Carba, we are having a collaboration with NG Biotech of France, and where you can have the 5 prime genes to be detected for carbapenem resistance, and here direct, not only from culture, but from direct urine and blood culture also you can detect using this NG Carba 5. So everyone in my previous, in this session were identifying the microorganisms, so we have started our sequencing services, and Sanger NGS along with bioinformatics we have in our bucket. And so for microbiome service, if anyone is interested, I request you to please scan this QR to get more information about this microbiome services, whatever we are at present providing to the customers. So I thank you all for be patient with me, and thank you so much for giving me this opportunity.
Thank you so much for keeping time, Dr. Sujata.
Now I request the chair and co-chair to share a token of respect to our industrial partner, High Media. Thank you to the High Media being the industrial partner for this international conference for us. Now I request the Dean Research and the organizing secretary of this conference, Professor Nabil Ahmed sir, to please join us on dais to share a token of gratitude and respect to our chair and co-chair of this session.
Okay, before that goes on, I would like to really thank local organizers, Nabil and Nirjara, and then as well as AMI for giving us the opportunity to chair and co-chair this session. Thank you very much, and thank you all attendants for being over here. Now it's session closed, but then if you have any specific questions for any speaker, please catch them during the daytime. Okay, thank you very much.
Yeah, if anybody has any questions, you are open to have those questions during the lunch time. Now I request Nabil sir to please share a token of respect to our chair and co-chair of this session. Audience, can we have a huge applause for our chair and co-chair for being giving this session to us. Thank you, ma'am. Thank you, sir. Now I request everyone to kindly proceed for the lunch at their respective places, and we will be back here in the next 30 minutes. So sharp at 3 o'clock, we'll be looking at you here for the next session. After the lunch, we have the session here. The session 8 would be at main pandal, that is microbes in agriculture and crops involvement. The session 9, microbes in traditional and contemporary food products, would be at Ayurveda Seminar Hall. Session 10, molecular frontiers in the microbial biotechnology at the Seminar Hall, first floor in the main building. The concerned person can respectively join their sessions, and the one who has to join for the main pandal, we're looking forward to see you at 3 o'clock sharp after the lunch in the main pandal. Thank you. सर और नमिता है ही नहीं, वो वहाँ पोस्टर प्रेजेंटेशन So good afternoon, everyone. I welcome you back after the lunch on the day 3 of this conference. In next 2 minutes, we are starting with the plenary talk. By the time, I just request everyone to kindly be seated at their respective areas. Thank you. शिवा कुमार होंगे, डॉक्टर शिवा कुमार, डॉक्टर अलका शर्मा और डॉक्टर सरकान सलकाट यही तो है बेटा। हाँ, अभी शुरू होने से ही हो रहा है। उनकी प्रेजेंटेशन खोल दी ना? हाँ, होंगे बस ठीक। उसके बाद जो सेशन है उसमें सिर्फ 3 लोग हैं, ठीक है? हाँ बस उनको तो बुलाने हैं, उनकी प्रोफाइल थोड़ी बोलूँगा. We need to start at 4:20. Sorry, 2:20. हमें 1 hour में. So good afternoon, everyone. I welcome you once again to the day 3 of the conference, to the 66th Annual International Conference of an Association of Microbiologists of India, supported by DBT, ANRF, DRDO, CSIR, and UCOST. Without a further ado, I request the chair of the session, Professor Dr. R.C. Kuhad, the founding chairman of AMSC, to please join us as chair for this session. Can we have a hidden applause for our chair for this session, please?
अभी खाना खा के आए हैं ना, तो तालियां इनकी बजती नहीं है कभी भी. और देखिए कैसे स्टैगर्ड बैठे हुए हैं आप लोग. ये पीछे वाले लोग जरा कष्ट करें, विद्यार्थी लोग आगे आएं. और जो सीनियर आगे बैठे हुए ना, इनको पीछे भेज दो. Come on, come on. अरे आगे आओ, आगे आओ. Don't feel shy. You'll be able to hear more. You'll be more serious. You'll be able to listen carefully. You'll be able to learn. चलिए आइए. अरे आगे आइए, नहीं तो मैं वहाँ आऊँगा फिर बुलाने आपको. देखिए या तो आगे आ जाइए या हॉल छोड़ कर चले जाइए. ऑप्शन आप लोगों के पास है. ये आपसे भी मेरी रिक्वेस्ट है, लेडीज एंड जेंटलमैन, प्लीज कम फॉरवर्ड. देखिए सीनियर scientists, senior academicians has come over here from almost 450 kilometers to deliver his findings, to share his findings. और आप लोग जो है ना इतने सिंसियरली उनको सुन भी नहीं सकते, देख भी नहीं सकते, तो इट्स ए पिटी. और समवेयर आपकी गलती नहीं है, ये जो हम टीचर्स हैं शायद हमारी गलती है कि हमने इनको कभी सिखाया ही नहीं. हम लोग खुद अपनी लैबों में काम करते हैं, रिसर्च पेपर लिखते हैं, इम्पैक्ट फैक्टर बढ़ाते रहते हैं, लेकिन स्टूडेंट को ये नहीं बताते कि थोड़ा सा ध्यान दीजिए, बैठिए, ये एटिकेट्स भी होते हैं। चलिए शुरू करवाइए।
I request my co-chair as well, Dr. Namita Singh, the General Secretary of AMI, to please join us on dais. Students, it is a time to listen to an eminent speaker. I'm telling you, he's a Padma Shri, Dr. Udas Ullah Khan. The rest of the thing will be taken here by my chair and co-chair. The profile is in these folders, sir. Over to you.
Good afternoon, everybody. Before we start the session, I have been given the responsibility to share with you that some of our colleagues, some of our great scientists have passed away. And I request each one of you to stand for a minute to pray God so that their soul may rest in peace. And of course, their demise will be a great loss to all of us. And we need to remember them, their contributions, and if you could preserve their contributions, I think that will be a great homage to all of them. And I just share with you Dr. Sisinthi Shivaji, who served as the Director Emeritus and Senior Scientist at the Professor Brian Holden Eye Research Center, Hyderabad. Dr. Subbanna Ayyappan, Padam Shri Awardee, and who has been DG and Secretary to ICR and Ministry of Agriculture. Also has served our President of Association of Microbiologists of India, Professor Ajit Verma, who was superannuated from Jawaharlal Nehru University from his regular services and then joined MIT University till the last breath. And Professor Jyoti Prakash Tamang has been a renowned food microbiologist from Siliguri. He has been former, say, as a Vice Chancellor also of Sikkim University. And then Dr. Rishi K. Bahel, who has also been a great scientist from Rajasthan University and he also served in Haryana Agriculture University, Hisar. And I think, if I'm not wrong, Professor Sani also left us this year only. So all these 5, say, great scientists, we are missing them today. Please mourn for a minute. I'll appreciate it.
Thank you, everyone. Kindly seat at the respective place. It is the time to introduce the plenary speaker now, so over to my chair.
Just to start this session, let me invite Dr. Asadullah Khan, who is faculty at Aligarh Muslim University. He is a distinguished professor in the Department of Biotechnology at Aligarh Muslim University, a leading researcher in the field of molecular microbiology and infectious diseases. Dr. Khan has made significant contributions to understanding antimicrobial resistance, pathogenic mechanisms, and novel therapeutic interventions. Dear students, with an extensive background in biomedical sciences, Dr. Khan's research primarily focuses on multi-drug resistant pathogens, molecular epidemiology, and the development of innovative strategies to combat bacterial infections. His work has not only advanced scientific understanding in these areas, but has also influenced public health approaches to infectious disease management. He is an educator and a mentor. Dr. Khan has guided numerous research scholars and postgraduate students, fostering a culture of academic excellence and innovation. He has a lot of credentials in his credit. Without taking much time, let's invite Professor Asadullah Khan. Please come on dais and share your findings. Thank you very much.
From me, I'm delighted to be here as a speaker with the chairs of Professor R.C. Kohart. So, I think we have a long association with Professor R.C. Kohart, sir. I still remember, just in a second, one of my PhD students, he is an examiner for PhD students, and he was so busy. I think it's 2009 or 10. This was the first time in a history it happened that I took my students, get permission from Vice Chancellor, and he identified some places in Delhi. Then we had a Viva. Sir, you remember these things. So we had a Viva there in Delhi. Well, thank you, sir, to be here. Today, I'll be talking on a very important area like AMR. Everybody knows what the AMR is because many of you not much aware about the antimicrobial resistance. So I'll just give a few brief introduction about what the AMR is. Antimicrobial resistance means like no antibiotic left to treat infection. We call it multi-drug resistance for students. The current scenario of AMR, if you look into, the global scenario is like by 2050, at least 10 million people will die every year. This was the old report. But I will share you some recent report by Lancet report, which published in 2024. Now, if you look into this, this report, you see the, can I have this marker? Pointer. So if you look at this data, in 2019, we have almost 5 million people died because of antimicrobial resistance, and almost 1.27 million people died because of the bacterial antimicrobial resistance, right? And that goes a little down, but now it is expected by 2050, we have 80 million people will die because of antimicrobial resistance. That's a big problem. Now, if you look at this plot, you see that the landscaping of from like neonates to 70 years old people, you find the increasing trend of the antimicrobial resistance, where you will find the death rate is increasing by elderly people, whereas in the neonates, the death rate is not increasing, but mortality is not increasing, but the ARGs are increasing continuously in the neonates also. We'll show some of the data. Excuse me, can I have this pointer? Is it working or not? Huh? Acha, okay. So, if you this brown one, this is age of 15 to 50 years people which are stable next 25 years, but the spread of ARGs is continuously increasing as you can see the data. Now, these are the priority pathogens where you find the carbapenem resistant A. baumannii, resistant enterobacteriales, rifampicin resistant tuberculosis bacteria, and aureus, baumannii, E. coli, and Klebsiella pneumonia. So these are the priority pathogens basically which we need to focus on in order to control the infection caused by these bacteria having a lot of resistance gene. This is the challenge what we are facing especially in the Indian system or Indian subcontinents, fragmented health system. You know, see in the time of COVID what happened, inadequate intensive ICU staff. Then we have inadequate universal wash access. We don't have any clean and hygienic water for most of the population of the country. Inadequate intensive care unit facilities and overcrowded ICUs. So these are some challenges which we are facing for controlling the infection. Now, this slide is showing that how the AMR spread is interlinked with the, we call One Health, that is animals, hospitals, and the drinking water or the sewage water. So we try to understand the transmission dynamic of this environment, human and the animal. If you understand the transmission dynamic, you can try to find out some checkpoints so that we can control the infection to some extent, and that is the best way to control the AMR, controlling the infection, rather than focusing on ARGs control, because it is not in our hand to control ARGs. It is a bacterial internal defense mechanism where the bacteria have the smarter than us to develop some new gene, acquire some new genes to defend. You know, this is just, I'm taking you some global view of livestock resistance, where you find how the livestocks carrying the ARGs, and the ARGs is spreading. This is alpha diversity. This is the whole metagenome sequence analysis of animals samples, where we found that I think the slides is not very clear, but I think most of the macrolides, aminoglycosides, almost all class of antibiotic, we call pen drug resistance sort of, should have high resistance in case of I think this is not visible, in case of a turkey, pig, and this kind of this is a broiler chicken. So this is increasing trend because of the continuous use of these antibiotics in the poultry or in the animal. Then we have, this is regarding the global distribution of MCR. MCR is discovered in 2015 in China, And this gene is causing the resistance against the colicin. So colicin is supposed to be considered as a last resort of antibiotic. After colicin, you have no antibiotic left available to treat the infection. But you find the, this is the pattern of ARGs is spreading in the, and we have got, at least they have got more than 200,000 samples, metagenomic samples done on these animals, and we found the livestock, how livestock is spreading. This is how they describe the fecal rhizostome. That also through slaughterhouse and through farmers, and they try to connect how the farmers and those who handle the slaughters can connect with this infection and how the infection is spread through these. So this is again showing this how the ARG is spreading different macrolides, aminoglycosides, and other kind of things. This slide showing the hospital, the patents abundance of antimicrobial resistance genes where the wastewater is involved. So wastewater is basically, our untreated wastewater is also problem because they are also showing the abundance of ARGs and the bacteria which are carrying the ARGs in that. So this is the global picture. Now, so my lab is focusing on surveillance metagenomics and trying to understand the mechanism, because I am basically by training as a biochemist. Probably you'll be surprised to know, because I'm not trained as a microbiologist, but I learned microbiology as a PhD and onward. And so we are in this field of antimicrobial resistance. We are working on some intervention approaches like vaccines, PDT, CRISPR-Cas inhibition of the biofilms. So today we'll just talk about some overview of AMR, what we are doing in my lab. So this is a paper we published long back, I think 2019 perhaps, yeah, 2018, where we found that NDM. So NDM was discovered in 2008-9 in India, and the name was given New Delhi methyl-beta-lactamase. And we found the NDM variants in the NICU patients are in ICU patients as well. And these NDM variants, which were discovered in 2008, later on published in 2009. Now we have more than 60 different NDM variants in the environment. Now see how it evolved frequently. This is the NICU data, neonatal intensive care data, where we have like NDM1, NDM1, then NDM4, and NDM5, and NDM7. And that too present in all like E. coli, Klebsiella pneumonia, Citrobacter, Enterococci, all kind of species, they have these NDM variants present in that. Now, just have a look at this slide. You see the different cefoxin, cefoxin, where the sulbactam is an inhibitor which is used. And sulbactam is an inhibitor. When the antibiotic is combined with inhibitor, the degradation of antibiotic is protected in the presence of inhibitor because it cleaves the beta-lactamase enzyme, which is main mechanism behind the resistance mechanism. But still you find the resistance goes to 1024 MIC. That shows you can compare like 1,000-fold higher MIC. And the real MIC is 0.5 to 1 microgram per ml concentration. And MIC goes to like 1,000-fold. That's very, very high resistance. Again, you have a Sedacia lepagi, another rare microorganisms found in NICU patients, like neonates in 2 days for a week or 2 weeks, days babies, and that shows very high MICs of these all drugs. So if the babies are being kept in incubator or in an ICU, they have got like high mortality rate because of the gene NDM in that. Again, we characterize certain NDM variants in an ICU where we found, we just shared the data that, see, don't go to the whole slide, but just look at the red one, 20 to 48 microgram per ml concentration of that gene. I mean, that's amazing. I mean, you will kill the man. So this is how, and then we further dissected through sequencing. We found that we've got ST33455 new strain, and that is the strain we published, we submitted that data into the Pasteur Institute for MLST, and this is the new MLST we found in this case. Now, this is a very pathetic situation of the two farmers from some nearby village, and they came to the patient, they admitted through UTI infection, burning sensation, fevers and all, and they got admitted, and after 3 weeks, they died. They keep on different line of antibiotics, imipenem, meropenem, and others, and ultimately they died. When we got the sample a week before they died, And we discovered that it is NDM6 variant. NDM6 is more severe variant than NDM1 or NDM4 variants. Now look at this, some of the studies from my lab that in 2014 we identified the NDM4 as the first NDM4, the first discovery from India, and that is the fourth variant which was first perhaps discovered in Italy and then in India. So somehow we have a lineage between these, the origin of these infection is spread from European country to India, maybe some medical tourism or something else. Then we have some metagenomic understanding about the antibiotic resistance genes of the sewage water. On the top C map, we've got a few sewage water from the West Bengal, Bihar border, then Delhi NCR and Aligarh itself. And we found that the discovery of the MCR 5.1. So the emergence of colicin resistance gene, especially MCR, in Indian system is increasing now. And this is the alarming threat for us. I mean, we should be careful because after the MCR, we don't have any drug left to treat the infection we have. We further identified some focus on the hospital settings, and we found that at least more than 300 different samples of colicin resistance. We found, we just characterized them, sequencing them, and then after getting sequence, we found that at least ST147 and ST357, which are most virulent strain of the Pseudomonas aeruginosa and Klebsiella pneumoniae, were found in this hospital setting. So this kind of an outbreak of these strains. And then we have taken the colicin resistance gene from publicly available data, metagenomics data, and we found in neonates 9.1 and 10.1 MCR were observed. So the emergence of these MCR genes are colicin resistance genes are one of the alarming signal in Indian setting, where the resistance is increasing and infection control is a big problem, and that is the reason the mortality rate is increasing. So they predict, WHO predict by 2050 we may have like, almost 2 million people die every year because of the antimicrobial resistance only, and that is to the bacterial antimicrobial resistance. Now, we also focus on one of the interesting study in the wastewater treatment plant, and generally wastewater treatment plant are supposed to treat the water and we can have a portable water. Although we don't drink that water, but we use that water for irrigation. All right, and we have checked the both conventional water as well as the conventional treatment plant as well as the advanced treatment plant where we have a UV system to treat infection and all. And what we observed that in both the plant, conventional plant, treatment plant and advanced treatment plant, we have got a lot of ARGs found at the portable water. And that is the link, as I have shown, one of the link between One Health approach, water, animals to human. This is how the infection is spreading, and the ARGs, antimicrobial resistant genes, are spreading from water to plant, consumers, animals, and so on. So we should focus on it, and I think there should be some policy should be made on it, and we must control this. And the carbapenem resistance gene, that is the imipenem and meropenem antibiotics, which is the sort of last resort of antibiotics, they all fail to treat infection because of these genes. So these are some key points. Discovery of MCR 9.1, 5.1, neonates, right, different ST types in the Indian system, Indian scenario. This is the global view, and the country view of the AMR spread, or ARGs spread. I have not shown more data because that make more time to me. So keeping that in view, we should think some interventional approach, because see, look at the GDP. We have $100.2 trillion GDP loss by 2050. That is a WHO report. Again, we have 10 million deaths by 2050, WHO report, costing around 66 trillion pound. Again, there's a huge amount of money to focus on. So my lab is focusing on such kind of thing. But the Lancet September 24 published a report, the 2 million deaths every year, and by 2050, 8 million people will die. But they also mentioned that 92 million deaths can be averted if we adopt some interventional approaches, control measurement, proper management, hospital management system, and so on and so forth. So from that perspective, besides so many approaches people are using, and my lab is also focusing on inhibitors, PDT, and nanotechnology, we are here today discussing about some vaccine approach, because vaccine approach is one of the best approach where you can reduce the antibiotic use and have more. So this is how, because of the limitation of antibiotics, we focus on the vaccine. So in order to prevent the infection, the vaccines can be developed, and we have here focus on the multi-epitope therapeutic vaccines. So you can have the prophylactic vaccine as well as the therapeutic vaccines. In multi-epitope vaccines, where we are trying to cover up the broad spectrum antimicrobial flora covered through different therapeutic protein. So we have adopted several approaches, proteomics approaches, and some, I'll discuss some of the other approaches where we identified some antigenic or epitope where you can have the best epitopes against the multiple bacteria like A. baumannii, Pseudomonas aeruginosa, Klebsiella pneumonia. So far we have covered at least 2 or 3 bacteria together to cover up these approaches. So these are recent paper we published on that. So we identified the multi-epitope vaccine candidate where against the Pseudomonas aeruginosa in immunoinformatics. Here we identified through proteomics approach the Fab protein, and the Fab proteins are basically the amyloid formation protein which lead to the biofilm formations. And if you control the biofilm formation, basically you can control the whole infection persistence basically in the infection site. Then we have another study where Klebsiella pneumonia and A. baumannii were taken into consideration, and we take the conserved multiple epitopes of the A. baumannii and Klebsiella pneumonia to identify these. So far we are just focusing on the bioinformatic approach through this, because we are moving to wet lab. We have not yet published any data on the wet lab because we've just started 2-3 years back this vaccine work on that. Earlier we are working on the inhibitors, beta-lactamase inhibitors, so we are not discussing that part today. So here we have a reverse vaccinology where we focus on the complete genome through genome-wide discovery of the antigenic epitopes, and these epitopes were identified from the bacteria, and then we use. Now we also use these antigens, we basically clone these antigens and then we express the protein through, and then after expression we get this protein to immunize into the animal. So this experiment we have not shown here because it is in a process, we have not shown that data. We also working into the cell lines, so looking the co-infection of the bacteria in the cell lines and where we find how the antigenicity is responding to these things. So this is how the case study is promising AMR vaccine, where A. baumannii, Pseudomonas aeruginosa, and K. pneumonia aureus, they show the target proteins. So identifying these target proteins is important because we have to look into the therapeutic approach, so we identified the virulence factors. And the best approach to get this virulence factor is basically the proteomics approach, right? Subtractive proteomics approach can also be used. And these are some targets we identified, and these vaccines are already in preclinical trials and phase 1 and phase 2. This is another study. We identified some multi-epitope vaccine on A. baumannii and Klebsiella pneumonia. This is in the stage of identifying the epitopes vaccines, and we have already constructed that, synthesized that vaccine, and we are in process of expression of the vaccines, and further in vivo studies will be performed. So this is some preliminary data we have shown here. Parrots, some softwares were used to identify these. So this is basically the epitope, the down panel, the colored bases. So this epitope were, so at least 13 to 14 different epitopes were identified from different bacteria and different proteomic approach. And we also characterize through antigenicity, immunogenicity, and toxicity, as well as we look into the structure part of it. So most of them, they are well in allowed region in the Ramachandran plot. The protein-protein docking was performed to look into the more insight about these, the interaction of these protein, because we have like extracellular protein, secretory proteins, and these secretory proteins and extracellular proteins were identified. And then we further, once we identified these, then we have looked into the TLR4 and B cell and T cell antigens. So B cell, T cell mediated antigenicity will be produced. And then we look into all these interaction, 16 hydrogen bonds and 148 non-bonding contacts were found in. So the data is not important here. What is more important, because this is the real data of my lab, not published. The more important thing is that most of the students are here and faculties are here. This is the good approach that to look into the reverse vaccinology and to find the way out how we can identify some best epitopes in order to develop a vaccine against the multiple bacterial infections. And that in that way there are two benefits. One is you can control infection and you can also control the use of antibiotics. So once you control the use of antibiotics, the antibiotic sensitivity will automatically be increased. For example, if ampicillin is stopped using now, then after few years you find the sensitivity may increase in the antibiotic because of the reduction in the selection pressure. Well, this is the final construct that we clone it, and so we clone that whole epitope, multi-epitope vaccine, and after cloning we express the protein into the BL23 and DH5 alpha using this approach. So this is the flow diagram approach where we identify the protein, epitope predictions, then B cell epitope, T cell epitope, MSC2. So we try to find out all immunomodulation of the host system, where usually we are using here the mouse antigen, mouse system, because I mean we cannot work on a human because it's a question of ethical issues and of course trial. So we are trying to find out into the activity in the mouse, and once we have successfully done it, then we can try to translate into the human system also. So we can cover up the all T cell, B cell and MHC molecules. Again, this is another study where we use the proteomic approach. So in the proteomic approach, we identified the at least more than 4,000 different proteins. Yeah, 4,000 different proteins from this bacteria, and these proteins were identified for the best possible epitopes. So we identified this all these small peptides epitopes, and we elucidate the structure of this epitope, which we further express into the protein for the wet lab experiments. Again, these are some studies were performed to identify the validation of these vaccines through, we found the 95% residues are favored regions through Ramachandran plot, and we cover like IED populations coverage. So we also look into the population. For example, it is important because when you go for a trial, you identified like 50 people or 100 people or 300 people. So you must have covered the whole population where you are using these vaccines for immunity or herd immunity. So finally, we end up with this interaction where we found a complex, and then we complex for showing different interaction, hydrophobic interactions, and simulation modeling also shown the stability at the 50 nanosecond RMSD, RMSF, and that shows the stability of the vaccine. This is the molecule that we clone, and we finally express the protein for further wet lab studies, which we have not yet done. It is in a process, so we have not shown the data. This is a flow diagram of the study that we performed. So we have taken the Pseudomonas aeruginosa, A. baumannii, extracted the protein through proteomics approach, and then we identify the multi-epitope, epitopes, and then antigenicity, toxicity, subcellular localizations, and physicochemical properties were identified. And then ultimately we got this construct, the down one, left one, and this construct was ready to express the protein. Okay, sir. Okay, so I'm just a few more slides. So this is another synergistic immunomodulation of the poly. So here we use the nanocapsulation because that increases the efficacy of these antigens, and these vaccine antigens have more bioavailability, vaccine system, and antibody modulation were enhanced by nanocapsulation. And we have using here is a curcumin, because curcumin is a household things and it's a non-toxic, so it's better to make the natural product as a nanocapsulation. So that nanocapsulated vaccines are in again process. The construct is already available, so we are expressing the protein for further studies is going on. What are the challenges with this is the pre-existing immunity, genetic diversity, because sometimes we have a lot of diversity in the bacteria, so that make you like a confusion whether the vaccine will work or not. Then immunological variability, immune stress, and microbiome resilience. Clinical trial success is again a problem, because sometimes you end up with the first phase, then your vaccine is failed. So again, all the efforts goes to like zero. So what we conclude, the AMR is a silent pandemic threatening and modern medicine. And vaccines provides a sustainable, cost-effective, smart solution, and prevention through the vaccination is the strongest antibiotic, because in that case you need not to go for antibiotics. And taken together, we can control the escape pathogens, which are most notorious pathogens, are deadly pathogens causing the infection caused by antimicrobial resistance. Okay, this is my lab, my students, those who passed out, and some of the students are working. And these are my grants. Without these grant support, I cannot carry out this work. So thank you very much for this, listening me, and I think if you have any question.
Thank you, sir. Now I request our chair and co-chair, Professor R.C.
Yes,
Kuhad.
thank you, Professor Khan.
Thank you, sir.
And it's a pleasure, and we really enjoyed your lecture as before as well. And I think very important area you have been working on, and vaccine is the only alternative could control this disease, no doubt about that. I have a small query that what's your take on, say, following the phage-based strategy to produce vaccines?
Yeah, phage is a very good approach, I think. So, but one of the limitation with the phage is that many phages are not. For example, if you are making phage against E. coli, so there's hundreds of E. coli strains, and these phages are not lysing all E. coli strains, because sometimes E. coli may have some defense system like CRISPR-Cas system. So they basically doesn't allow to
organism specific.
Yes,
data.
yes.
That's the only drawback.
This is the problem. But we can have a cocktail. People are working on that. They can have a cocktail, so multiple phage can make a number of like 3, 4, 5 phages, and they have a broad spectrum cells to kill. Yeah.
Thank you.
Yeah.
Thank you very much. So nice of you. If any questions from the audience, please.
Yes, sir. Please.
Yeah, the questions from the audience are open. I request my team to kindly pass on the...
Professor Kanwar from
this is really a
Himachal
great
Pradesh
talk that has been shared by you. Actually, my group is working on oral microbiome.
Sorry?
And my group is working on oral microbiome, oral microorganisms, and we are getting some samples from our dental college, Shimla, IGMC. So there we are looking into actually multi-drug resistance, and somehow there is extreme drug resistance among Pseudomonas strains. And they are in fact effectively very strongly resistant to macrolides, beta-lactamases, and couple of frontline antibiotics also. But they are susceptible to fluoroquinolones. So how you take it, I mean, when the organism which is extreme drug resistance is in the mouth, and maybe when you talk or sneeze or something like this, so what is the extent, in fact, what kind of precautions we expect and what to do that as a future perspective? Yeah, please.
Basically, I mean, this is very, basically, if you talk about the oral microbiome, there's more than 600 different species are present in oral microbiome, and major species are the gram-positive cells. Yeah, not gram-negative, but in case if you got the Pseudomonas aeruginosa through some means, oral microbiome, right? Then this Pseudomonas itself has a lot of resistant gene in the genome. And that is how the horizontal gene transfer of these genes are not possible, because Pseudomonas cannot transmit this gene to some other bacteria. Whereas other than Pseudomonas, like in E. coli and Klebsiella, they have a capacity to transfer through horizontal gene transfer on the plasmid. So in your case, if you find this strain, and if you got a sequence, I'm sure you may have some genes which are present in the bacterial genome, which showing the resistance, like, but macrolides may not find. I mean, they may not have the plasmid for having the gene inside to resistance, and that's how it's become sensitive for the macrolides. It is a very unpredictable thing. Bacteria, which genes or which plasmid is acquired by the bacteria, and when it is cured off, right? So it's kind of a, you pick the same area, same site. Sometime one bacteria, they have a different status, another time it has a different status. If you talk about the microbiome itself, the microbiome keeps changing. And it's a very dynamic, and so if you have some longitudinal study on that, you can find that variations and dynamism in the change in the ARGs spread in the microbiomes. We have shown some studies in microbiome also, not in oral microbiome, but like other microbiomes we have shown it.
Any other questions?
Sir, some other students has a question at the back side for you.
Yes, ma'am.
Hello. Good afternoon, sir. My question from personalized medication system. सर हम इतना एडवांस हो चुके हैं कि पर्सनलाइज्ड मेडिकेशन दे रहा है, तो क्या इसमें AI का क्या यूज है और इसमें मेजर क्या चैलेंजेस हैं पर्सनलाइज्ड मेडिकेशन किसी पेशेंट को देने के लिए?
देखिए, I mean, your question is valid. AI is basically not an intelligent idea. You'll be surprised to know these things. AI is artificial intelligence. It is not intelligent. You know, what is it? It means AI is dependent on total database, right? Whatever database you are providing to AI tools, they will predict out of it. आप Chat GPT use करते होगे या और सॉफ्टवेयर्स होते हैं, I'm not much aware about it. What it does, it is assistance. आपने कुछ डाला, इसने respond किया, तो वो लिखता है searching, searching, searching. It means they are looking here and there. So from where they are getting? आप उससे कोई ऐसा सवाल पूछिए जो पूरी दुनिया में वो चीज़ हो ही ना मौजूद, नहीं बताएगा आपको, because the data is not available. So AI is dependent on the database. So suppose if you have a good question, if you have a database, comprehensive database of all kind of all scenario, all conditions, or then you can use like ANN, random forest, artificial neural network, any kind of ANN to develop some algorithm, and that give you some prediction about the possibility of the drug. On the other hand, you can also use the AI tool for screening the drug molecules based on the chromophore, pharmacophore, based on the structure-based drug targeting. If you are working on, they can give you the best possible molecule that can fit in the target. So that is possibility. You can target-based drug design. AI will help you a lot. Yes, ma'am.
I have also one question. May I? So, you have shown a lot of data on reverse vaccinology, and most of the protein which you have shown, they are basically conserved and they are on the surface of the pathogens, basically. So my question is, the situation when we work in an in vitro system, those proteins, they might be on the surface, but these escape pathogens, they are very smart. Once they go inside your body, they modulate the expression of large number of proteins which are on the surface. They reduce the number from 100 even to the 0. And if we design any kind of a vaccine, which is a protein on the surface, which is having B cell and T cell epitopes, but in a live situation, these pathogens, they try to shut them down. So how we can overcome such kind of situations?
Frankly speaking, I'm not much in this area because I've just started a few years back, trying to some attempt. I think adjuvant, one thing is that you can use some good adjuvants that can help you out. And if you're looking into the cell line, it has a definitely different response compared to if you're using some mouse model or some for human trial, for example. If you are bringing them, definitely it will show different immunomodulations, or it will show you different response from the immunity, T cell, B cells, it behave in a different way. So the only thing is that at the time of screening or the time of identifying the epitope, you have to find the best precise possible, the best possible epitope that could have. That's how we have used the MHC1 and 2 and T, all like possibility of the responses of immunomodulation. So this is, I think, if you're lucky, you can get it. If you're not, then you miss it, the file. Many times vaccines are also fail and they can be drawn out from the market sometimes. So yeah, this is fine. That's a good question. Yes, sir.
Sir, very good lecture, very informative.
Thank you.
Thank you. My just basic query, that these genes which we talk about, antibiotic resistance gene, they came into existence several years ago, even thousands of years ago.
Yes.
They were present. Then how come they were present when antibiotics were not evolved, not discovered? So these genes were present that time also. So why there is a havoc about the antimicrobial resistance? How come it is becoming more and more problematic problem for us
to find antibiotic to combat the resistance against these genes. So what is your opinion about those genes, which were already present 1,000 years ago?
Yeah, you are correct. Genes are, I don't know if it is 1,000 years ago or more or less, but
there are reports that they were present already in the nature.
Even you will be surprised to know that, please ma'am, please have a seat. You will be surprised to know even in archaea bacteria, where thermophile and extremophile bacteria are present, they have ARGs, and they have the same ARGs as what the mesophile or pathogenic bacteria. So there is somehow link between, because of horizontal gene transfer on the evolutionary scale, the exchange of these genes. This is one thing, that's true, they are existing. But the thing is that why there is a havoc, your question is, havoc, the reason of havoc is that now the infection is increased. So infection control measurement is not proper. And population is also increased. We also spreading the infection through person to person. For example, in communicable diseases. If you take an example of COVID-19, how it is spread, right? Or some other viral infection, influenza virus. Similarly, various tuberculosis is increased. Leprosy is not like in dormant state. Leprosy is still cases are coming in leprosy. Leprosy is a very live disease. Now people thought in India that leprosy is like khatam ho gayi hai. Leprosy is not khatam hui hai. Leprosy is still there. I am a member of one of the ICMR committee in SAC in leprosy center, and regular cases are there, and they are very worrying about how we can control the leprosy in certain regions. So things are there, but I think the infection, since infection is increased now, and no drug is left to treat infection over a period of time. Earlier there was ampicillin, you can use ampicillin, you can use cephalosporin first, second, third, fourth generation, then you can use the carbapenem, imipenem, meropenem, doropenem, then you can use recently 2015 onward colistin. But now since all resistance happened, and because of the, NDM1 discovered in 2009, and now we have 68 or 64 variants of NDM1, highly drug resistant against all drug of resistance, even against sulbactam. Sulbactam is an inhibitor, beta-lactamases, but it is against that showing resistance. And sulbactam is made for serine beta-lactamases, and this is the metallo-beta-lactamases. With slight change, it's not working. So due to the mutations in these genes, and due to the mutations and evolutionary changes, it might lost, like antibiotics are not working against such kind of a bacteria having these markers, these genes. That is the reason. No, it's, you can say it's a man-made, but it's not a man-made. You can say in a man-made, it's not a man-made. The reason, because man-made is because we are not complying the proper treatment. We are not approaching the good physician, correct, original, sahi physician. ये नहीं साहब जो physician नहीं उसके पास जाके आपने दवा लिखा ली. We are not completing the course. We are not basically using the good quality drugs. आपने जो ड्रग्स मार्केट से खरीद स्पूरियस ड्रग्स हैं, भोपाल में पूरा मार्केट है ड्रग बनाता है, वो 2 नंबर की ड्रग्स होती हैं, वो ड्रग्स जो हैं हमने खाई, हमारे साल्ट जाना है 50 मिलीग्राम 500 मिलीग्राम टू किल द बैक्टीरिया, हमारे पास साल्ट जा रहा है 1 मिलीग्राम 2 मिलीग्राम, रेस्ट इज पाउडर. सो बैक्टीरिया विल परसिस्ट एंड डेवलप ए रेजिस्टेंस. बिकॉज आपने, अगर आप मुझे मारना शुरू करें यहाँ, तो मैं भी, मेरे अंदर भी एक स्ट्रॉन्ग एक ताकत आएगी, भाई मैं भी फाइट करूँ, मैं भी डिफेंड करूँ. तो इट इज़ ए काइंड ऑफ ए डिफेंस मैकेनिज्म इन बैक्टीरिया. आप जब उसे मारेंगे, बार बार दवा देंगे, बुलेट देंगे, तो बैक्टीरिया एक्वायर्ड द मैकेनिज्म ऑफ ड्रग रेजिस्टेंस मैकेनिज्म इन ऑर्डर टू डिफेंड फ्रॉम दिस सिस्टम. सो दिस इज़ हाउ वी डिड इट. ओवर द काउंटर ड्रग, ज़रा सा नज़ला हुआ, नाक बही, भाई एजिथ्रोमाइसिन खा लो, क्लावनेट खा लो, अमॉक्सिक्लेव खा लो, 2 दिन खाई, छोड़ दी. घर में अब्बा लेके आए, बच्चे ने भी वही खा ली दवा, बच्चा लेके आया, बच्चे की आई दवा, अम्मा ने भी वही दवा खा ली. आई मीन दिस इज बेसिकली द रीजन हाउ वी आर मेकिंग थिंग्स मेस्ड अप. सो इट इज नॉट थ्रू अस, बट वी आर डूइंग दिस थिंग्स.
Sir, for a layman, what's the best way to prevent this AMR infection?
For a layman, देखिए 2 चीज़ें हैं. There are 2 interface. One is the policy makers and infection. Second is the scientists and infection. Policy maker इस पे काम कर सकते हैं. साइंटिस्ट जो है सिर्फ बता सकते हैं आपको, और जब तक ड्रग बनाएंगे 10 साल में वो ड्रग फिर रेजिस्टेंस हो जाएगी उसके अंदर. So scientists are not much effective. Scientists खाली डराने के लिए हैं. मेन चीज़ जो है इसमें आप पॉलिसी मेकर्स हैं, जैसे मान लीजिए ओवर द काउंटर, केरला इज द ओनली स्टेट वेयर द ओवर द काउंटर ड्रग इज कंप्लीटली बैंड. एंड इफ वी adopt the same policy, I am telling you, in next 3 to 4 years, you will see the reduction in the drug resistance. So main use is overuse and misuse. आप एंटीबायोटिक यूज़ करना छोड़ दिए. मैं एंटीबायोटिक कभी यूज़ नहीं करता. मैं आयुर्वेदा, यूनानी मेडिसिन यूज़ करता हूँ.
Say that's one thing. What other preventive method we can use?
Preventive method तो ये है कि we should have wash. WHO uses the word wash. Wash means clean, hygienic and clean water. हाइजीन एंड सैनिटेशन इज़ वेरी इंपॉर्टेंट फॉर इन्फेक्शन कंट्रोल. आप इन्फेक्शन को कंट्रोल कर दीजिए, ड्रग की ज़रूरत ही नहीं पड़ेगी. रेजिस्टेंस का सवाल, इन्फेक्शन जब होगा तभी रेजिस्टेंस का सवाल खड़ा होगा. जब इन्फेक्शन नहीं है तो रेजिस्टेंस ही नहीं होगा. तो यू स्टॉप इन्फेक्शन. हॉस्पिटल मैनेजमेंट सिस्टम होना चाहिए. आप पहले नहीं होता था, अब तो यहाँ पर MBA वगैरह उन्होंने हॉस्पिटल मैनेजमेंट MBA स्टार्ट किया है, मैनेजमेंट सिस्टम स्टार्ट किया है, इन्फेक्शन कंट्रोल मैनेजमेंट सिस्टम स्टार्ट किया है. So I think we should focus on the control of infection, clean water, hygienic water, clean food, wash your hand every time whenever you take food. हम लोग क्या है, हम माइक्रोबायोलॉजी पढ़ाते हैं, लेक्चर दे रहे हैं, अभी खाना स्टार्ट होगा, ऐसे ही जाके खाना शुरू कर देंगे, हाथ वॉश नहीं करेंगे for some reason. So we have to wash hand, clean hand. बल्कि अभी मैं बता रहा था, अभी मैं एक हिप्स एक दिल्ली इंडिया हैबिटेट सेंटर में था मैं, वहाँ दे वाज टॉकिंग अबाउट, एक ग्रुप है वो डिस्कस कर रहा था, उन्होंने शायद केरला ही से था वो, दे हैव मेड सम गाइडलाइंस, ब्लू पिंक समथिंग सॉर्ट ऑफ दैट. उसमें उन्होंने ये कहा कि सर वॉशिंग हैंड, इवन यूरोप में भी बहुत से आप देखिए, आफ्टर वॉशरूम व्हेन दे कम आउट फ्रॉम द वॉशरूम दे नेवर वॉश द हैंड्स. आप उन्होंने हाथ साफ करे, पोछे उन्होंने, और दैट्स इट. सो दे आर फोकसिंग ऑन दैट, वॉश योर हैंड, वॉश हैंड. WHO का मेन इज मैंडेट वॉश का है, WASH, वाटर, सैनिटेशन एंड हाइजीन. सो दिस विल रिड्यूस द इन्फेक्शन. Thank you.
Thank you, sir. As we are running very short of time, if anybody has further questions, sir will be here with us downstairs. You can meet and interact with them. Now I request my chair and co-chair to kindly share a token of respect and gratitude to our speaker for this session, Professor Khan, a Padma Shri awardee. Can we have a huge applause? Thank you, sir, for enriching session, and thank you to my chair and co-chair for being here for this session. It is a time for the next session, that is session 8, Microbes in Agriculture and Crop Improvement. This session highlights the crucial role on beneficial microbes enhancing the cultural and agricultural productivity and sustainability. Experts discuss how microbial communities contribute to the soil health, nutrient cycling, plant growth, and promotion and disease resistance. The session focuses on recent advances in biofertilizer, biopesticides, plant microbe interactions, and genomic tools to develop eco-friendly agricultural solutions. It is a time to introduce the chair for this session, Professor Sunil Pabbi. Professor Sunil Pabbi is a Principal Scientist and Head Division of Microbiology at Indian Agriculture Research Institute, New Delhi, and a core committee member of Association of Microbiologists of India, AMI. A renowned microbiologist, he has a significant contribution in the field of cyanobacteria, biofertilizer, and sustainable agriculture biotechnology. With this, I request my chair to kindly please join us on the dais, Professor Sunil Pabbi. Can we have a huge applause, please? Thank you, sir, for joining us. Welcome to Dev Bhoomi Uttarakhand University, sir. It is a time to introduce the co-chair of this session, Dr. Shiv Kumar. Professor Shiv Kumar is a distinguished academician and researcher serving as a professor and head of the Department of Agriculture Microbiology at Tamil Nadu Agriculture University. With a vast experience in microbial biotechnology, plant microbe interactions, soil microbiology, and bioconservation technology, he has been instrumental in advancing sustainable agricultural practices through microbial innovations. With this, I request my co-chair to please join us on the dais. Please can we have a huge round of applause for our co-chair of this session, Professor Shiv Kumar. I welcome you once again, sir, to Dev Bhoomi Uttarakhand University. Since we are starting with the first keynote session, so I request once again to my co-chair and a keynote speaker, Professor Shiv Kumar, over to you, sir, for your presentation.
It's already loaded. Yeah, in a second. Okay.
This is the pointer, you can just slide up and down this. Sir, if you are able to see your screen on that.
Okay, okay. Well, thank you very much. Indeed, it's a great pleasure for me to be here to meet and greet you all on the occasion of AMI conference. Here the title of the talk is Biocatalysts for Biomass-Derived Chemicals. So we have been working on this for the past 2 decades. We started working on lignin degradation and then started working on the enzymes associated with lignin, cellulose, and then now we are focusing on the valuable chemicals production using biomass material, more particularly on cellulose and biocellulose, cellulose and lignin derived materials. To brief about the university where I belong, it's a Tamil Nadu Agriculture University, one of the top ranking agriculture university, and the department was started in the year 1979 as agriculture microbiology. So where we do lots of activities relating to plant microbe interaction, biofertilizers and biocatalysts, biomolecules, food fermentations and so on. As far as the department is concerned, we have a privilege of having 3 different centers of excellence, centers of excellence by ICR and the Center of Excellence by MHRD, now it is Ministry of Education, and another Center of Excellence by UNDP FAO. This is how I divided my talk. The first half will be focusing on biomass, where I will skip some of the basic slides, and the second half will be focusing on the enzymes and associated process that releases the biomolecules, more particularly lignin-derived aromatics and the cellulose, hemicellulose-derived products. So here, being a tropical country, we receive huge amount of sunshine. More than 12 hours of sunshine in a day help us to produce more biomass. So with that biomass, we can produce the biomass-derived material. Only issue with this biomass materials are the complexity, recalcitrancy in the structure of the material. Otherwise, it's a very good material to proceed with because of its renewability, availability, all those things are very good. And with this biomass material, once we develop any process that converts the lignocellulosic biomass into either fuels or chemicals, there are few challenges. One is the lignin. Lignin is being a recalcitrant molecule, this has to be carefully removed. And while removing lignin, when you use either acid or base or any chemical, mechanical methods, it produces lots of inhibitors. So inhibitor generation needs to be reduced in the first process of pre-treatment. And the second process is bioconversion, which is otherwise known as hydrolysis or saccharification. So here we have to use mostly of thermophilic enzymes. So these thermophilic enzymes are very effective. And these are the two major issues. Once we have these issues, primarily we can target on the conversion process. And of course, the conversion process is also a multi-step process where we have to intervene on so many ways of developing the enzymes, cocktail of enzymes, to proceed with the converting them into monomers or dimers or trimers. And to start with the biocatalyst, we have been working on various thermophilic enzymes of lignin degradation and thermophilic enzymes of cellulose hydrolysis. And these enzymes are really very, very active. Catalytic efficiency, site specificity, and turnover, all these things are very good. That's the reason why we always focus on this. In due course of my presentation, I will be explaining how these enzymes have been taken up. So this is the steps by which the biomass can be converted into either fuels or chemicals. To start with the pre-treatment, these are the requirements which we have to follow while selecting a methods. And if you look at different methods, there are so many methods available, physical, chemical, mechanical methods are available, of which we focus on the enzymatic process because we have been working on these enzymes for several decades. And more particularly lactases. Lactases are oxygenating enzymes. We focus on the lactases. So if you look at what are lactases, they are oxidases, multi-copper oxidases. They have a site-specific action. And here often it is called a green catalyst. The reasons are like it uses oxygen as electron acceptor and reduces the product of your choice into reactive radical. And this reactive radical could be used for various other purposes like dimers, trimers, tetramers. So initially we have worked on that with the bacterial domain, archaeal, and as well as the fungal. In all the sources, the red highlights are the works that we have done in the past. And we started as early as 1989 on the lignin degradation, so where we wanted to use very hardest material, pine needles. So these pine needles were source for isolation of the lignin degrading organism, and then we characterized the enzymes. Even during 90s, we have developed some mutants, oxotrophic mutants, protoplasmic recombinants. All these things were used for making biocompost during that time, and that biocompost could be a very good slow release fertilizer in organic agriculture. And then with the collaboration of University of Florida Gainesville, we have developed the first archaeal lactase. The model Archaean Halophrex vulcani was used for that. And this enzyme happened to be a solvent stable, thermostable, and salt stable. And more interestingly, this enzyme doesn't, even though the organism requires 2.5 molar of salt concentration, the enzyme is active at 0.2 molar. So that's an interesting feature. And the solvent stability made us to think about developing a process. And since my tenure at the University of Florida was over, then I moved back to India. And we were unable to use that organism because it was US patented one. And then we worked on the culture which we have isolated from the forest ecosystem, Hexagona hydra, and this enzyme is also recognized as a hyper-secreting lactases and solvent stability. So with that solvent stability, we were able to develop a process called Enzol. So that process could be used for developing very good enzymatic delignification process where we can get 40 to 50% of lignin recovered and used for other applications. And then we have also developed a protocol how to produce this enzyme using submerged fermentation and solid state fermentation. So we have developed all the protocols which we have used for optimizing the standardized condition. On the right hand side, you can see the stability of this enzyme to various extreme conditions, and we were able to get 1585 units per ml, and in the same organism is producing very huge amount of 3500 units in liquid culture. So that's an interesting feature about this organism. And we have also checked whether this organism is able to deconstruct lignin. So this observation was confirmed by using FTIR and GCMS and scanning electron microscopy, how these enzymes are penetrating into the biomass molecules under the specified conditions. And then we have also checked the reduction of hemicellulose, reduction of lignin, and increased quantity of cellulose. All these things were confirmed by multiple times. And interestingly, we found some of the metabolites like lignin-derived compounds were observed, and that made us to think on how best we can work on the lignin-derived aromatics. And this was a process, again, it was patented by us, and this process is unique process in the sense that we use the enzymes here, and we use solvent because the enzyme is solvent stable, and also we use a steam in a sequential manner, not at one time, in a sequential manner. This process works very well on corn cob and wood biomass, and we were able to get around 35 to 40% of lignin reduction, and it takes a lot of time, like 17 hours to delignify the biomass material. That was the biggest problem that we faced, and to overcome this, we have developed a reactor, a hydrodynamic cavitation reactor. So once we feed the same material under optimized condition, we were able to reduce the time, but the lignin reduction was not increased. So here we were able to get the same amount of lignin reduction, but within 1 hour we were able to get the delignification done in the biomass material of various kinds. And then we have also evaluated the same process on the material which is grown as a wasteland crop like Erianthus. Erianthus is a sugarcane relative, and it is growing almost like a sugarcane, not exactly sugarcane. It doesn't require any water, fertilizer. It grows as a wasteland crop. So we wanted to take this crop and check whether the lignin part of this biomass material could be removed by this process which we have developed with the same hydrodynamic cavitation reactor. So we were able to get around 49% lignin reduction. On the right hand side, you are seeing several lignin-derived compounds like benzoic acid, benzaldehyde, salicylic acid, all these things are co-produced along with the delignification process. So that is an interesting process which we have developed. And then we wanted to work little more on the lignin-derived compounds because having worked on various enzymes, so here this is to show you that how important the lignin-derived aromatics. You name any process, biochemical process, either in the cosmetics or pharma or any industries, all fragrant industries, flavor industries, aroma industries, in all the spheres, lignin-derived compounds serves as a platform chemicals. So with that idea, we wanted to use, so there are several players who are already working. So currently this lignin obtained from either paper or pulp industry or any of the industries could be used as a low value process. More particularly, it can be used as a fuels, binders, and so on. So here we wanted to get these high value chemicals produced, provided we have to get some separation process optimized in this. And then this is to show that some of the unique compounds like vanillin, which is a flavor enhancing compound, hydroxy, benzoic acid, all these things are antimicrobial compounds. This could be used in various organic, agriculture as well as pharmaceuticals companies, and their functions are listed on the right hand side. So with that idea, we tried several materials, several substrates, like whether this lacase alone is working on wood biomass, whether this lacase could be used to delignify the craft lignin, black liquor, all these things were tried. And here in this case, we tried treating lacase with the biomass material, wood biomass material. So interestingly, we were able to get some compounds which are also unique in nature. They can be used for antimicrobial action, anti-tumor compounds, and so on. And then we have also tested this effect of lacase on the craft lignin. And then here also the treated ones showed so many compounds. Say for example, hexadecanol, benzaldehyde, all these things were present only in the treated ones, not in the untreated ones. So this suggests that this enzyme is working on the polymeric lignin, depolymerizes without getting repolymerized, and this could be used. Only thing is we have to have some separation methods. And we also tested black liquor, which is a waste that is obtained from paper and pulp industry. And this black liquor was initially processed to have a unique, have a optimum pH and other salts removed. And then we treated the lacase, and we were able to get benzoic acid and the hexadecanoic acid unique, and they are present only in the treated ones, not in the untreated sample. So this also an interesting information. Still, we were not happy with the way in which the lacase is being treated onto the lignin-rich material. So we wanted to have some mediators. So these mediators will activate the rate of enzyme. And here we used HPT, hydroxybenzothiazole, and this activated the enzyme. Even we could get lignin removal to an extent of 70%. So with that idea, we used one of the wastewater where the phenolic content were removed to an extent of 70% in the treated ones. And also we tried a different concentration of this mediator. So the role of mediator is to activate the enzyme, and rate of reaction could be increased. And the same thing was confirmed with the GCMS data and the FTER, that these derived products are being accumulated more in the treated ones. And then we have also had a collaboration with the Russian team, where they wanted to have a catalyst with the biocatalyst. We know that catalyst and biocatalyst could not be combined, but in a sequential manner. So once the material of birchwood of their own material, corn cob of our own material were subjected to catalytic process, and that catalytic processed material could be subsequently treated with the lacases. And we were also able to get that done and compared with the enzymatic process with the organosolve process. And it is interesting that the catalyst treated ones, when they are subjected to biocatalyst, we were able to get 76% of lignin derived monomers, and we have had the good number of aromatic compounds produced in this process. Only thing is we have to follow the strategy, and this is not readily available now. Still we are working on it. And then we, having worked on various substrate materials and mediators, and we wanted to take one particular biomass like cotton stalk and see whether the lignin from the cotton stalk could be recovered and used for various applications. So this work is also going on. It is an ICR funded project. So what we have done here is, we have taken the lignified material. So the lignin portion of cotton stalk was taken, and then extracted the lignin, and the extracted lignin by either acid or alkali treatment could be used to extract using various solvents. Here the lignin derived monomers, dimers, tetramers were extracted very well, and we tried several solvents which is listed on the right hand side. So here the methanol is used to get very high number of lignin derived aromatic compounds. So that work is also given here, and you can see here the number of compounds: vanillin, benzoic acid. So many antimicrobial compounds are produced in a high volume. So here we are working with a chemical engineer. We wanted to develop a membrane process, so that will help us to separate these compounds and scale it up to the next level. Okay, so the next part of presentation is the cellulases. So how the cellulases or xylanases could be produced from various sources, and these produced enzymes, how best we can produce some of the products using the xylon and the cellulose material. So we started working on the thermal springs of Manikaran, Vasist, Kalat, all those things. We went there and then did some in situ enrichment using biomass material, natural polymers, and isolated the organism. So all these organisms were potentially thermophilic, and these organisms have multiple functions like cellulase activity, xylanase activity, pectinase activity, amylase activity, and so on. So with this organism, we were able to get one organism that showed very high level of beta-glucosidase activity. So this is one of the best organisms which we have now. And in addition to that, we have also did some enrichment in the compost where the temperature rises to 60 degrees Celsius or 55 degrees Celsius. And assuming that the thermophilic organisms could be isolated from the compost, and we have isolated the Cytalium thermophilum. So that also had multiple enzymes like exogluconase, endogluconase, beta-glucanase, all the enzymes are being produced with this organism. So this organism is also characterized and it is available with us. And then we have also isolated some organism like Aspergillus fumigatus. This is the one which is used for cello-oligosaccharide and xylo-oligosaccharide production. And we have isolated, characterized, and we have standardized the optimum condition in which the enzyme could be scaled up. And we have also characterized the stability part, and it is available now. And the production levels are given on the bottommost slide. Like endogluconase is very high level, which is some of the companies are communicating with us so that they can take this culture with them for scale up to the next level. And similarly, the same organism is producing xylanases. See, if you look at the xylanase level, initially the organism produced 445 units per ml. And after optimization, this could produce 624 units, which is very high level, and this could be used for xylo-oligosaccharide production. And then we have also used this organism to produce enzymes in solid state fermentation using various biomass material. So interestingly, cellulose could be produced using banana fiber among the various polymers we have tested, and the xylanase could be produced using what is that? Wheat bran. So we have taken this selected wheat bran for xylanase production, banana fiber for cellulase production. All these conditions were optimized, and then with that, we wanted to produce cello-oligosaccharide and xylo-oligosaccharide. Before going to cello-oligosaccharide, I just wanted to show you one more product here, xylitol. So xylitol is a natural sweetener, and you know, it is very, very important sugar molecule. So this could be produced using yeast-based system where the enzymes like xylitol reductase is hyperactive in Candida tropicalis and Saccharomyces cerevisiae. So there is no system to screen xylose reductase directly from the plate. So we have done an alternate method of selecting xylose isomerase producing organism on the plate, and then further tested the xylose reductase onto the gene level, and this gene was taken and overexpressed onto E. coli based one. And we were able to get the xylose reductase produced. And this xylose reductase being a reductase organism, it requires NADPH for its activity and also convert the xylose part into xylitol. So instead of that, we wanted to use glycerol here. So the glycerol based system, done sir, 2 minutes. So the glycerol addition has yielded about 6.4 grams per liter. So that is an invention here. And the oligosaccharide part, everybody knows that it is a probiotic. It has got so many applications. And this is how we designed the process of cello-oligosaccharide and xylo-oligosaccharide. And Ensol, which we have discussed, whatever we have developed for corn cob and wood biomass, is same thing applied here to produce. And then to extract xylon, we used two-step process of alkali and hydrothermal assisted alkali extraction. And okay, I will skip this. So this is a key slide of how the xylo-oligosaccharide could be produced. So the initial material is A, and then we processed it and extracted process is freeze-dried, and it is available. So we are now in the process of toxicity study. One of our hospitals in Coimbatore, they are taking up this, whether this could be used for animal feed application or human application. At the same way, cello-oligosaccharide we have produced, and the freeze-dried material is given to the hospital for testing its toxicity level. And this is the mass balance of both the cello-oligosaccharide and xylo-oligosaccharide. So if you take 1 kg of biomass, you can get 15 grams of xylo-oligosaccharide. Similarly, if you take 1 kg of banana fiber, you can get 59 grams of cello-oligosaccharide. So this is how we are working. So we are also closely working with the engineering team in designing the reactor and scaling up to the next level. So with this process we have developed in the pilot scale, we wanted to scale it up to the next level. So fortunately, we were sanctioned with the Bio-E3 call for upscaling cell-oligosaccharide production as well as enzyme production to the next level. So they have sanctioned around 2 crores for this project. So with this, I wanted to thank our team of students, research scholars, postdocs. So they are not only microbiologists, they have a chemistry background, and renewable engineers are there, plant physiologists, plant pathologists, all of them are working together in our team. And our mentor, funding agency, then place where I did the postdoctoral research. So we have collaboration, strong collaboration with Russia, UK, Netherlands where we have learned the microbial volatile organic compounds, how this could be a signal or biocontrol agent. And the last 3 part, University of Tokyo, South Africa, University of Zululand, Changan University, all are active collaborations. Currently they are working with us. And I would also like to thank the funding agency who has generously funded for our research. Thank you very much.
Thank you, sir. Now I request the chair of this session to kindly share a token of respect and gratitude to our speaker for this session. I request sir to kindly proceed for that. And I request the further speakers to kindly complete your presentation in the given time, as we are already running late. Thank you so much. Audience, can we have a huge applause for this panel session? Over to my chair for the next speaker.
Okay, thank you. Thank you, Dr. Sivakumar, for such an informative talk and applied aspects of agri-residue management, I would say. The next speaker is Professor Alka Sharma. Professor Dr. Alka Sharma, she is a distinguished academic and expert in food science and technology at Guru Jambeshwar University of Science and Technology, Hisar. A professor in Department of Food Technology, she also serves as a chairperson of the Women's Cell and an expert panel member of the NBA. Her PhD in Food and Nutrition from Hisar, HAU Hisar, Professor Sharma's research focuses on food processing, post-harvest technology, and the development of functional foods and nutraceuticals. A prolific researcher, she has published over 85 papers and secured multiple government and World Bank funded projects. Her excellence is recognized by HAU Merit Gold Medal and ICR Research Fellowship. So may I request Dr. Alka Sharma and also request her to stick to the time of 15 minutes.
Thank you so much. A very good afternoon to everybody here. And I'm thankful to AMI and Dev Bhoomi Uttarakhand University for giving me this chance, and of course my university who sanctioned my leave and allowed me to be here. And a very happy Children's Day to everybody here. There are few kids also who were all deep into science since morning. I was listening to them, but I think they have left now. But everybody has a child in him or her. So best wishes to all of you, and including me also. And don't let the child in you die, ever. About today's talk, it's a bit different because, could you please help me? How do I move?
this one next.
Okay, next. So this talk is not the usual talk like we were listening since morning and for the last 3 days. It's a bit different for everybody, and it makes me remind of an advertisement where Pankaj Kapoor used to say it's different. So it's going to be different, no deep science into it, and it's just to remind us what we actually know and what we are inside our body, and we tend to forget. And one more point I would like to add after listening to today's sessions, that many a times it happens that we just are, like the scientists over there were saying, that we feel helpless. What to do now, because microbes, they cannot be controlled by anybody all the time, because they are the superpowers. They are going to stay when we don't stay. So we need to concentrate on things like where we can also do something. And today's talk will be revolving around this factor only, how food, how immunity can be built up, how our lifestyle, it can help us treat with all those things, from all those like disorders in our body when doctors fail. So we need to be reminded about that. One more thing, we Indians, we have a tendency to forget or to go to the, like we ignore things. We always get feelers, we always get signals from our body that this is the time we need to think about what something is going wrong. हम डॉक्टर के पास तब पहुंचते हैं जब बात हमारे हाथ से निकल चुकी होती है and things are gone bad by that time. So we should be more caring about ourselves, and we are here to live our life well. So doctors are fine, scientists are also fine, but we should also do our role, and we should understand our body, and we should remember that we need to work about ourselves also. And one more reminder about this thing, that nature has made us very strong. We have enormous potentials. There are times when people have done, the humans have done the thing which nobody expected them to do, like how much weight can be carried by a person. So remember your energies and use your potentials to live the life well. And body, you know that it works on the principle of maximum economy. Body has its own way of correcting itself, and if we identify them, we can do well. Now coming to the main theme, that is the gut-mind connection, and of course microbiome is involved into that. There is an axis which connects the body inside us, the mind there, and the microbes revolving all around us. So in our body, why gut health is important, everybody knows. I won't go deep into this, but you know that for nutrient absorption, for better immunity, for good skin, for good health, for improved mental health, and for good moods also, everywhere mental health is important, and microbes as well as the lifestyle has a lot to do about that. And gut, when we mean gut means it generally it includes when we refer to stomach as well as the intestine portion, that is what we call gut. And how do we know that our gut is healthy? Actually, everybody knows when you are not able to pass your stools in the morning well, because passing the stools is even more important. Like if not that, it is equally important than having the food. तो आप देखिए एक बड़ी प्यारी सी ad भी आती है about some laxative or something, how good he or she feels after when the gut is clear. And when gut is clear, the good microbes are there, the probiotics are there, and we work for those probiotics, everything will be fine. And I think most of the diseases, more than 80%, I can say, of course it is my value, they can be treated, or their reason or their etiology, it is around the food and the gut health. So gut integrity, good fiber, good bowel regulation movement, good sleep, all of them, they are interrelated also. And who controls in our body? The young ones, the students at the back, if they are sitting and they are listening, they need to, many of them, they might be from like physiology or microbiology, or they might have studied some physiology, but yes, we do have centers in our body that control how much food to eat and when to stop eating food. Like we have the hunger centers, the satiety center, and after eating food, the blood glucose level, when it rises, it leads to the activation of the satiety centers and we stop eating. So a balance is required. And our history, our literature, our epics, they also say that we should be balanced in our life. So food, microbes, microorganisms, our brain, all of them, they can be worked and we can do good to our body. Then we have so many hormones also working in our body like leptin, ghrelin, their balance is required, they are produced by our adipose tissues, but if in a way it is good that more of adipose tissues, more of leptin, but there is, if imbalance is created, it leads to, it leads to obesity also. And empty stomach, this is responsible for ghrelin. These are the ones which actually control what we eat, how much we eat, when we stop eating. So there are so many other factors also that are responsible for maintaining the blood glucose level. Insulin everybody knows, but that reduces the blood glucose level after we eat and blood glucose level increases. But there are many other factors, so many hormones they are included. Which are involved into this. The incretins are there, then you have your glucagon is very important, steroidal hormones, the stress hormones, there are so many factors. Other than that, there is one thing, like there is a nerve also, it is the longest nerve in our body, it plays a very important role. It is called as vagus nerve. And why I am stressing over this nerve is that some of the exercises which we can do daily, they can activate this nerve and many things they can be corrected. Like this plays an important role in the inflammatory diseases, and nowadays inflammatory diseases are on the rise. The joint related, the pain related. So if this vagus nerve is active, then up to a great extent the things can be controlled. It is connected to gut health, to digestion, to thyroid health, to organ functions, to stress, to sleep, heart rate, blood pressure, so many things, and we need to be careful about our lifestyles and our exercises and whether we are able to give sufficient movement to these muscles in the body, the joints in the body, and this vagus nerve activation. I'll be just referring to what can be done. I won't go into detail because the time given is too short. And as the title has indicated, I need to connect it to our literature also, to our itihasas also, जो हम भूल बैठे हैं और हमें जानना चाहिए, and more validation, more evidences can be collected because old evidences are lost, and the scientists and researchers, those into food technology and other than those also, we can at least have a look at what our forefathers have said or what our literature is saying, whatever is available with us, whatever is still left with us, and we can do more scientific studies and we can get the honor or respect also, because many of the people, they say that allopathy is the only best treatment. We should not go for the parallel way of treatments like the Ayurvedic or homeopathic, or there are so many lines of medicine. But yes, if sufficient studies are conducted, the things can go good, and when the doctors fail, these things they can work. Then I'll try to connect and give just few examples of how our forefathers, how our rishis, and how our very old scientists, which I call itihasas. Many of people, they don't call it history. What we learn in history is what we read in our books. They do not consider literature, our Granths, our Vedas, they are not considered history, but I personally, I consider them. So they deserve to be more exploited, more explored, and more validation. If it is done, they will also get their due respect. So I request the young ones and the senior ones also to be, to at least have a look and try to find out what is actually scientific into that. Because many of them are still considered to be food fads or the fallacies or the wrong notions. But many of them, they have proven to be correct, and scientifically they have also been validated by many. But still they need to be, attention is required to them. Then there are so many exercises, I'll just touch some of them, like there is Patanjali Yoga Sutra. I'm not talking about the company or the trading person Patanjali, I'm talking about the Rishi Patanjali and the documents given by Patanjali Yoga Sutra, in which the scientists, they have connected and they studied about the connection between vagus nerve and how it helps in regulating. So there is a concept of Ashtanga Yoga also, which has 8 elements, which include Yam, Niyam, Asan, Pranayam, Pratyahar, Dharana, Dhyan, Samadhi. This you can always study. Information is available everywhere. So out of them, some of are very important. Few of them, they have been taken from Ashtang, and other ones are also taken from different literatures, which include pranayam or the breath control or the breathing exercises. They can help in the gut flora also, they can help in health also. Pranayam, the meditation part, the discipline, the fasting, all of them, they are connected to our body. The contentment, the gratitude, the ahimsa, non-violence, all of them, they have a connection with our health. So in a way, we can say that through all these we can naturally tone the vagus nerve, and we can harmonize digestion. And if digestion is good, so many things can be prevented. And one major thing is obesity also, which is getting more and more. Obesity and depression or the mood disorders, they are on the rise, and we need to take care. How the vagus nerve can be toned, it includes the breathing, diaphragm. There are so many types of breathing, and it includes the gargling also, laughing exercises also, the vocal activities, the temperature treatments. Diaphragmatic is the one in which the diaphragm gets to move. Instead of being lethargic, if we are able to move those parts of the body which are generally not mobile. So that mobility and the blood circulation reaching each and every part of the body, right from the head portion and till our toes, towards all the exterior portions of the body, then things will go wrong. So stretching does help, and these small tips, they can also help. And these are not just tips. Actually, these have been practically studied also, and I personally very much believe in this because I have been experiencing also. So lifestyle changes, I will be touching a bit about the diet, because the kind of fiber, the soluble and insoluble fiber in our diet, 25 to 40 grams per day is sufficient to take care of our requirement, and the gut will be clean. Microbes in our gut, they will be healthy, and if along with that we take probiotics, prebiotics, symbiotics, then postbiotics. There are so many terms emerging day after day. So they are all targeting towards the gut health. So they can be like considered to study. And gratitude, one should be feel blessed. One should be thankful about what we have and not always craving about what we don't have. Then probiotics and omega-3 fatty acids, you know that they are so much popular these days, but we need to think them, and more can be studied also. The grain, here I want to add one thing with regard to the microflora which is developed in our body. When we are born, our gut is sterile. But once we are outside in this world, outside our mother's protection, the microbes, they start entering our body. The first source is mother. So the health of the mother and what is given to the child just after birth. In many of the families, there is a concept of giving good tea also. And what is given to the child other than the mother's milk, that is also very important. I have seen recently in the hospital, even doctors, they don't bother about other than mother's milk is fed to the children, and after that the colostrum in the mother's milk won't work, because the digestive system will start working, and whatever colostrum which was being absorbed intact as protein, now after the digestive system is working, has started working, it will be broken down into just amino acids, and it will be just a good source of protein, not the colostrum. Then immunoglobulins तो बाहर से इंजेक्शन दे के ही लगाने पड़ेंगे, क्योंकि कैन नॉट बी टेकन orally, it will be digested, it will be broken into amino acids. So mother's milk and the first feed of the child, in everybody's family there are small children, small kids, they will be born sooner or later. So be careful about what is given to them in the beginning, and that has an impact. And this is proven also that what microbes are developed in the beginning, the ones which colonize in our gut, they have an impact till we die, till the last breath, because they have a tendency to colonize, they stay there. So first feed is very important. So we can be more careful about that. Other than that, the food we eat, the nutritional as well as the non-nutritional factors, and the stress. If there are children over there, yes, you study good, but you don't have to stress about that. Stress can be of multiple types, though the stress, if you concentrate on some exercise, I'm not talking about that go to gym and अपने वहाँ पे सांस होने की ज़रूरत नहीं है. ऐसा नहीं है कि आपको बहुत ज़्यादा जिम करना है या बहुत ज़्यादा heavy exercises करनी है. Heavy exercises are never recommended because more will be the oxidation, more free radicals will be produced, and these free radicals, if they are not taken care by antioxidant natural ones or the other ones which we are taking from outside in the form of fresh fruits and vegetables, then they are going to overpower us. I don't know where I was. I'm moving to next now. Right, this I've already told. And all of the gut microbes, they have an impact on our molecules of emotion also, like the serotonin, the dopamine, which are secreted naturally in our body, and they should be maintained for some time in the body. So for that, so many things can be done, and good food and probiotics is one of them. I need not talk about depression, but one of the reason is improper health and lack of sleep and a lot of stress, which is leading to depression. The values are given there. You can just have a look or study later on also. So now coming to the last point of my presentation, in which I was just trying to link how our literature, how our granth, they say that how important the gut health is. In the earlier times, the doctors, they did not used to go for scan or go for so many testing and diagnostics. They just used to check the stomach. They used to ask, did you pass your stool? The gas or the bloat is okay or not? It is more or less than that, what is required actually. So a good food was always recommended. When food is pure, the mind also becomes pure. I need not mention that. And about the Ayurvedic principles, the Agni, the digestive system, the acids produced in our stomach, the Jatragni which you call, they are also important, and they also transform. If food is digested well, we are well. So Satvik Ahar is always recommended, but yes, no Ahar is bad, because as per the requirement of the body and as per the profession in which we are into, we may need to have the food, a different kind of food, because Tamasic food and Rajasic food, they also had a meaning. But in general, for teachers who are into a profession of just teaching, they should at least go for Satvik Ahar. This is the last one. I think I should stop here. This is a shloka from Srimad Bhagavad Gita about what type of food we should eat, like Yukta Har Viharasya, Yukta Cheshtasya Karmasu, Yukta Swadapno Bodhasya Yogaha, Bhavati Dukhaha. The meaning you can read, or I can read it for you in the end. The translation goes like this: Dukho ka nash karne wala. I think you read it, because in a hurry I'm not able to read in a hurry. But good food is the one which is, and a sattvic food is the one which is recommended for most of us. And there are so many references, I need not go into details, but there are so many literatures, so many granthas, they tell us about this. So the one last thing is, we should also not waste food. That is also important. We need to take a vow. We need to consider it sacred, and we should avoid wastage of food and treat food with gratitude, avoiding wasting, avoiding insulting, and just taking it for granted कि खाना मिल रहा है. This is the message which I give to my students, to everybody who meets me and who gets to talk to me for some time. I don't miss this chance to tell that please don't waste food. Wasting food is not just a crime, it's a sin, and nobody is going to, like, कोई माफ़ी नहीं है उसके, sin के लिए माफ़ी कहीं और ही मिलती है. So this is the last one, I think. The mindful intake, the proper digestion, wholesome food, they will be good for our body. Thank you. Thank you for listening to me.
Thank you, ma'am, and thank you so much. Being into the hospitality, that is something I already know, that wasting of food is almost not a crime, it is a sin. And that is what students should take in forward. Thank you so much for such inspiration session.
Yes.
I request my chair and co-chair to kindly share a token of gratitude to our speaker, please.
And I'm thankful to the chair and the co-chair also, I forgot to mention earlier.
Pleasure is mine, ma'am.
And thanks to everybody who at least decided to stay back.
Thank you, ma'am. Over to my chair, please.
This paper I can take it back. Thank you.
Thank you. Now we have last, I would say, talk of this session. This is an invited power talk, and the speaker is Dr. Saikat Mukherjee from Department of Biochemistry, Manipur University. The title of the talk is In Silico Predictions of Biosynthetic Gene Clusters in the Genome of Agroactive Pharmacologically Potent Bacterial Endophytes associated with Ethnomedicinal Plants of Manipur. So just to introduce Dr. Saikat Mukherjee is Assistant Professor in Department of Biochemistry, Manipur University. He's an alumni fellow of European Marie Curie Science Commission Human Frontier Science Program, and he has significantly advanced the fields of microbial biotechnology and protein biochemistry, and he is recognized for his purification of keratinase from A. mycoleptopsis species MBRL40, having the potential for degrading beta amyloid fibrils, which basically helps in Alzheimer's disease. And then he has also worked on mitochondrial membrane of mitochondrial tRNA import protein complexes or Leishmania species. And he has, to his credit, many prestigious publications, projects, and patents. So I request him to be short and brief and try to finish in 10 minutes.
Good evening, Honorable Professor Sunil Prabhi sir, Professor Shiv Kumar sir, and respected audiences. It's really an honor for me to be in this platform because as a life member of AMI, I had been associated with as a student member for a long time. I mean, only recently in 2021, I got a faculty position in Manipur University. And since then, I am trying my hard to set up a lab over there because, as you know, Manipur is continuously, it is under too many conflicts, economic blockade, consumables doesn't come on time, scholar has to take her all samples to other places of India to get the analysis done. So all these things we are facing, but luckily I have got I got one research scholar and she has done tremendous effort. Her name is Shantirani Thokchom. Just now she has given her oral presentation, and I am lucky to have a scholar like her that in spite of all these difficulties, when Manipur is burning, since even that time also she is visiting the lab, working late at the nights and trying to complete whatever assignment has been given to her. So whatever data I'll be showing to you today, although it's written that the in silico predictions, but I'll be giving some glimpses of what actually the research activities I am trying to build up in that small setup of mine. And whatever data you will find here is mostly done by my research scholar in this last 4 years since I have joined there. So although my topic is in silico predictions of biosynthetic gene clusters in agroactive and pharmacologically potent bacterial endophyte, so I'll give you some glimpses initially, then I'll talk to, in details about what we had been doing over here. So as this is the Manipur, and I invite you all to have a visit there in Manipur. And we have we have a small lab, but it is equipped with at least with basic facilities that I have got from Government of India projects. And this is, as we all know, Leonardo Vinci has mentioned, nature is the source of all true knowledge. And I'll show you how it applies very much in case of Manipur. So what is the extent of biodiversity that Manipur is having and how actually we need to explore them much. I was just now talking to Professor Syed Dastagar. We all know he is such a reputed microbiologist and how he has been also been instrumental in the development of microbiology in various aspects. So as we know, this is a small quotation about Imphal, a little paradise on earth, and Manipur belongs to the Indo-Myanmar biodiversity hotspots. As we know, there are 4 biodiversity hotspots in India, and Manipur belongs to one of them. And I'll show you what is the extent of biodiversity that you can be finding here. So we got one paper in Alzheimer's and dementia a few years back, about in vitro degradation of beta amyloid fibrils by microbial keratinase, and I am the corresponding author of the paper. This is actually it is not shown here. Something is written in red. So far, this is the only report of a microbial keratinase degrading beta amyloid fibrils. There has been no report so far globally that a microbial keratinase can degrade beta amyloid fibrils. And we isolated that particular keratinase, we purified that from an actinomycetes amycoleptosis species, and we got that from one of the hill areas in Ukhrul in Manipur. We got that. Now it is very difficult to get in these hill areas. After there has been the conflict between hill and valley, it is very difficult to go in these hill areas and collect samples. But luckily, before 2023, we could go there and my scholar could collect those things from there. Then, so these are some earlier photographs where we detected beta amyloid fibrils, and we used the particular, we call them two keratinases are there that we purified, CAR1 and CAR2, and we could detect that they are degrading the beta amyloid fibrils. I'll just skip them because these are all there in the publication of us. So I'll give you the link, you can download it from there. And there is some HPLC spectra which shows that when beta amyloid fibrils which are generated in vitro is incubated with the keratinase, it can degrade the beta amyloid fibrils. So these are the HPLC spectra, and there is some conjugate absorption spectra also. So these are all from the literature that we are, the paper that we have published, and this is so you can understand that there are so much, I mean, this kind of, we don't know, maybe we can come out with such kind of very potent enzymes later on also for further different other activities also. So we need to explore. Given a conducive condition, we may explore and we may find out that such kind of new proteins coming up which can have various metabolic, I mean antioxidant activities or anti-diabetic activities. So such kind of biodiversity, I mean we need to explore such biodiversity in Manipur. So another one is the fermented foods of Manipur. So we have a lot of fermented foods as you can find here, and this is another topic of interest in my that lab. So this is some photos that we have taken when we were collecting those fermented foods in the different fermentation conditions. And we could get some axenic cultures from one fermented bamboo shoot of Manipur. We did some in vitro probiotic potential experiments as per the guidelines. And we could also test on the zebrafish model. And we have some zebrafish set up in our lab. And we could see that when it is treated with a pathogen, Enterococcus faecalis, the zebrafish embryo couldn't hatch, as well as the egg also get disrupted. But when I am treating it with some probiotic, those bacteria, we got it the lactobacillus species. So we could found that it is actually recovering. So this is one we are doing, and the third one is now we are looking at so this is the third one also we are now looking at. The third aspect, the diversity in biosynthetic gene cluster in agroactive and pharmacologically potent bacterial endophyte. So as we all know about the biosynthetic gene cluster, I won't be going into those details because we are all aware of that. So the polyketide synthase biosynthetic gene cluster, NRPS gene clusters, and we all know that bacteria and plant, they are having the horizontal gene transfer system. So many plant metabolites are actually there, maybe producing now by the bacteria itself. So this I think I can skip because we all are aware of this. And different compounds are produced by these biosynthetic gene clusters. And there are some, as you might be aware of, there are some online resources, computational tools are there to detect such biosynthetic gene clusters like Anti-Smash, Bigscape, Bagel4, and Cluster Finder, and those can be valid. So you need to first do the whole genome sequencing for the students. You need to do the whole genome sequencing. Then you can put those sequences accordingly to the different this biosynthetic gene cluster detecting softwares. Anti-Smash is the best because Anti-Smash takes care of all the different clusters. Almost 80 clusters are there, biosynthetic gene clusters. So it can take care of. So you can, so Anti-Smash will predict that what kind of gene clusters which are producing the different secondary metabolites, which can, like it can have antibacterial activity, antifungal activity,
other potent bioactive compounds could be produced, and those prediction has to be validated by MIBIG database is there, which is considered to be a gold standard. So students, you can take care of this if someone is interested in detecting of working on biosynthetic gene clusters. So this is a very interesting piece of, interesting area of work. Gradually, people are trying to come up with heterogeneous cloning of biosynthetic gene cluster after the identification. So many different systems are being developed nowadays, and many recent papers are coming up. So this can be an era of heterologous cloning of biosynthetic gene clusters and coming out of those products. It's very difficult to purify the natural products from the system itself. So better we can go for cloning of these biosynthetic gene clusters and trying to purify that compounds from the heterogeneous system and test their activities. Some of the good reports are coming up. I found in 2020, so many papers are coming out where many new antibiotics can be gradually actually being cloned and purified, expressed and purified from different bacterial artificial chromosomes and yeast artificial chromosomes. Right now, I'll show you after we, now that we have predicted some biosynthetic gene clusters, we are actually now trying to go for the heterologous cloning of them. So this is my scholar. She just presented her talk some time back. So she, you can see her. So she is actually, so here she is showing only one of these bacteria, Cellulose-Microbiome cellulus MUKR5 that she has isolated. So Iris levigata fish is it is only reported in Manipur. So it is the very newly reported Indian flora. It is only reported in 2021. It was not reported earlier, having various, neurological, it can in hysteric patients or like epilepsy. In those cases, it is found to be having a very good effect. Actually, my scholar has been instrumental. She has actually taken 8 medicinal plant, and so although she has presented on Iris levigata, but she has worked on 8 medicinal plant which are having ethnomedicinal uses. She has got many different bacteria, many bacterial isolated, and actually that was her research work. Although I'll just won't go into all those details. I'll just talk about the in silico detection, what she has done. So some of this bacteria she has already identified, whole genome sequence has been done. Many are there. Actually, we have done almost close to 10 bacterial whole genome sequences. So all those data are there, and then she has analyzed what could be the possible biosynthetic gene cluster. Those are present in these compounds, and using anti-smash and then validating by MIBIG database, and you can see all those different. So as you know, in biosynthetic gene cluster, what happens, there are 4 different type of enzymes are there. Core enzymes, tailoring enzymes, regulatory enzymes, and transport enzymes. Only when these 4 enzymes could be there present in the software could detect, only then a biosynthetic gene cluster can be annotated. And once it's annotated, you need to be validated using the MIBIG database. And if it is identified, then you can properly get a clear picture that where exactly from where to where your genome sequence, your genome cluster can be there. And then you can go ahead with the cloning. So these are different bacterial strains. So we have all got the different whole genome sequences already done. We have already got the biosynthetic gene clusters. So I'll not take much time. So my sincere thanks to ICMR, ACRB, and ISRO for providing me the funds to carry out this project. So now, after I have got a huge number of bacterial isolate that I have got from 8 ethnomedicinal plant, and many of their 16S ribosomal RNA sequences has been done, as well as many of them are whole genome has been characterized, and the gene cluster also we have got, few of them we have got the gene cluster analysis also done. Now my plan is to go for some selective gene cluster analysis. I'll just, this thing I don't want to, I'm skipping this because these are some backup slide, how we have done all this gene cluster analysis, and this some kind of anti-proliferative activity also we have tested. We have got many compounds which can have proper antifungal potential, plant growth promoting potential, and anti-proliferative activity as well. So based on that, we have shortlisted all of them. So I won't be going. So she is working on this plant growth promoting activity also. So as much in silico prediction we have got, we have also tried to come up with wet lab experiment also to confirm that whatever has been predicted from the anti-smash and MIBIG, some of them can also be validated using the wet lab experiment. So I am in the process for identification, for further analysis on this aspect, and I'll be happy to collaborate because it's very difficult always to be inside the lab, working under some constrictive conditions. So thank you so much. Thank you for giving me the, thank you.
Thank you, sir. Now I request my chair and co-chair to present a token of gratitude to our speaker. Thank you, sir. Now I request our Vice Chancellor of the University, Professor Ajay Kumar, to present a token of respect and gratitude to our chair and co-chair. I request you, sir, to please join us on dais. We are thankful to our chair, Professor Pabbi, and our co-chair as well for being a panel of this session. Thank you, sir.
Hello, hello, hello. Hello, hello.
Hello.
A very good evening to one and all present here. I welcome you back for the last panel discussion of today's session. It is a collaborative dialogues panel discussion creating enabling ecosystem in HEIs. It is focused on fostering innovation, research and entrepreneurship within the higher education institutions. This session brought together the distinguished academicians, industry leaders and policymakers to discuss strategies for building supportive environments and nurture creativity, startup culture, and interdisciplinary collaboration. The discussion emphasizes on the importance of institutional support systems such as incubation centers, research funding, policy frameworks, and academician partnership. It highlights best practices for the successful models across India and explored the ways to empower faculty and students to translate the ideas into impactful innovations. With this, I would like to invite the moderator for the session, Professor Prince Sharma, the President of AMI. Can we have a huge applause for our moderator for this panel discussion? With this, I would like to invite the panelists on this dais as well. Professor Ravi Toteja, the Principal ANDC UOD. Professor Sandeep Sharma, the Pro Vice Chancellor at Dev Bhoomi Uttarakhand University. Professor R.C. Kuhad, the Chairman AMSE, and Dr. Anveta Gupta. I request Dr. Anveta Gupta, the panelist for this panel discussion, kindly join us. Okay, she's here. And a Pro Vice Chancellor of Dev Bhoomi Uttarakhand University, Professor Sandeep Sharma. Over to my moderator. Thank you, sir.
Okay. Thank you so much for inviting us. I think AMI's, this particular AMI conference, one of the highlights has been that it has included, besides lectures, lots of panel discussions on important topics. Previously, we already had held panel discussions on industrial collaborations, startups, and yesterday we had also on next-gen education programs. And today, another important issue is, which the education is facing, the HEIs, higher education institutes are facing, is do we need to produce the graduates, the degree holders, or do we need to produce the enablers? Enabled to face the world. Enabled, skilled to find jobs, to start their own businesses, contribute to the economy of the country, contribute to the development of the country. It is this particular burning issue on which we decided that we will have another panel discussion where the eminent gathering from here, India and abroad also, we have one person from abroad also, that what is the scenario today? What is the scenario of this making enablers or creating enablers in higher education systems? With this, because we are not having much of the time, there is much left for the day, I will go directly to Dr. Tuteja first. He is the principal of a college and he is very much involved in this particular education system. So I would ask Dr. Tuteja that how do you think that our country is progressing and our colleges are taking up this issue, that now from now on we have to produce enablers, we don't have to just produce the degree holders.
Thank you, Professor Prince. Good evening to everyone. I am Professor Ravi Tuteja, Principal of Acharya Narendra Dev College, University of Delhi. As Sir has rightly said that we have not to produce graduates who are knowledgeable, but those who can think beyond the box. Those are skill enabled and they are confident and they can take part in decision making. And this is possible only when other activities are included in our curriculum. So to make them enable, we should have skill-based courses, which has been introduced in National Education Policy 2020. We have skill-based courses. In addition to this, that entrepreneurship plays a very important role. So higher education institutes should have the entrepreneurship cells. They should start with the incubators so that any student who come up with the idea, institute is able to mentor that student so that to make them job ready. In addition to this, integration of internships and research is also important into our curriculum so that we make the job ready students, and also this helps in making the student informed decision. So NEP 2020 is doing its part, and earlier all these activities used to be, we used to say these are extracurricular activities. But now I think it's the time that we should include all these activities not as an extracurricular but into the curriculum of our education. And yeah, this is how we will be able to make citizens which will be able to make the future nation very progressive. Thank you.
Thank you, Dr. Tuteja. I feel even if the colleges are in a remote place or in a tier 1 city or a tier 2 city, all of them must be vying for now that to remain in competition, to have the good grades also, rankings also. Maybe this may become a vital component that, okay, we are not just producing the degree holders. Yes.
Another thing I would like to say that with the coming of ranking, it has more of become a competition. So it should not be a competition. We should work on those areas where we are lagging. and should improve those areas so that students can get the maximum benefit. It should not be competitive that this particular college has got the first ranking and other college has got the 20th ranking. So should work on those areas where we are lacking so that students can gain the maximum.
Thank you. I will extend this same discussion to Professor Kohar. He's a renowned educationist, being the Vice Chancellor of a reputed university also, and to many committees, policy making committees also in the UGC. Sir, now again, one thing is established that this is an important issue, creating the enablers. How do you feel that the higher education institutes are facing some challenges in creating such enablers?
Yeah, I think you are very intelligent, Professor Prince Sharma. Before you decide to put a questions that what a person is thinking about. That's one thing. Let me share with you that today itself I have joined as Vice Chancellor of one of the private university, that's Swami Vivekananda University at Kokrajhar, Assam.
Congratulations.
And I seek your support and say help in the time to come. I will remember that I was at Dev Bhoomi University and I took this responsibility, and of course we have to work on that. It's very important, Professor Prince, that when we are talking about that enabling the educational ecosystem, in universities or higher education institutions. Fine, we are in a process for the last 5 years developing the curricula, and I'm afraid that many of the universities and the institutions, they are simply developing the syllabi, not really developing the curricula, and which is, say, one of the very important, aspect of the ecosystem, if you think about. And then when we know about the various facets that we have to train the students, not only in transmitting the knowledge, not only enriching their knowledge, but skill, make them, say, good trainers so that they go to the market and get the employment, or we may inculcate the scientific temperament or the research temperament, whichever the area you think of. If we really say we are planning to make them to know our Indian knowledge system or the life values or human values or ethical values, because our whole education system, as per the national education policy, is focusing on holistic development. Therefore, in order to really developing this ecosystem, every institution need to do a SWOT analysis. What is your existing manpower? Up to what level they can handle the curriculum you have developed and the outcome you have decided, where you feel that they need, say, training in a particular area, that also you need to decide in between, in the beginning itself. And most important is the challenges. Because, Professor Prince, if you agree with me, many of the institutions, while developing these, say, curricula, they have not identified the challenges. If they identified, they are not bringing forward in front of, like, the administrators, from where you need help. With that, I think we have to get a courage to come forward that, hey guys, these are the challenges. We are ready to meet out the outcomes as per the credit framework or the national higher education qualification framework you have put forward. We will be able to achieve. And then, when we develop this ecosystem and the target we are fixing, I think we should work and develop a strategic plan, say, in a phased manner. In a phased manner. If now we are entering into the interdisciplinarity, then we should say that after 5 years, we want to go to the multidisciplinary. Or maybe next 5 years to the transdisciplinary. It's really very, very important. And of course, University Grant Commission has been providing us guidelines, say for example, establishing the Research Development Council or Research Development Center. Not only that, the kind of incubation centers, maybe startup-based or entrepreneurship, innovation centers. But I think simply having the incubator center is not going to serve the purpose. The institution has to work for that, the similar kind of infrastructure as well as the expert. We don't have the experts. Therefore, we need to go beyond, say, our boundary, and we need to find out the experts we need from industry or corporate sector if you have the business management courses. Good researchers. Like every institute should think of, I think, having one very important component in your educational ecosystem, a center for lifelong learning. I tell you, whether you agree with me or not, but the teachers who have been superannuated, they are the real treasure for us. And if we are not making use of that, the same way as we have not been able to create the data about the wisdom of our elders, similarly we will be the losers in the next year's time if we are not going to focusing on making use of the knowledge, the skills, and the art our senior, say, the faculties or the researchers, they do.
It's very true,
It's
sir.
very, very important
Yes,
if you have to really strengthen, say, the whole education system.
It's very true, sir. We'll come to Anvita. She's from USA and she has studied in Stanford. My point of bringing her to this panel was that when Indian education system is going through such a change now, how about you think that in your university or in foreign universities, are these enabler systems already, or what kind of system you have to create, which are already creating enablers, or you have some different system or different thing calling it not enablers or something else?
Yeah, that's a wonderful question. And first of all, hi everyone, I'm Anvita, and thank you to Dr. Prince for inviting me to this panel. I feel very honored to be with such distinguished fellow speakers, and certainly I'm not a professor yet, but I come from a family of professors and educators. My family in the US has a very strong business and education technology working with the government, and I'd done my bachelor's and master's in computer science from Stanford and worked very closely with the entrepreneurship system there, the education system there, and I'm also a Knight Hennessy Scholar, so that's a very small community of students in Stanford who they give the next level training to and a lot of opportunities for self-development. So I see a lot of that. I think some things that we have in the US that I would say the Indian education system can also benefit from. And these things are growing in the US too, they're not already there. One thing though is remote learning. So that took off in a big way after COVID, because in the US they shut down all the schools. I think in India, luckily they didn't do that. But for at least a year and a half in the US, schools were completely shut down. So all classes were online. And it's very hard to teach students over Zoom, so teachers struggled a lot, students struggled a lot, but they got the systems in place to have remote learning access to courses and curriculum available to students 24/7. And so that also lets teachers do more activities in the classroom, because in the classroom you're not then just giving knowledge, the knowledge they should have studied, they should have read the textbook before they came, right? So now with remote learning, when they come into the class, they can actually do activities with the teacher. So that's very useful. I think, in the age of AI, knowledge-based teaching will become less important than skill-based teaching, right? And I don't know if that's one of your questions that's going to come up. So getting students involved in research early is another very big key factor. So I think that allows students, and I've taught coding camps to young girls in India, in Chandigarh, New Delhi, and in the US. And students, we teach them coding, but then they have to come up with a project. They have to make a website about an issue that matters to them. They have to develop app prototypes. And so they come up with amazing problems they see in their community. So they make, websites to help teens with mental health. They say, our classmates are struggling a lot with their mental health. We want to make a resource for them. So they make that website. Some girls say, in my town, we have this problem of female infanticide, so we want to make an app to report the doctors who do it. We'll publicize them, and they come up with these creative ideas. So that's I think getting students involved in projects early is very, very important. The last thing I'll say, which is pretty unique, is that the Knight Hennessy program at Stanford, which I'm a part of, they give everybody a storytelling class. So every Monday they make us come to lunch, and they're not teaching us, there's no knowledge being imparted here, but they say, come up and we'll teach you how to tell a 3-minute story. Because, in life, the most important things is humans are storytellers. So you can do all the research and understand everything, but if you can't tell what you're doing to an audience, then nobody cares, right? So if you think about Steve Jobs, all these people, they're great storytellers. So I think, in India and even in my own family, we focus so much on knowledge, and we have all the knowledge. We have more knowledge than 99% of the people out there in the world, let me tell you. But if you don't tell the story of that knowledge, then you fall behind, right? And then you get frustrated that I have all this knowledge, why don't people care about it? It's because you couldn't tell the story. And this is a skill you can learn, you just need to practice it. And I think that, students love it, so why not have something like that in schools?
I think you're very right that creating enablers right from the school, not why wait for HEIs, and secondly to have the communication skills, like you say, the sellable. You should be selling yourself actually. Yeah. If you can't sell yourself, no point of having a good research and keeping it in your house actually. Wonderful. That's there are some lessons where India can take from US or the vice versa. Other countries can take from us. I come to Dr. Sandeep.
Sandeep.
Yes, you are from a private university, and you see that in this these times, when the universities are competing, I can see that when I came to Dehradun, I think there are 18 private universities in Dehradun, something like that, is it not? Yes. They're all competing so much with them. Now, with this such a fierce competition, do you find some challenges, and how are your universities coping up with that? That you have to create enablers, not some degree holders only.
Thank you for that. For the community here, I'm not a microbiologist. I am probably here because I am the one who's responsible for spearheading NEP implementation at the university, and I bring a perspective of a layman as far as microbiology is concerned. And maybe sometimes
it's just not about microbiology, it's a general discussion on creating enablers.
So you see, what I have always felt is, and what I have strived for, is India has been the land of skill-based ecosystem. Ancient times, our the division of work or the labor or everything was skill-based. So the Varnas were nothing beyond but the skill-based. Coming down to the question, sir, yes, the competition is fierce, and we at Dev Bhoomi are implementing a national education policy to the very dot. The challenges that we face are what fellow colleagues have already spoken about a little. The policies are there in place, the systems are there. The curricula, by and large, in the age of information and technology, is not very difficult to cope up with. But the problem is the last mile delivery. Now, what happens is, across the globe, these things work because they are supported and supplemented by industry. So the kind of projects that are required for the next level of research are shared with the academicians, and the students get direct hands-on working there. So the next line of workers is getting ready while they are still studying in their UG, PG and PhDs. What happens in India, unfortunately, a large number of industries, they are not really very confident of the Indian academic systems, if I may be allowed to say so, right? So what happens is the learning of the faculties themselves limits to the books or to the knowledge available, thanks to the remote learning accesses, what my fellow colleague mentioned a little while back. A lot of things have improved, but still what remains is the motivation and the depth of getting the things done and implemented, so that the real student, the students like of those who are sitting over here, they get the pie of that and they appreciate and start moving in that direction. In addition to that, I will also like to bring upon a challenge that now is the age of experts with multidisciplinary flavor as well as interdisciplinary flavor. So they not only have to fit in a system where people from different disciplines are contributing, but they also have to understand those different disciplines so that they can bring to the table their perspectives and their learnings to make meaningful contributions. So these challenges from Indian perspectives, I believe not for category A universities like IITs, IICs or the likes, but everybody below that domain will have to struggle till the industry finds a way to accept us as partners and start contributing there. So that is the biggest challenge that I foresee. And then the students, of course, they have to also understand that they are in an age of startups. They're no longer searching for government jobs where they will retire at 60 and then pension will follow suit.
Yes.
Yep. Thank you.
I think sooner they learn this, the better is for the students that don't go for the jobs, go for your own businesses or own entrepreneurship. Now, anyone from the audience has to make a comment on this, particularly the students, that are you getting ready to be the enablers, or you are just going for the rote learning and getting a degree only? How many of you want to be enablers? None of you.
Can we have some?
Or you do not understand what's an enabler, that you want to be an enabler, that enabler means who is able to face the world once he is out of the college, who has got everything, that the world will come to him, he will not go to the world. Who wants this?
All the students should raise the hands.
Who wants this? Okay, very few.
Good.
Very few. Let's say that. Let's say that. Yeah, we should be doing that actually.
Professor Prince.
Okay. Any particular comment can come from one person? You want make a comment quickly. We have set the ball rolling, probably to make people understand what we are doing.
Hello. Hello. Yes, myself Jaideep Chauhan. I'm pursuing my master's over here. Simple question on the dais I would like to put.
It's like nowadays we see one small kind of what we can say problem into the industry and academia relationship. It's like many of the time I have seen one small thing like whenever student is going out there as an individual to ask for the internship or looking for some hands-on training, the corporate or the industry demands for certain amount of money, right? Like at the same time, there's the one single question, like student whenever he is going to there and to learn some things and to have some experience over there, at the same time he might be delivering some certain amount of work from his side. So what kind of expenditures my industry might be facing for the having like to train that in a particular intern, and what kind of what we can expense corporate needs to pay for that particular learning?
Okay, I think, okay,
Yeah.
Dr. Kuhar can answer that.
Sure. I think very good, say, kind of query or a inquiry you have raised. First thing is, because industry are also wanting to earn from the students, those who come as interns. On other hand, government has really defined a special scheme under CSR that they should come forward and give interns facilities for a student. But majority of the industries, they don't have created that kind of ecosystem what we have been talking about. Therefore, say government needs to monitor, say, such kind of infrastructure, and they should make it compulsory. Because now industries have developed, say, some agencies outside the industry from the market and using their CSR by these agencies to train, say, students. And several have been found fake. So that's the scenario is going on. Dear friends, it's very important that if we have to enable you, I think every institute has to see first what maximum we could do to enable our student. Say, begin with the soft skills. Okay? Don't think of going industry for every kind of skills. And everybody should not think of that unless until industry is not coming, we won't be able to make you skilled. That we should, these are kind of, along with this NEP, institutions, it's a big challenge. And when it's a big challenge, why we are only passing on the bugs to the industries? Say, for example, professors have said very rightly that the A category institutions, IITs. If I as a student ask here to all of you that if I go to the IIT because I got an opportunity and I as a student of IIT get all sort of facilities, so why a student who is admitted in Dev Bhoomi should have the different kind of, say, the training or enable parameters? That's the question. And that question you youngsters should, I think, ask everybody. And if we will equalize these facilities, I think we will start forgetting slowly by the industries, because we should be self-sufficient, institution as a whole, so that we can enable maximally our students. Then, after certain grade, you need, say, training. Then, of course, industries should come forward. Can we think of, like, I think several lakhs of students we do have. Do we have the industry or the industrial setup so that they can come forward and train our youngsters? I think we should be very clear about these kind of things. ये हम बोलते रहते हैं, अखबारों में छप जाता है, फिर एक होता है, मैं जब स्टूडेंट था, then I used to say that academia and industry, they have been brought at one platform. And today also we are talking about that industry and academia. Although there is a progress, But when I was student in 1980s, master's student, it's 45 years. I don't see personally any, say, major progress has taken place. It's very, very important, I think.
Yes, sir. To continue with this, actually, let me tell all of you, you are from the business administration and there are other people from different fields. The times are changing now. A time was there when industry used to visit the institutions, find out the best talent and hire. But now the times are changing, even in outside and now in India, and Anvita maybe can tell whether I'm right or not. Now what is happening is that industry doesn't want degree holders. Even if you have not completed the degree, doesn't mind. They don't mind. They want a talent. They want you to come to their industry for internships. There is a fierce competition. Let's say they have selected 10. They pay for them. And the best ones, they will give the jobs to them. Even if you have not completed your degree. They prepare you as per the industry needs rather than what education you are getting in the institutes. The times are changing now. I think you may have got your answers now. So it's a fiercely competitive world actually. I was wanting to come to this point at the last, but because of your question, so I have, maybe Anvita can tell more about this on this aspect. Is this true or not?
Half a minute to bring one more problem which I have, say, experienced. It's very true that even if you haven't completed your degree, you are going as interns, they will appoint you. But first thing, you develop the habit of to be absent from your course. And this trend has entered not only from engineering institutions, but the usual, say, the courses in various universities. Absentees has become a trend in our higher education institutions. Specifically, all B.Ed courses in this country, और जितने सब लोग बैठे हैं सबको पता है, जितने हमारे से बाहर बैठे हैं पंडाल से उनको भी पता है. If our teachers are coming, having the degree without attending classes or having any training, then what kind of the enablers we are expecting in these schools? That's very important. और देखिए इस चीज को कौन सबसे पहले सोचना चाहिए और बोलना चाहिए, आप लोग बैठे हैं और ये टीचर बैठे हैं. Otherwise, I think simply we'll be using in terms of our words that we want to improve the education standards, we want to enable these our students. I think there are several problems which we need to discuss and take into consideration and work on.
I would agree, definitely you should finish your degree, number one. And for industry, you can take the example of Zoho, right? They are now hiring students right out of 12th grade.
Yeah.
They train them in their academy and they, get them ready to work. And so yeah, you don't need a degree to get a good job now if you're smart. That being said, I do think you should finish your degrees as students because it's for yourself, right? If you have 12th grade, you go to Zoho, tomorrow Zoho decides for some reason to fire you, what are you going to do? Now you're 30 years old and your 12th grade pass, or I don't know how they say it in India actually, but 10th grade pass or whatever. But you need to have your degree for yourself so that you have that security, right? Job security. That if, this job doesn't work out, I can go get another one. So education is very important, and it's the one thing that nobody can take away from you as a student.
I think it's right that you may get the job, but sometimes in India also I have seen that some people get a good job after a master's, but they don't get the promotions. They get stagnated to that place only. If they finish their education, do their PhDs, then they go up the ladder also. That's very true also.
I'd like to add something. Sorry,
no,
please.
please.
Thank you. You see, the NEP that we are talking about, that has provided solutions to this kind of a problem.
Right. That's what I was about to say.
Just like the West, I had a co-worker with me while I was at GE Power Conversion, working with them in the R&D. The guy started as a diploma enthusiast in the company, and he did his masters, and he grew with the company alongside. So that kind of infrastructure and that kind of policy points have been put in the NEP, and where we call it a multiple entry, multiple exit.
Right.
People can exit at will, they can serve in industry or the jobs they like, and they can come back. So that is what we are heading for. In future, what will be happening is that we will have smaller certificates, and gradually, like the culmination of those certificates, the requisite credits will translate into a degree. And I think that is a very important thing that has come out of NEP. And if we are able to play it right, we will see the change, the kind of worries our senior panelist here has raised. They're very valid, but then those concerns will be addressed through that only.
I think NEP has been thought of very well.
Yes.
But if it is implemented in a proper way,
in the true spirit.
In the true spirit,
Yes.
actually. And that brings me to Dr. Tuteja. He's also wanting to say something, but I will also say that because this, yesterday also we were talking about that this maybe needing an overhauling of your syllabi and courses in such a way that we fulfill the mandate of NEP of producing startups or create enablers rather than the degree holders actually. So what exactly you think is going about how to change, or what changes you think we should be making the syllabi?
Let me finish that query again, that the positive point of NEP, another positive point of NEP 2020 is that you can store your credit in academic bank of credit. So it's not that earlier what used to happen that student has to finish all the 3 years of graduation or 4 years of graduation from the same university. But now there is a provision that a student can gain the credits from one of the institute, and maybe because of any issue he or she has to move to some other institute within the India, those credits will be counted. For example, the student has completed first year in X institute, the credit will get stored in an academic bank, and second year the student can do from Y university. So this is again very positive thing about NEP 2020, and definitely NEP is evolving. It is just a starting phase of NEP, and as we know, whenever the new policies are put into place, there are teething problems. We are also facing some teething problems. Definitely, we are going to solve this. And now regarding to your question, sir, regarding curriculum. So it is very important that curriculum has to be designed in such a way so that the needs of industries have to be taken care of. So the main thing is that there should be a strong collaboration between industry and academia. And also the institutes have to develop R&D cells so that what is the need of industry, that should be put into our curriculum. And as I earlier said, that research is very important. So mentoring and how to submit the research proposal, there should be a guidelines which should be laid down by the institute so that it should fulfill the mandate of NEP. NEP, as we all know, believes in experiential learning, hands-on. Hands-on is very important. So the curriculum should be designed in such a way that more of hands-on activities should be there. As we all know that there is a very important saying, I hear, I forget, I see, I remember, I do, I understand. So for making anything understand, one has to do hands-on. So for the holistic growth, all these components should be integrated into our curriculum.
Right, thank you. So I will be having on the last part of this because we have lot more following this. One part, quick from all of one question, a quick answer from all of you that I think we direly need industry advisory boards who can guide us making our syllabi, making changes in our syllabi, and how do you think, what is your take on that, that they should be guiding us, what kind of syllabi should be there?
Yeah, definitely there should be industrial advisory board so that education institute should be guided what is the need of industry.
Quickly.
Yeah, this is, so one institute should have advisory boards, definitely.
Dr. Kohard, we agree to that. And how to implement?
There has to be, yes, each institution can have, say, one industry advisory board, but I think that for each science-based, engineering-based, technology-based, or mathematics-based, say, the department, they should have in their board of studies or committee of courses at least 2 persons from industries.
Yeah.
Because at institutional advisory board, say, will have, say, specific role, but to develop the curriculum from the grassroots itself, there should be experts from, say, related, because each department should define their, say, areas of expertise.
Truly, sir.
And based on that, you should get the experts, which is very helpful. Not only from, say, industry, but, say, management and your commerce.
Truly.
You should get people from the corporate sector.
Industry means their management also, or
Yes,
production
but
management.
now we have to see, because when we talk about the educational system, we, being from science, we only talk more and focus on these things, because there are subjects left from social sciences, arts, humanities. As far as our students say concerned, this flexibility system is very good. I'm taking just a minute with your permission. Exit and enter. But I have a say question to all of you. Say, for example, I complete first year of BA Political Science and I exit. What I'm going to get in the market? I'm a student of BA Hindi. I complete just 1 year and say leave and come. Even I'm allowed for 8 years or 10 years as per national education policy. Because idea is very good, scheme is very good. That facilitates, say, our students. But on the other hand, you call the examination branch and ask them what kind of the problems you can face. That's why just in the beginning I said, NEP, I am the greatest advocate of that because I have been involved from that. Because scheme I बहुत बढ़िया scheme. But now the whole planning has to be developed, how it has to be executed. I think that's in our hands.
Right.
That's in our hands.
Truly.
तो
Implementation.
हमें तो वो syllabus अगर सोशल साइंसेस का बनाना है तो हमें ये एक्सरसाइज करनी पड़ेगी कि 1 साल में एग्जिट लेके जाता है बच्चा, एक सर्टिफिकेट लेके जाता है, तो उसको कोई ना कोई मार्केट में ऐसी जॉब अवेलेबल होनी चाहिए. दैट्स द ड्यूटी ऑफ द फैकल्टी. सिर्फ देखिए एनईपी को प्रशंसा कर देना और हम भी बैठ के ये कहेंगे बहुत बढ़िया सिस्टम है, बट व्हाट द बेनिफिट वी आर गोइंग टू गेट आउट.
Yes, sir.
क्वेश्चन है यहीं पे.
Yeah, that's why in the first year, when what courses we have to give in first year, they are very selectively they have to be and judiciously they have to be thought of, so that if the student goes after first year by taking the certificate, he takes benefit of those courses.
Your every course
Yes.
should be skill oriented.
Right.
In the first year. Yeah.
Sir, one thing.
Quickly, sir. Just quickly.
एक और क्वेश्चन है मेरा. एक स्टूडेंट 1 साल
में सर्टिफिकेट लेके चला गया, 2 साल बाद आ गया, एक फिर आया डिप्लोमा लेके चला गया, फिर एक आया 3 साल पूरी करके डिग्री लेके चला गया। एक स्टूडेंट लगातार 3 साल में वो डिग्री लेके जाता है, तो देखिए बेनिफिट उसको है जिसको आपने फैसिलिटी दी कि उसके पास 3 सर्टिफिकेट। अब मैं एक 3 साल तक लगातार जो है ना डिग्री करता रहा, मेरे पास एक सर्टिफिकेट। डोंट वी टीचर्स, डोंट वी academicians should think of this? Anyway,
Okay. So
yes, sir, I would like to add one thing that every institute has a IQAC. So if we see the composition, it is a mandate that 1 or 2 members should be from the corporate world. But with NEP, with the advisory board, it will become more regulated kind of thing. But still, at present also, there are corporate members in the IQAC, and every education institute should deploy those corporate members in their respective IQAC, and those members can guide us how to go about.
Right. Okay. But last question to Anvita is that because you have been in India, you have been in USA, that when we talk about these creating the enablers in higher education institutes, how important is to build up the international collaborations and exchange programs, how vital a role they can play in this creation?
I think I'm very in favor of international collaborations. So my company, AI Novo Biotech, I founded after getting a lot of international exposure. I did an international research trip in Switzerland at ETH Zurich. That was a fellowship paid for by the Swiss government, and they have this program to bring students from the US and all over the world to Switzerland and do research. And so I got the opportunity to work in AI for drug discovery at a very great lab there. They were working a lot with Merck and Genentech and Roche, all these pharma companies. And seeing the success that AI was having in drug discovery in this environment, and seeing how they had really combined computers with wet lab. So they had both in their lab working side by side. I worked the whole fellowship with medicinal chemists. I'm a computer scientist, so they really had a different skill set. So I think international collaboration gives you that exposure for students, that how research is done in different parts of the world, how research is done globally, and then they can bring those ideas back to their home countries, to their ventures, to their labs if they're in academia. So that's really important, I think, to create enablers, that if you see only one thing your whole life, you don't know that there's more out there, right? It's like the frog who lived in a puddle versus the frog who lived in the sea. Yes. That story is there. So
That's very true about that. So anybody wants to have a question or is anything, a comment from the audience? Again, the same person. Lastly, because that one we are going to finish that now.
Thank you so much for providing me this opportunity. Sir, I don't have any questions, but I just have one small point to put on the table.
Very good. Okay.
It's like I do personally feel like when we talk about NEP 2020, more or less we are focusing on the higher education. But from my perspective, it should start from the standard 1. Like if we are training students in a way from the school system, because after studying the same thing and in the same pattern for 12 years, students are not ready to get out of that hedge and get their hands on the training. So that's the small thing.
You are very true. It's very true that the culture should begin right from the start, from the toddlers, from the school only. So with this, I think we have one last point from sir. Closer. It's not.
There is a lot of emphasis, dear friend, in schools. Even NEP has been taken up more seriously in schools of our country than higher education institutions. Thank you.
Thank you so much. Thank you. I'm thankful to all my panelists here and thankful to the audience. And this panel discussion is again, I am reiterating that again, AMI from now onwards has been thinking that only lectures will not make a conference, it will not complete a conference. There should be more of industrial partnerships, or they should also come forward to take part in such conferences. Startups should come, such panel discussions should follow. And with the time, more and more AMI conferences when we'll have these kinds of discussions, they will be more and more rather than the lectures. So thank you so much again. Thank you.
Thank you, sir. Now it is a time to share a token of respect and gratitude to all the panelists here. I request Dr. Digvijay Singh, the Dean Student Welfare at Dev Bhoomi Uttarakhand University, and Mr. Sachin Sharma, the Deputy Registrar at the Dev Bhoomi Uttarakhand University, to please join us on dais to share a token of respect and gratitude to all the panelists. I request everyone to join in front. I start with Professor Ravi Tuteja, the principal at ANDC UOD. Audience, can we have a unit applause for one of the panelists for this panel session? The next panelist, Professor Sandeep Sharma, the Pro Vice Chancellor at Dev Bhoomi Uttarakhand University. Audience, I'll tell you, Kohart sir is still on the stage, so the clap should not stop. Next, I request sir to please give a token of respect to our Professor R.C. Kohat sir, the founding chairman of AMSC. Now I request sir to kindly share a token of respect and gratitude to Dr. Anveta Gupta. And I'm thankful to the moderator of this panel discussion, Professor Prince Sharma, the president of AMI. I request sir to please kindly give a token of respect and gratitude to Dr. Anveta Gupta as well. Thanks to all the panelists and the moderator for this panel discussion. Thank you, everyone. Now it is a time for the AMI Journal Body GB meeting and award ceremony. Now I request Professor Namita Singh, the General Secretary of AMI, to kindly take over the further sessions. I request panelists to please join us on dais.
Good evening to everyone. Now I request President, please come on the desk for the proceeding of 66th annual meeting of AMI. Yes, वो उसमें से निकाल देती चल रहा है ना. Now AMSC Chairman Professor Kohad. सर कहाँ गए? कोहाट सर कहाँ गए? Past President Professor, sorry, and Dr. Pabbi. Dr. Dudeja, past president. Please come on the desk. Sir, आ जाइए. Yes, sir, please. Welcome to Punjab. नहीं ठीक है. It's fine. इसकी ज़रूरत नहीं है. With the permission of chair, may I? Okay. So I, Professor Namita Singh, General Secretary, welcome all the AMI life members, our President, Professor Prince Sharma, immediate past President, Professor Sunil Khare, he is not present right now, and past President Professor Sunil Pabbi, past President Dr. Yoginder Singh, Professor R.C. Kohad, AMSC President, and Professor Rooplal, past President, Dr. Ganju, Dr. Meenu Sharaf, Dr. N. Vasudevan, Professor Shamsher Singh Kawar, Dr. Anil Panghal, Joint Secretary, Dr. Meenakshi Prasad, Editor-in-Chief, Dr. Livlin Shukla, not present. So, with the request or permission, I start the proceeding. Before I start the proceeding of the 66th General Body Meeting, I present the executive summary of our annual report. The Association of Microbiologists of India was established in 1930 and in Lucknow and is one of the oldest organization, most knowledgeable scientific society in India. Since its inception, the society has contributed appreciably to the development of microbiology and has recognized it as an essential branch of biological sciences. The association also publishes a quarterly research journal, Indian Journal of Microbiology, which is now at its 65th volume, issue 3, September 2025, published, and it's currently co-published with the Springer Nature Limited in 2024, all the INGM issue published, and 128 total article, total downloads totally 2,18,900. Total subscriptions are 6,400 presently. Total royalty we received this year is 48,74,389 rupees. From December 2024 to November 2025, 4 Central Council meeting has been held, that is on 11th May 2025, 31st May, 28th September and 2nd November 2025. Various awards are given by the Association and Academy of Microbiological Sciences. AMI Lifetime Achievement Award, AMI Young Scientist Award, AMI GB Manjrekar Award, AMI JB Bhatt Award, and AMI Rana Memorial Award for the Best Poster Presentation, AMI BN Johri Award, student travel grant in various categories selected through peer review have also been declared. 24th special awards announced by the AMI Springer Award out of 24, 12 for the oral presentation for the young scientists and 12 poster presentation for the young scientists. Each award having the poster certificate and the €300 book to each of the winners. The association is progressing well with all 3 issues of volume 65. Journal has a 5-year impact factor 2.5 as an impact factor of 2024 is 1.6. The journal has also contributed significantly to association total income 48,78,389. A large number of new members have been enrolled with the association. So presently, member stands at the 5,683. That is on the 1st November 2025. The members added from the 2020 when I take over the charge as GS, that is a 1,350 new members join our association within the 5-year span time. This growth reflects the association expanding visibility and influence within the scientific community, both in India and internationally. The association has successfully submitted its income tax return for assessment year 2024-25. Annual book of account has been audited and the balance sheet has been compiled by the auditor employed by the general body made. Within the country, the new AMI units were established. One is the AMI Northeast unit at NEU. Now a total 45 AMI unit established. And recently we also received request from Mumbai unit and one more unit. The Delhi unit, which has been inactivated since 2019, has now been active. The proceeding is approved in, all proceeding will approved in the CC meeting. The member of association have also brought admiration to themselves and glory to the association by getting fellowships from the various scientific academy of the country and other prestigious awards. They have been nominated for prestigious fellowship from the BRSI, NAS, NASI, INSA, and AMI members have also nominated as the editor and academic editors in the various reputed journals. This year, MI organized various webinars, field trips for the students and training programs for the students. We also organized different units, also organized different activities for the young professionals or the young students. Over the last year, we have lost a couple of life members. For that, I request all the members to stand forward and the 1 minute more. you. AMI also congratulate Dr. Mishra and Dr. Joshi for establishing new units. And we also congratulate Anil Punia, President, AMI elect 2025, for selection as a Director of NRCC Bikaner. We aim to encourage the formation of idea and the development of creative thoughts. Researchers are giving sufficient exposure to enrich their knowledge and expand their imagination, enabling them to excel in science and technology. Our association endeavors to provide the best possible learning environment to achieve excellence in research and other fields with global perspective. All members and participants, please note that the microbiology is not the basic science. Advancement in modern biology and their application will deeply affect society, making microbiology as leading industry in the future. On behalf of the AMI and the organizing committee, I extend a warm welcome to all the participants and wish everyone a safe and comfortable experience for this ongoing conference. As we know that we are following the legacy of our senior, that is the AMI held a very successful meeting of researcher, educationists, students, technocrats around the world at their annual conference in 2025. The AMI International Conference Conclave was organized under the title Next Generation Microbiology Education, Innovation and Research for Economy, Energy and Environment, in collaboration with the Dev Bhoomi University, Uttarakhand, from 12th to 15th November 2025. The conference aimed to unite microbiologists, students, and scientists from around the world to encourage collaboration and the exchange of ideas within the global microbial research community. This year, Indian Journal of Microbiology continue to serve a vital publication platform, fostering research collaboration, interaction, and community engagement. Journal of Microbiology received 2 prestigious awards from the Springer Nature. One is the Growth Impact Award given by the Springer and Large Partnership Award. We acknowledge the contribution of all the past and present president, editor-in-chiefs, editors, and contributors of INGM for their valuable contribution that have led our journal to achieve remarkable recognition from the Springer in this prestigious category. The President, Chairman, AMSC, and other CC members released the report titled Technical Insights and Reflections on the 65th AMI International Conference and School Conclave on Prospective of Microbes for Human Welfare, hosted by the Guru Jambheshwar University of Science and Technology, Hisar, from November 14 to 17, 2024. That is under the ISBN number. The capacity building, each year AMI hold an annual general body meeting during the AMI annual conference. This year, AMI organized a conference at Dev Bhoomi University, Dehradun, in both online and offline mode, comprising 10 themes and 43 presentations. At this conference, more than 100 eminent microbiologists from India and abroad are participating in the hybrid format, with 40 from different parts of globe and 77 from India. In this conference, more than 400 people registered for the conference and workshop. The workshop were conducted under the AMSC, and the following activities were organized on the topic School Children's Activity on the eve of Children's Day. Hands-on bioinformatics workshop for genomic analysis. Patent writing to granting hands-on workshop under UCOST. Conference lead many formal discussions on the industry and academia, next generation education in microbiology, Learning Enabling Ecosystem in Higher Education of India, Biomanufacturing. We also go for the outreach activity. As a GS, I participated in the different discussions, group and discussions to educate related with the small farmers group. On the hydroponic, it is possible to, you can say, biotechnology, बिना जमीन biotechnology, हाइड्रोपोनिक वर्टिकल खेती, वैकल्पिक खेती की प्राथमिकता क्यों? पर्यावरण के अभाव में विकास मानव व प्रकृति विरोधी है. मानव संसाधन बेस्ड पलायन, marginal farmers group involved in all this, you can say, discussions group, and any other activities organized by the different units of AMI. Hisar unit organized the 65th annual conference along with the 3 workshops: microbial forensic and application, nanomaterial for drug and diagnostics applications, and research proposal writing, school children conclave. Under this, the different competition, poster, quiz competitions, and Ragoli competition organized for the school children, and they expose with the high-ended equipment related with the microbiology, slogan writing, and quiz. Bangalore unit organized the conference on recent advancement in chemical and biological sciences, sustainable future, 7-day faculty development program, skill development program on sustainable innovations in pharma, industry visit come 1-day training program on mushroom cultivation, skill enhancement program of next generation sequencing, application and data analysis, biohackathon, cooking without fire, and vegetable craving competition on World Food Day. Hyderabad unit, 1-day seminar on igniting young mind for successful future, 6-day workshop internship program on the research techniques, international conference on the Golden Jubilee of the international emerging microbial technologies for One Health, AMI Burdman unit, social outreach and community engagement programs organized, Botany Department, International Research Networking in February-March 2025 under the guidance of Professor Rajbir Bandyopadhyay, AMI Mysore units, pick and speak competition, poster presentation, program on the school children awareness program, webinar on the antifungal resistance, NSS awareness program, model making competitions and study visits for the school students. AMI Srinagar Gadwal unit organized 1-day national symposium on trend for TB, emerging pathogen and environmental management. LPU unit successfully conducted 1-day national symposium of mushroom research, mycelium to medicine. World Cancer Day, 4th February, celebrated for awareness campaigning by AMI LPU unit. And financial assistance for the AMI unit, the financial assistance amount of academic activities, AMI unit to spread the knowledge of microbiology and increase from 20,000 to 40,000 maximum with the following condition. AMI unit will take the registration fees from the participants, minimum 4 lectures per activity organized by the organizers, minimum 10 life members of the AMI come from the unit. Activity under the umbrella of AMI and AMSC only. That I propose before the GB kindly approve the same. I begin with the announcement of the award result for the year 2025. The following is the list of awardees for this year. Before proceeding with the announcement for the 2025 awardee, I request our dignitaries to felicitate our awardees. Please stand forward. So first, I will start with the Young Scientist Award. Young Scientist Award under the Agriculture Microbiology, total 3 applications received. And Dr. Shri Niketan Puranik, scientist, Indian Institute of Pulse Research, Kanpur, UP, selected for this. I request Dr. Renu to come on the desk to read the citation of Mr. Puranik.
Dr. Shri Niketan Puranik, scientist ICR, Indian Institute of Pulses Research, Uttar Pradesh, is recognized for his contributions to the field of microbiology. His research revolves around reduction, valorization, and utilization of agro-residues into compost and other value-added products. Decontamination of arable soils and deciphering the semiochemicals involved in plant microbe pest interactions. Also, he has worked on understanding the role of gut microbiome of agricultural pests in their metabolism. Having authored several publications, he is also a reviewer for reputed journals. He has been an active member of microbiological associations. He was previously conferred with DST Avsar Popular Story Award and Best Researcher Award. He steers his research towards sustainability by providing robust biological technologies in agricultural pest and disease management, thereby reducing chemical usage. Also, he aims to increase soil fertility by promoting microbial inputs. In recognition of his valuable contribution in advancement of microbe-based agricultural sciences, Dr. Shri Niketan Puranik is confirmed the AMI Young Scientist Award, Agricultural Microbiology 2025. In view of the contribution in area of molecular microbiology, which are of both fundamental and applied values, Dr. Shri Niketan Puranik's efforts are recognized by AMI in conferring him AMI Young Scientist Award, Agricultural Microbiology on 14th November 2025.
उसको पीछे कर लीजिए. Now, environmental microbiology, total 4 applications received. Dr. Sonam Palia, Alexander von Humboldt Fellow, Aachen University, Germany. Please come on the desk. And I request Ms. Shreya Maheshwari to read the citation of Sonam Palia. Where is the Sonam? She is present.
Dr. Sonam Paria, currently an Alexander von Humboldt Fellow at RWTH Aachen University, Germany, has an outstanding contribution to the field of bioremediation, hazardous waste management, and environmental sustainability. With over a decade of research experience, Dr. Palia has made pioneering advances in the biological remediation of persistent organic pollutions and biovalorization of waste. She earned her PhD from the Academy of Scientific and Innovative Research working at CSIR and EERI, when she developed microbial processes for the degradation of biomimetic flame retardations and provided valuable insights for policy frameworks on POP management in India. As a national postdoctoral fellowship and later a Humboldt Fellow, she has innovatively designed in situ remediation technologies integrating microbial and physicochemical methods to remove hazardous pollutants from wastewater, established scalable, sustainable, and eco-friendly solutions for real-world applications. Dr. Palia has published extensively in high-impact journals, including Environmental Chemistry Letters, Bioresource Technology, Journal of Cleaner Production, and other reputed QI journals with over 700 citations to her credit. She is also a prolific author and editor of international books and book series with Taylor and Francis, Springer Nature, and Elsevier, reflecting her commitment to advancing environmental biotechnology and education. Dr. Palia has made significant contributions to the field of environmental science and waste management, both at the national and international levels. Her extensive list of conferences, papers, posters, and invited talks reflects her dedication to promoting sustainable waste management practices and environmental awareness. In view of the contribution in area of environmental microbiology, which are both fundamental and applied values, Dr. Sonam Palia's efforts are recognized by AMI in conferring her AMI Young Scientist Award in Environmental Microbiology on 14 November 2025.
Industrial Microbiology, Dr. Ajay Kumar, Assistant Scientist, Department of Microbiology, College of Basic Sciences and Humanities, CCS, HAU, Hisar. I request Dr. Ranjana to come on the desk to read the citation of Mr. Ajay Kumar. थोड़ा ब्रीफ कर देना, ठीक है, और छोटा
Dr. Ajay Kumar, Assistant Scientist, Department of Microbiology, Chaudhry Charan Singh, Haryana Agriculture University, Hisar, has made remarkable contributions to the field of microbiology. Dr. Kumar obtained his Bachelor of Science in Agriculture from CCS HAU Hisar, and he qualified for ICR's All India Junior Research Fellowship in 2014 and secured All India Rank 1 in ICR's All India Senior Research Fellowship examination. During his postgraduate and doctoral research, Dr. Kumar explored innovative microbial and nanotechnological approaches to mitigate industrial and environmental challenges. His PhD research on microbes-mediated rice residue management in the rice-wheat cropping system provided sustainable alternatives to crop residue burning through microbial interventions. Dr. Kumar has been an active researcher in the field of microbiology and engaged in 2 externally funded research projects. Dr. Kumar's research findings have been published in several high-impact national and international journals, underscoring his significant contribution to the field of microbiological sciences. Beyond research, Dr. Kumar has been an inspiring teacher and mentor, guiding postgraduate and doctoral students in microbiology. In the view of contribution in the area of industrial microbiology, which are of both fundamental and applied values, Dr. Ajay Kumar's efforts are recognized by AMI in conferring him AMI Young Scientist Award, Industrial Microbiology on 14th of November 2025.
Dairy and Food Microbiology, Dr. Harmeet Singh Dhillon. I request Dr. Navnidhi Shikara to read the citation of Ms. Dr. Harmeet Singh Dhillon. He is from Patiala University, am I right? He recently joined the Patiala University, or Amritsar, sir? Amritsar, okay.
Dr. Harmeet Singh Dhillon is working as assistant professor in the Department of Microbiology at GNDU Amritsar, Punjab. He done his PhD in dairy microbiology from NDRI Karnal. His research focuses on interaction of microbial ecology and animal productivity with a particular interest in comparative metagenomics of bovine samples to explore microbial assemblies linked to the milk yield. Dr. Dhillon's academic journey reflects a dedication to bridging international fundamental microbiology with applied dairy sciences. In addition to his scientific pursuit, he is actively participating in different activities, advocating for diversity and inclusion in science. Dr. Dhillon is keen on collaborative research and aims to contribute to global initiatives addressing the food and dairy security. In view of contribution in the area of dairy and food microbiology, Dr. Dhillon's efforts are recognized by AMI in conferring him AMI Young Scientist Award in Dairy and Food Microbiology section on 14th November 2025.
Dr. Vishu Chaudhary, Assistant Professor, University Institute of Biotechnology, Chandigarh University, Punjab. Okay. Okay, she is not attending today's function. Okay, so now medical and veterinary microbiology, total 3 applications received. Successful candidate is Dr. Anish from Department of Biotechnology, Chaudhary Ranbir Singh University, Jeend. I request Dr. Nirjara to read the citation of Dr. Anish. Dr. K.S. Patel to read the citation of Dr. Patel. Read the citation of Dr. Anish.
Dr. Anish is an assistant professor in Department of Biotechnology, Chaudhary Charan Singh University, Jind, Haryana.
He attended his BSc Biotechnology in 2012, MSc in 2014 from Kurukshetra University, MDU Rohtak respectively. Subsequently, he earned a PhD Biotechnology in 2022 from the MDU Rohtak. He is awarded his PhD degree for Best Thesis Award by Honorable Vice Chancellor of India in 18th convocation during 2023. He successfully completed his PhD and served a research associate between 2020 to 2020 in ICR. He developed various technology in the molecular biology field like DNA, RNA isolation, ELISA, PCR and RT-PCR and many other for the Mycobacterium tuberculosis and diagnostic. He has accumulated 327 total citations with H index of 9, I-10 of 9. His research expertise include molecular technique and diagnostic method. He is a good communicator, adept at handling multiple projects and developed scientific proposal and writing grant both independently and a part of a team. Dr. Anis is a dedicated researcher with expertise in developing innovative diagnostic technique, and he made valuable contribution to field of microbiology and TB research. In view of contribution in area of medical and veterinary microbiology, which are the both fundamental and applied value, Dr. Anis' efforts are recognized by AMI and conferring him the AMI Young Scientist Award in area of medical veterinary microbiology on 24th November 2025. So we congratulate him. Thank you.
Molecular Microbiology and Biotechnology, Dr. Utkarsh Sood. Assistant Professor, Department of Zoology, Kirori College, University of Delhi. I request Dr. Souvik Kumar to read the citation of Dr. Utkarsh Sood.
Dr. Utkarsh Sood, Assistant Professor in the Department of Zoology, University of Delhi, is recognized for his outstanding contribution to molecular microbiology, genomics, and microbial biotechnology. His research spans microbial systematics, comparative genomics, and genome-based reclassification of bacterial taxa, significantly enhancing our understanding of microbial diversity and evolution. He has authored over 40 publications in reputed international journals, including M-Systems, Frontier in Microbiology, Microbial Biotechnology, and Journal of Natural Products, and contributed 3 genus chapters to Berge Manual of Systematic of Archaea and Bacteria. Dr. Sood's work bridges molecular microbiology and biotechnology, integrating comparative genomics, bioinformatics, and microbial ecology to address environmental and biomedical challenges. As an educator and resource person, he has led numerous national and international workshops promoting computational biology and microbial literacy. He also served on the editorial board of ISME Communications, communicating to the disseminations of cutting-edge microbial ecology research. In recognition of his impactful research, academic excellence, and commitment to advancing microbiological sciences, Dr. Utkarsh Sood is conferred the AMI Young Scientist Award, Molecular Microbiology 2025. In view of the contribution in the area of molecular microbiology, which are of both fundamental and applied values, Dr. Utkarsh Sood's efforts are recognized by AMI in conferring him AMI Young Scientist Award, Molecular Microbiology on 14th November 2025. Thank you.
Now the Professor B.N. Johari Award. Professor B.N. Johari Award, total 3 applications received. And Dr. Shilpi Sharma awarded the Professor B.N. Johari Award. She is Professor, Department of Biochemical Engineering, Biotechnology, Indian Institute of Technology, New Delhi. I request Dr. Nabil to read the citation of Dr. Shilpi Sharma. I request Dr. Anil Kumar to read the citation of Dr. Shilpi Sharma.
It's a pleasure to read the citation of Dr. Shilpi Sharma. Shilpi Sharma is a chair professor at the Department of Biochemical Engineering and Biotechnology at the Indian Institute of Technology, New Delhi. An alumnus of the Department of Botany, BHU, she completed her PhD from Ludwig Maximilian University, Munich, Germany, followed by the postdoc at Helmholtz Zentrum, Munich. She is an environmental microbiologist working in the area of the plant-microbe interaction for attaining food security and agriculture sustainability. She focuses on tailoring the plant microbiome for stress mitigation and development of next generation bioformulations. She has authored more than 120 international publications and 20 chapters. She has been bestowed upon several coveted awards, including the Young Scientist Award by AMI India, Innovative Young Scientist Award by Asian PG PR Society, Women Scientist Award by the Indian Botanical Society, and many more. She featured in She is a 75 Women in STEAM, a joint honor conferred by the Government of India and British High Commission to commemorate 75 years of independence. She is the inaugural winner of the Tata Transformation Prize in the Food Security 2023. In view of the contribution in area of microbial diversity, which are of the both fundamental and applied values, Dr. Shilpi Sharma's efforts are recognized by AMI in conferring her AMI Professor B.N. Johari Award on 24th November 2025.
This year we also announced Professor G.B. Manjrekar Award to 2 applications received, but none of them found suitable for the award. Professor J.B. Bhatt Award we awarded for the best citation of articles in the field of research articles and the review articles. Muhammad Abrar Yusuf, Nagarjun Prakash Dalbanj, I think none of them. S.K. Praveen Kumar, none of the candidates are present, so we can move further. The Best Unit Award, the total 3 units submitted their report, and that we awarded the Best Unit Award first, second, and third. The Best Unit Award, third prize, to the AMI Hyderabad unit. Anyone from the Hyderabad unit? Please come on the dash. Hyderabad unit. Check bhi kaat diya. So we give the 3,000. No, kitna 3,000. So along with the certificate, we awarded 3,000 rupees cash prize also. Yes, come on. ऐसे कहते कोई बात नहीं सर ऐसे ही दे दें सर तुम्हारे आगे नहीं And second prize, the AMI Bangalore unit. How much? 5,000, no sir, वो online transfer करेंगे. 5,000 rupees and the certificate. सारे बच्चे आ जाना, बोल दो, सारे बच्चे आ जाना, सारे बच्चे आ जाना. Award money sir, online. अपना गैंग तो बुला दो, कब ना है अपना गैंग. Now the first prize for the best unit award is Hisar unit. All the Hisar unit members come on the desk. Come. आ जाओ. जल्दी आओ, Dini. And along with the certificate, 7,000 cash prize also. Want to them. जल्दी जाओ बच्चों.
Congratulations, Dr. Saab.
आवाज आ रही है?
I think AMSc fellow already awarded during the inaugural function. I will just read the name of the award. नहीं बस ये अमित वगैरह बता देते तो. हाँ जी. That Dr. Amita Gupta from Department of Biochemistry, University of Delhi, South Campus, New Delhi. Dr. Naveen Gupta, Department of Microbiology. Sir, खड़े हो जाइए. Naveen Gupta, Department of Microbiology, Punjab University, Chandigarh. Professor Naveen K. Arora, Professor, Department of Environmental Sciences, Baba Saheb Bhimrao Ambedkar University, Lucknow. Dr. Ram Prasad, Associate Professor, Department of Botany, Mahatma Gandhi Central University, Motihari, Bihar. Professor Surjit Das, Department of Life Sciences, National Institute of Technology, Rourkela. The CC authorized for the Student Travel Award, CC authorized the AMI General Secretary to scrutinize the grant for the student travel grant to attend the AMI conference as per AMI rules and regulation after the conference. I congratulate all the awardees on their success and hard work, and I also request that all AMI unit office bearer submit the report well in time so they can also include it during the evaluation process. I propose before the GB kindly approve the same. Now the Rana Memorial Best Poster Award awarded in the 2024 conference to Ms. Renuka and Malvika Sehgal Memorial Best Poster awarded in 2024 65th conference, Dr. Manvika Sehgal to the Lucky Duhan, Department of Biotechnology, MDU Rohtak. I propose before the GB kindly approve the same. Propose activities which are approved by the CC. AMI organized following activity under the AMI MSc flagship. AMI organized the International Annual Conference at Dev Bhoomi University, Uttarakhand, which was Dr. N. Vasudevan submitted some Southern Zone one-day conference or seminar. GS proposed one workshop on startups at Hisar unit. CC plan list one, all the CC members requested to submit at least one activity at their own institute, and it is most important before the March their term completed, they have to submit one activity and organize at their institutional level. I propose that before the GB kindly approve the same. The confirmation of 65th annual conference GB meeting proceeding was held in 65th International Conference, circulated during the conference. I propose before the GB to kindly approve the same. The detailed report I am proposing, that is the 6 meetings held during the year, AMSC and AMI. We signed an MOU because from the very next year in the 2026, instead of 4 volume, our journal will publish 6 volume a year. I propose that before the GB kindly approve the same. One new journal proposed for the comprehensive review in microbiology under AMI MSc was proposed and submitted to the Springer as per the approval of the 65th GB meeting. I propose before the GB to kindly approve the same. As per the CC recommendation, an online-offline editorial meeting was held on 11th November 2025 at the Effortal Hotel of Dehradun. The EIC will present the meeting proceeding after I complete you can say report. I propose before the GB to kindly approve the same. Now the contribution of academic contribution of AMI. During the session 24-25, AMI and AMSC organized different activities. Instead of AMI life member, that was approved by the CC, that all the CC members and all the life members who are interested to propose any book proposal, we are ready to process their book proposal through our association. And if they are interested, they can submit to, a proposal may be submitted to the GS office, and that will be approved by the CC. And if CC feels that it is suitable to process further for the textbook or textbooks or the book series or any of the edited book, we will definitely help them for the publication. Under the AMSC academic proposal, Chairman AMSC proposed a lecture series on the different fields of microbiology, and all the CC members were requested to submit the potential speakers list so that final schedule may be released. So this is my humble request to all the life members, all the members who are present here in this hall, may submit their, may propose any of the person who is potential to and ready to deliver a lecture, online lecture, and that lecture will be uploaded on the our association's website. And action taken report related with the book proposal. Me, Dr. Anil Panghal, Professor R.C. Kohat, and Mandeep Kumar, we propose one book on symbiotics, that 2 volumes in process. It is completed very soon. Within a month, we will release with the name of AMI and AMSC. The revolutionizing health and disease management, and another harnessing probiotic and prebiotic for nutrition. I propose that GB kindly approve the same. The 3-member committee, including GS, President and Chairman AMSC, and President and 4-member, President-elect Dr. Anil Punia, was constituted to finalize the rules and regulation of AMI and AMSC, because during our process of election process, we find out some discrepancies in the rules and regulation. So immediately proposed by the President that we have to be modify or include all these, remove all these discrepancies, and that will be very soon. We will finalize rules and regulation. I propose before the GB to kindly approve the same. Related with the inactive members, CC members, if any officiating CC member does not attend 2 consecutive meetings without a valid reason and does not deliver for the improvement of the AMI, they should be replaced by new active members for the benefit of the association on the recommendation of the CC. I propose before the GB to kindly approve the same. AMI membership drive related decision taken by the CC. CC informed that House that to initiate the membership drive, the Treasurer will issue the receipt book for annual and life membership to the CC members. The CC member will update the progress of their respective zone to CC in periodic CC meetings. The organizers of the 66th AMI annual conference will also include the membership drive with conference registration. I propose before the GB to kindly approve the same. GS to inform the House that a few FMSC fellows have requested new certificate. CC discussed that AMSC should issue the new certificate to the fellow members, subject to the submission of the AMSC membership fees, which is 5,000 rupees. So I request if whosoever present an AMSC fellow kindly deposit the fees of your membership as a AMSC fellow. I propose before the GB to kindly approve the same. GS informed that House that CC, Thorough and GC office should compile the minutes of meetings and as action taken report of last 10 years, and that CC should revisit annually, and we will find out the whatever the resolution passed by the previous meetings should incorporate it during the updation of our rules and regulation. I propose before the GB to kindly approve the same. AMSC activities proposed during the AMSC meeting सर प्रिंटर काम नहीं किया, हमें यहाँ निकालनी थी इसलिए और सीसी proposed activity सर resolutions इसमें दिया है ना, सीसी मीटिंग अटैच नहीं की है. We will provide you. We will give. Renu Gangi, I will give you, sir, don't worry. AMSC, Dr. Amulya Panda, President and the AMSC Executive Member, commended the societal contribution of AMSC, highlighting several key initiatives, including the syllabus benchmarking to maintain academic standards, efforts to promote microbiology education at the school level, engaging young microbiologists in writing books on microbiology, enhanced press coverage of AMI and MSc activity, active involvement of young researchers in initiatives aimed at the uplifting of the microbiology. Several expert lectures online and offline should be organized, organizing different activities at the unit level and then CC members. Recently, one book is already published in collaboration with our association with the help of Springer, the Plant Microbe Interaction for Climatic Resiliency Agriculture. This is, okay. This one, okay. So now I am going to present the AMI-MSc minutes of the meetings. The meetings of the Executive Council of Academy of Microbiological Sciences was convened on the 15 hours 16th October 2025. The following members participated in the meeting via online mode. Professor R.C. Kohar, Chairman, Professor Prince Sharma, President AMI, Professor Namita Singh, General Secretary, Professor Naveen Khanna, Member, Professor P. Gunashekharan, Member, Professor Bhagyaraj Joseph, Member, Professor Rooplal, Member, Professor A.K. Panda, Member, Vijay Kumar Chaudhary, Member, Professor Sarman Singh, Member, Professor Praveen Rishi, Professor Surjit Singh Dudeja, Professor R.C. Sobati, Professor Siba Samba Rao, Member. Professor Sunil Khare, Professor Jagaved Agrewala and Professor Yogender Singh could not attend the meeting. The chairman welcomed the members and briefed about the various program and activity organized so far and shared the list of events to be organized in the time to come, which are as follows. The Academy has already begun its program to popularize microbiological sciences in a school and college in different parts of country. However, this plan needs to be strengthened and extended in the near future. The members suggested that experts should prepare themselves in such a way so that the school students gain clarity about microbiology and their applications. Furthermore, few members suggested that they may strive to make and public awareness about both harmful and beneficial aspects of microbes through newspapers, social media or outreach programming involving UG, PG and PhD students. The matter about bringing reforms in the evaluation process of the nomination of the fellows of Academy was discussed. The members suggested to constitute a committee to work on developing a methodology to elect fellow and authorize the chairman to do the needful. The following committee has been constituted: Professor Naveen Khanna, Professor P. Gunashekharan, Professor Vijay Chaudhary, Professor Praveen Rishi, Professor Anil K. Punia, It was also decided to initiate an expert lecture series in order to develop a policy development and Executive Council of the Academy constitute a committee under the chairmanship of Professor P. Gunashekharan, member Executive Council AMSC. The committee has submitted its report, identified thrust area of research in microbiology for which we can start lecture series under our program Microbiology Connect. The committee also suggested to identify the expert from different specialized area using a performer to initiate willingness to deliver the lecture online to the General Secretary AMI. It was further resolved that the lecture calendar annual may be prepared and circulate among members of prior information. The Academy also developed a system of providing remuneration for the expert lecture online. Whenever lecture is to be organized offline, we must keep a provision of TA and honorarium. A committee of following members has been constituted to formulate such provision: Professor Prince Sharma, Chairman; Professor Sarman Singh, Member; Professor Namita Singh, Member, Convener; Professor Hari Shankar Mishra, Distinguished Professor, Biological Sciences, NIMS, Mumbai. The matter of implementation of NEP 2020 was also briefly discussed among members, and it was informed that the Academy and AMI had jointly taken up the responsibility to develop and benchmarking the curricula of UG PG microbiology aligning with the NEP 2020 and UGC credit framework. The members were informed that this exercise is almost in its final phase of completion. Hopefully, we will be able to bring out the full curriculum by our next meeting. Accordingly, members also discussed that the Academy should take up the matter to facilitate UG PG student of microbiological sciences to receive research internship opportunity in universities, industries, and higher educational institutes. The committee resolved that a full-fledged plan be developed in tune with the process being followed by other National Science Academy in order to develop the procedure or planning. The committee has been constituted. Professor Naveen Khanna, Group Leader, IGIB, New Delhi Chairman; Professor Amita Gupta, Head Department of Biochemistry, University of Delhi, South Campus; Professor Rooplal, Hansraj College, University of Delhi, Member. Finalization of result about selecting Fellow of Academy of Microbiological Sciences, AMSC. The Chairman presented the evaluation report based on the making received from different experts for their observations and discussions. After a healthy discussion, the member unanimously decided upon the 70% cutoff for selection of Fellow of AMSC. Some members observed some discrepancy in case of Dr. Ram Prasad, Department of Botany, Center, University of Bihar, Motihari, and authorized the chairperson to ask him for his full publication record and submit to the expert concerned in order to complete his re-evaluation. The final result may be notified after receiving the evaluation. Consequently, the following candidatures were approved for the award of Fellowship of Academy of Microbiological Sciences: Professor Amita Gupta, Department of Biochemistry; Professor Naveen Kumar Gupta, Department of Microbiology, Punjab University, Chandigarh; Professor Naveen K. Arora, Department of Environmental Sciences, B.R. Ambedkar University, Lucknow; Dr. Ram Prasad, Department of Botany, Central University of Bihar, Motihari; Professor Sujit Das, Department of Life Sciences, NIT Rourkela, Odisha. The meeting came to close with formal vote of thanks to the chair. That proposal also submitted an inviting academic ambassadors, one from each state, so that then organizing to conclave to promote the innovative idea, research startup and interdisciplinary research, one in North India, Eastern India, South India and the Central India. Proposal to be submitted, rupees 5 lakh each conclave. I submitted to GB, kindly approve. Initiating a scheme of AMI and MSc to facilitate PG student of microbiology for research and internship. Scheme to train younger professionals, researcher and research methodology. I submitted this proposal to the GB, kindly approved. To initiate all these AMSc and AMI activity, we need a permanent office, and then permanent office for these purposes taken on rent in Delhi NCR region, because from the last 5-6 years, every time we approved for the permanent office. So finally this time we finalized one office at NCR region of Delhi. So I submitted this is to GB to approved, please. Now I request EIC to present the report. मैडम आप ब्रीफिंग कर दीजिए, ब्रीफिंग रिलेटेड विद द EIC मीटिंग। ट्रेजरर की ट्रेजरर है नहीं, वो उनकी मैं पढ़ दूँ? Okay. So now I am presenting the treasurer report. As AMI treasurer, I hereby inform that AMI is presently maintaining 2 accounts in different banks. The saving account with Punjab National Bank, 2 crores 21 lakhs 5,762 rupees and 43 paisa. While the current account with the State Bank of India holding 64 lakhs 54,067 rupees and 50 paisa. Accordingly, the total funds available in both accounts, that is 2 crore 85 lakhs 59,829 rupees and 93 paisa as on 6/11/2025. Additionally, MI has one fixed deposit with PNB value 1 crore 62,696. With regard to membership, MI has making continuous efforts to enhance the strengthen the both annual and life member category. During the period from April to November 2025, 48 annual member and 64 life members and 5 AMSc fellow registration taken place. Consequently, the total number of active life members now stand for 5,683. The long pending issue pertaining to the AMI Pune unit since 2019 has been successfully resolved. The Pune unit has remitted a check amount 1,37,670 rupees and 18 paisa in favor of the Treasurer Association of Microbiologists of India for further necessary action and utilization. So I really congratulate.
Madam, actually in table this is not shown. The AMI has received from Pune unit. That amount is not reflected in the table.
सर वो अभी अकाउंट में नहीं आया, इसलिए नहीं आया है, अभी 4 या 5 दिन पहले ही आया।
Okay.
That's why. हाँ. Regarding Jhansi conference, I had a talk with informed that she has a talk with Dr. Rishi Saxena. He is not denying for taking 2 lakhs rupees only for organizing, but he was not aware that he was to return them secondly. He was saying that there is no written document where he had signed this as an agreement. Still, I asked him to solve this matter because we give the 2 lakhs rupees as a loan amount, but he is denying to pay back to the association. Although he was very much aware that this is amount is the loan amount, he was present in these CC meetings where this amount sanctioned, and his sign is in our register also present, but we are trying to get back from him our best. Yes, although he is not ready to submit the bill for the 2 lakhs also. So we are facing problem how to adjust this 2 lakhs rupees as an you can say account for. So I submit that what the suggestion from the GB. What can you just inform? Expenditure statement. Total amount is yes. Transfer of the conference earning to AMSC amount. The matter of financial support to AMSC was discussed, and the earlier CC decision was retained that CC advised that the transfer of saving from the AMI annual conference to AMSC should be regularized and followed in the future. Moreover, Professor Kohad reminded us about the decision of CC and GBAMI, as also presented the black and white in the rules and regulation of AMSC that 20% royalty from the INGM will be transferred till it becomes self-sufficient. PAN of AMSC, it was proposed to obtain a separate PAN in AMSC name to maintain a distinct financial account for AMSC activity. This measure is intended to streamline financial management and optimize tax reliability. Liability matter will be discussed in the following physical meeting. I propose before the GB to kindly approve the same. ये तो सर हो चुका है. हाँ. This is from my side. Yes. Okay, the proposal submitted for the 67th Annual International Conference. The following proposal were received for organizing the 67th Annual Conference of AMI from the C.G. Bhakta Institute of Biotechnology, Ukta Takshila University, Surat, Gujarat, Godwal University, a Central University, Assam Agriculture University, Jorhat, Assam. DRG Jhansi Road, Gwalior, Sunandan Dwivedi School of Science, Nims, Mumbai. The above proposal were received and evaluated by CC as per the rules and regulations, but only the eligibility criteria were fulfilled. That CG Bhakta is not eligible because it doesn't have any AMI unit. Hemvanti Nandan Bahuguna University eligible for the organizing the conference. Assam Agriculture University, Jorhat eligible as a proposal submitted for the establishment of Assam local chapter unit on 28/10/25. DRG Gwalior Jhansi not eligible as it does not have any AMI unit. Sunanda Divetia School of Sciences eligible as the proposal submitted for the initiation of the Mumbai chapter revival on 16th November 2025. The CC feels as the Dr. Mishra from the Mumbai, he said that they can cater only 300 strength in their organization to organize an international event. Okay, so We finally approved the Assam Agriculture University, Jorhat. I propose before the GB to kindly approve the same. Any? Yes, you can. You want to say you can? मैंने बोल दिया ना 60 1 से लेकर 60 तक everything approved that we have to take the permanent okay, here I have all the correspondence with me. Okay. The conference is expected to host approximately 300 participants, including 10-20 foreign delegates from Asia, Europe, and America. Okay. The scientific program will feature planning, lecture, invited talks, oral and poster presentation, and interactive session designed to foster collaboration and knowledge exchange. So, this is the decision of CC. If CC feels, then it's okay, it's fine. You can.
Yes, Mr. Mishra, we received your proposal and we had a long discussion. Even day before. Hello. Last, say before, like Monday before, we discussed and we wanted very much, but the genesis, say, which emerged, that after a very long time we have got one proposal from Northeast. We have been trying to get some kind of the activities to organize in Northeast because we want that Northeast people should also come forward and join Association Microbiologists of India. They have been fulfilling, say, as far as the application is concerned, all those conditions. So Central Council resolved that the first preference they should give, and they will be given, say, a couple of months' time. And if we feel, even the committee will go and will take a look. If we feel that, yes, they would be able to, I think, organize it successfully, or with the interest they are coming forward. So it has been, say, approved in the favor of Agriculture University, Assam, Jorhat. But your interest is very right. I have a suggestion here before House, because just now we have got approved proposal of 2 conclave, specifically to motivate people for innovation, research, startup. So I think one of the conclave, you can give the new proposal. One of the conclave you can have, say, in Bombay. As far as time is concerned, Central Council again will have a look on it. And what time, say, Jorhat people would like to organize it in the coming year. Certainly we'll find out the way, and both will have the different objectives. Because that will be a subject conference, and here it will be a specific kind of conference where you can, say, add maybe one day more program along with the conclave. So
okay,
that kind of
the
permutation combination.
CC and Council of AMI, I'll respect the institution since Council has taken a call that Jorhat will organize, so I accept it, let Jorhat organize it. However, Mumbai chapter is the oldest AMI chapter, along with the Lucknow and rest of the country, has got nearly 400 members. The chapter was not active for a long time, I fully agree. After Namita has mentioned to me that it should be revived, we have actually activated. Now here is a merit in the discussion point. There is a chapter which has been just created. Please, Jorhat people should not mind it that I am contending against them. I am only putting the logic. Here is a chapter which has been just constituted, and here is a chapter which has been existing for years. Mumbai region also and Maharashtra never had a AMI at least for last 30 years, which I know that. So there is a merit and there
Professor
is a...
Mishra, with all considerations, because sometimes we have to, because we always try that one time this conference goes to the south, one time it goes to the north. But there have been occasions where, say, even continuously 3 times, say, it has been in north. But there is an attempt by Central Council that we should also have, say, some conferences elsewhere as well. Northeast has been, say, I think maybe in the years sometimes 1998, if I'm not wrong, we had in Guwahati. And since then, you can say 27 years. And we have made the efforts so seriously, we could not get even a single Central Council member from Northeast. We had to nominate, say, some representative from them. So I think
हैदराबाद वाली दिया ना सर को बुला के दे देंगे वो हैदराबाद वाली वहाँ से उठा के सर आ गए हैदराबाद से सर आ गए ना हैदराबाद यूनिट से उनको बुला के देते ना
if I can add something to this, I have just an idea came that but it has never happened before. Isn't it possible to have 2 AMIs 1 year? Why not? I propose it, and if people think that it's possible, and for the organizers to do that, why not do one? If this situation has arisen and there are more applicants, yeah.
Sir, I respect a lot on the institutions, and this council is one of the institution of AMI. Whatsoever decision has been taken, I will not like to contend it. I was only putting the logic. Please accept that way. If the council gives me an instruction that you can conduct in 2026, I will do that. If it gives the instruction you conduct in 2027, I will do that. If council decides no Mumbai chapter should not have an annual meeting, I will respect the decision and keep doing all
See, you are not getting it actually.
Mishra ji, you are taking, I think, in a very different direction.
What?
देखिए, हमने आपके सामने एक अर्ज के साथ एक वो रीजन रखा, लेकिन you have felt it, I think, in a different way. ऐसा नहीं होता है. देखिए, एक ट्रेडिशन होती है किसी भी ऑर्गेनाइजेशन की. उस ट्रेडिशन में हम आपको भी चाहते हैं कि आप भी शामिल हों. मैंने पहले ही कह दिया कि we wanted very much, लेकिन हमारे सामने भी तो एक जो है ना कोई ऐसी चीजें आती हैं जिसको हमें देखना पड़ता है. उसका जवाब हम दे नहीं पाएंगे. अगर मेरे आपको ये बताया, मैंने यही कहा दूसरी एमआई कॉन्फ्रेंस की बात करें, और आपके सामने एकदम हमने आप लोगों से अभी अप्रूव कराया कि हम 2 कॉन्क्लेव करेंगे, एक नॉर्थ इंडिया और नॉर्थ ईस्टर्न रीजन में, एक सेंट्रल और साउथ इंडिया में. तो हम आपको एक कॉन्क्लेव के लिए बात कर रहे हैं. तो कॉन्क्लेव इज आल्सो लाइक ए कॉन्फ्रेंस. और इस कॉन्क्लेव में हम इमीडिएटली आपको जो है ना 5 लाख रुपए भी देंगे.
करेंगे.
हमने तो आपके लिए एक प्रोविजन बनाया अलग से. अब बात आती है कि क्या हम 2 AMI कर सकते हैं कि नहीं. अभी तक इस तरह से ट्रेंड रहा है, करने को 4 AMI कर लो. एक मिनट बात पूरी होने दीजिए. करने को 4 AMI कर लीजिए, हमें कोई दिक्कत नहीं. ऑर्गेनाइज आपको करना है, फाइनेंस आपको ऑर्गेनाइज करना है, लेकिन एक ये होता है कि नंबर ऑफ पार्टिसिपेंट्स जो होते हैं वो ऑलमोस्ट वही होते हैं, उनको चाहे बॉम्बे ले जाएं हम, चाहे नॉर्थ ईस्ट ले जाएं। अब बात ये आती है कि अगर यू आर सो कीन एंड वी आर आल्सो रेडी टू सपोर्ट यू, तो आप अगर 2027 फरवरी में करें तो हमारी 2027 की जो रेगुलर कॉन्फ्रेंसेस होती हैं, हाँ वो उनको कह देंगे हम कि वो नवंबर की जगह, ये डिपेंड करेगा कि वो सितंबर में कई बार वेदर बड़ा ऑड रहता है, तो क्या अक्टूबर में कर सकते हैं वो हम उनसे बात कर लेंगे। उसी हिसाब से आपको जो है ना, अगर वो अक्टूबर में कर लेंगे तो आप फरवरी में कर लीजिए, 4 महीने का टाइम रह जाता है। इन केस, हाँ इन केस वो ये कहें कि हमारा तो नवंबर में है तो मार्च में कर लीजिए, इतनी बात है बस।
ठीक है सर, मैं आपके मतलब मैं जब काउंसिल का रेफरेंस किया, इंस्टीट्यूशन का रेस्पेक्ट किया, तो उसमें मैं एग्रीमेंट दिखाया है। मैं बिल्कुल कंटेंट नहीं कर रहा हूँ, मुझे जरा सा भी खराब नहीं लग रहा है, मेरे वे ऑफ टॉकिंग में भी लिटिल हर्टिंग टू यू। तो पहले
हमको खराब लग रहा था ना कि
मैंने पॉइंट, मेरा जो ओपिनियन था, मेरा जो स्टाइल था, वो एकदम उसी कंकरेंस में है जो आप मुझे सजेस्ट कर रहे हैं। आई फुली एग्री सर। मैं मैं एक सेंटेंस बोल देने दीजिए सर। देखिए, मैं AMI ऑर्गेनाइज करने के लिए मुंबई में, when we two came here, we must have done lot of underground work, so much popularization to the people. I am someone, I mean, if I am given the chance, you will see what kind of meeting I am organizing, how precise is that. I am actually have done lot of international meetings organized by the other societies. I am not contending, bringing out all the point. तो सर मैं बहुत सारा वर्क करके आया हूँ, इतना पॉपुलराइज कर चुका हूँ पूरे रीजन को कि वी आर गोइंग टू कंडक्ट द मीटिंग। तो इससे हो क्या रहा है, थोड़ा सा फीलिंग तो हो ही रहा है कि आई एम नॉट गेटिंग द फीलिंग, बट वो मैं एक्सेप्ट करता हूँ। आप जो भी सजेशन आप करेंगे, मुझे देंगे, या मतलब AMI देगी आपकी सेंट्रल ऑफिस, मैं उसको करने के लिए बिल्कुल तैयार हूँ। आप मुझे फरवरी 2027 दीजिए, आई एम विलिंग टू दैट। अगर आप मुझे बोलते हैं फरवरी 27 में हम अक्टूबर नवंबर में करेंगे, हम उसको भी करेंगे, कोशिश करेंगे। अगर मैं नहीं रहूँगा तो आई हैव गॉट अनदर स्ट्रॉन्ग मैन विद मी।
आई रियली अप्रिशिएट। लेकिन मैं आपको ये भी बताता हूँ, देखिए आपके जो है ना प्रिलिमिनरी, मिश्रा जी, अरे मिश्रा जी बात सुनिए, आपके प्रिलिमिनरी प्रपोजल को भी सेंट्रल काउंसिल ने ऑनर किया और ये प्लान भी हो गई कि चलिए बॉम्बे जाके चलते हैं, वहाँ भी देखते हैं। क्योंकि देखिए आपका भी प्राइवेट इंस्टीट्यूशन और ये जिसमें बैठे ये भी प्राइवेट इंस्टीट्यूशन था। यहाँ से हमने कुछ अपनी ऑब्जर्वेशन ली हैं, उन ऑब्जर्वेशंस को हम आपके सामने जरूर रखेंगे और ये सब कुछ उन सब्जेक्ट टू दोज कंडीशंस होगा। ठीक है ना? तो टेंटेटिवली फॉर 2027 your proposal is considered, and I'm submitting it to the House as well, that if you could give approval, then Central Council in the next meeting can say work on it.
Actually, if I can add, in the CC meeting, we also had this thing that in the Jorhat, it is still to be approved because some condition was there which they have to fulfill. If they fulfill, again, I reiterate that September can be given to this and February, March can be given to the you people.
So you keep your
Yeah.
keep yourself ready. What the president says that some condition is there, so you have to be ready. Or maybe if November is there, you have to be ready for that.
Yeah. If by chance Jorhat people it doesn't happen for some reason, then in November you are ready, or in February you are ready, or March ready. Yes. Probability. Yeah.
Just to add, in fact, I mean, all four proposals they were put before TC, and we had certain reasons because of which we have chosen Jorhat.
And as we are so stressing, in fact, I must place on record that if it's possible, you just could have given the reason that we already done the whatever the homework you have done, work for planning this workshop. So even now, I request, I mean, if you have all this valid reason, just place it before the CC, and we'll take a care actually already. This is projected. Yeah.
I request Dr. Bhima, please come on the desk. मैडम आप आ जाइए एक बार, थोड़ा सा ब्रीफ तो कर दीजिए आप एडिटर इन चीफ. मेरे पास आई नहीं है.
Okay, sure. Yeah, you are here. You'll be here tomorrow. Okay, no problems.
So, fine. Look, actually, the CC also approved the conference proposal format. I prepared it a little bit large, but if we use that format, I don't think so. That incompetent unit can fill the form. Okay. So, automatically the competition will be very healthy if they fill the form in a proper manner for the conference organization. Okay. Yes, definitely, we will fix. Yes, you can. So, there is some issue.
All the speakers have been arranged by AMI, and they are, because of that platform we are coming, not for the private institutions or any organization. Thank you.
Thank you. We will take care of this issue, madam. Any other?
Madam, good evening. I think it would have been better to circulate in all the previous meeting, before you were presenting the things. It would be better to circulate all those things. Then it could have been easily, you could have been looked into it actually. Okay. Because the last time also, I think you have circulated. The previous time also it was circulated. Now when you are telling, we are not having anything to refer actually. That is the thing.
Sir, what we discuss about this private organization, we are facing some problem for the printing, and nearby we are not getting any photocopy machine also, na? Okay, so we will.
I am talking about the proceedings, madam. Actually,
yeah,
just
yeah,
now,
I
am talking about the proceedings.
Only that I am able to only take the print out only 5-6 copies.
I think it is soft copy would have been there in our group itself. We could have been looked into actually.
Definitely.
If it would have been put in the WhatsApp group or it would have been good actually. That is what I was mean.
Okay.
WhatsApp group is more universal kind of a thing. As President has suggested, it will be put on our website, AMI website.
Okay, sir. Okay.
तो फोटोकॉपी नहीं अवेलेबल हुआ सर, हमें यहाँ पे नियर बाय कोई है भी नहीं जहाँ पे हम जाके करा दें. But no connectivity available here to use the. This is the limitation. Just why I was thinking that they can connect and I can display the whole meeting.
Yeah, excuse me, madam. Regarding the traveling for the invited speaker, it was, I think, in anticipation. We came here that traveling will be reimbursed. But here I I could know that traveling they will be not considering. So if it would have been like this, so we would have taken prior permission from our institute for the traveling. This will be embracing situation for us. So AMI, I think, has to consider regarding the traveling expenditure to reimburse for the invited speaker. Otherwise, the individual will come according to their own plan. Regarding this traveling expenditure,
They must have mentioned in your invitation letter
yeah,
that they will provide you DA.
all the expenditure and hospitality they wrote, they will be taken care.
I think they will give if they have committed, they have written, they will give.
No, no, they may deny like that Rishi Saxena.
Sir, actually, that's why we propose that whatever the sponsorship comes from the private sector, it should be come to the AMI account, because we are not gaining a single penny from this organization.
No, but it should be clear actually to speakers or the invited
Sir,
speakers.
actually, this is the responsibility of the organizer, because we are not gaining any single penny.
But
I
organizer was talking that everything was again and again decided by the CC member only. So they are talking something different.
They are inviting you.
Accordingly, you can make plans.
The invitation
to your institute to reimburse.
No, that is not problem, but the prior permission is required.
Sir, this is the reason why we deny Dr. Mishra.
डॉक्टर साहब आपने तो ऑर्गेनाइज किया हुआ है, आपको पता है कैसे होता है.
नहीं नहीं, वो तो ठीक है, लेकिन इस तरह के कंफ्यूजिंग स्टेटमेंट या कंफ्यूजिंग सर्कुलर नहीं जाना चाहिए. That is
Sir, that circular not, you can say, submitted by us or the sent by us.
the letter for the invitation which we received.
From the organizer.
Yeah, that is written.
Not from us.
So,
I organize 65th, I reimburse all.
yeah, you reimburse, and even if you ask again, even after a few months back, you repeatedly ask regarding the reimbursement from all the speaker.
Because it takes little bit long time.
So, in anticipation of the same pattern, we thought it will be done. तो ये तो मजाक हो जाएगा.
No, you cannot anticipate every time that if it had
अरे
been
नहीं
previously.
sir, this is a reputed society. This is not a joke. मतलब हर बार हर रूल बदलता रहेगा ये
It's
तो.
not, sir, the society which is changing the rules. It is the organizers who have to do. They may do, they may not do. You have to clarify it before coming. I think.
Once they are inviting or they are writing, it is suppose they are honoring their word. If you are not honoring your word, it means your ethics. Suppose the Jhansi people, they are saying that I am not aware about the return of the loan.
No, they have written it, they have signed it.
And I have faced the music regarding the JNU Atul was Johari also.
Everything was, we got everything.
When I was in CC.
Yes, we got everything.
So for 3-4 years he was rejecting, escaping himself.
But ultimately we got everything.
You cannot get escaped.
Sooner or later.
हाँ.
So I think it is request from my side, the CC and AMI has to consider the situation of the speaker, and the organizer also should be clear regarding their communication or invitation.
In future, we will tell the organizers that if they are inviting, it should be clarified.
Yeah.
Sure. Well
So,
taken, sir.
this should not be. मैडम वही बात तो हो रहा है, आप बोलेंगे ही. Thank you, sir. Please, you, sir. Okay.
So now the time to felicitate our outgoing CC members. Sean. Women first. Yes, ladies first.
दोबारा भी आ सकती हैं आप। अच्छा ठंडी लगेगी।
Now, thank you to all life members, annual members, and AMI family.
Thank you, everyone. Ma'am, it is on that side. I request all the dignitaries to please join us for the dinner at the SOHMT restaurant, and the faculty and the students, if somebody is pending who has their dinner, they can also join us in the respective areas. All the dignitaries, the senior members, I can request you to please join us for the dinner in the hotel management restaurant. Thank you.
म्यूट कर दे